Okay, I think hope you can hear me. Very good morning for everyone, right? Can you hear me? Right? Can you hear me? Just drop a message on YouTube Live, or you can drop a message in discussion group, right? Okay. So I will post the link of Telegram discussion group in the YouTube Live chat. You can get it there, right? Okay. Now today the entire day will be for English medium students. So we'll discuss 500 MCQs, right? Okay. So we have posted the Telegram discussion group link. So then you can drop messages in Telegram discussion group, right? Because sometimes YouTube Live may be frustrating or irritating because of the behavior of some fellows, right? Like, okay, there are some fellows who are who are who are looking forward for a live. Never mind whether it is a class or anything. Doesn't mind the subject. They just need YouTube lives to interact or chat or expose themselves. Then that times it may be difficult, or it may be kind of irritating for you to attend the session, or might be some kind of distraction. But so therefore, if there is someone who's not like to attend the class through the YouTube live, or just engage in the class through YouTube live, then he or she can go with the right Telegram discussion group. I have uh, mentioned the link there in the YouTube live. Okay. Right. Okay. Before you start in the class, send me that you are. Do you ready for the class, or do you ready for the session? This will be an important session, right? Okay. Really important session. Really important session. So please send a ready message to the Telegram discussion group, or either to the YouTube live. Okay. Really important. Now today is the beginning of our end game, right? So today is the first class, and it is the English medium super MCQ discussion, that MCQ 500 discussion. So you can share this live, or you can share this class to anyone, any of your colleagues or any of your friends to attend, right? Because this will be very important for any particular students to attend. And actually for most often for the English medium students, you don't find any. It means you rarely find some good classes, no. So therefore, this will be a quite interesting, or this will be a quite kind of important session for you guys to catch up with the missed things, right? Okay, we'll go around the entire syllabus within the day by discussing these questions, right? Okay, so we'll come to the question number one. Okay. Mm, okay, right. All fine. I think the PDF. So for students who are unable to join with the PDF opening from one end, and unable to attend the session, I have share screen the question paper. So therefore, no worries. So if someone can uh, arrange with the share screen, if someone has printed paper with yourself, then you can continue with it, no matter, right? Okay. And the PDF relevant for the today's session is there in the website. There in the website, you can go to vwbio.lk, and you can get the PDF from the endgame course. There's a course called endgame. You have to go to the correct one. It means you have to go to the English medium one. You have to enroll. These are free enrolling sessions. Then you have to go there, and under the courses category in your website, there will be a card, or kind of a class known as the end game, right? So that it will be there. Okay, right. uh, right, fine. Then we'll start the session. Okay, right. Which of the following is not a field that is studied under the branches of biology. So this is the field that is not studied under the branches of biology. Okay, so there are field like so what are the fields? So just cope around with this fact, right? Field. Now there are three main branches. You have to go. You have to just kind of revise on branches and field areas of fields that I studied. No, right? Then branches here. When you come to branches, there are three main branches. No, right? Zoology, botany, and microbiology. And under those branches, you study right around seven fields. No, seven fields starting from cell biology, right? Then histology, then kind of anatomy, right? Then physiology, biochemistry. Likewise, then what else? Genetics, ecology. Those are the areas of fields that you are studied. No, fields you are studied. So therefore, that is what you have to keep in mind. Then there, so cell biology, histology, physiology, and biochemistry. Those are uh, they are fields, right? So therefore, molecular biology, there is no such field under the branches of biology, right? Okay, got it. Then fine. The answer for the first one is fifth one. Okay. So English medium students, some students are asking that the PDF is not available, right? But you can say that the PDF is already screen shared, right? Screen shared in the session, so therefore no worries, he or she can join. Just mention it for those who saying that they can't log into the website or any kind of issue. But if there is some kind of issue in the website, our technical team will assess that. So if someone is just saying the same thing that can I get the PDF? I need the PDF. I can't log that. Likewise, so such kind of things you can just drop them in the discussion that already the PDF has been shared screen. So therefore no issues of joining the session, right? Later they can download the PDF now. Okay, right. Then second one, which of the following is correct regarding lipids? It is a large biological macromolecule. The thing is, lipids are not they are not macromolecules, right? Lipids are just only large molecules, right? Macromolecules you come to study as polysaccharides. Then what else? Proteins, no? Then what else? Nucleic acids. Those are the macromolecules. But lipid, you keep in mind that is a very important point. Even though they are large molecules, they are not macro. Macro means consisting of polymerizing monomers. No, polymerizing monomers. There is no such monomer for lipids. They just made up of different types of molecules like glycerol. Either can be fatty acids, can be they can be phospholipids there, right? So several different types of groups are collected to form lipids. So therefore, they are not macromolecules, right? The second one, it is a diverse group of not hydrophilic, right? They are hydrophobic because they are not water liking, no water disliking. Now, when you get some oil kind of thing or kind of a fat that no kind of a fat, they are water insoluble, no. Therefore, they are not hydrophilic, they are hydrophobic. So they are hydrophobic. Therefore, the second one is incorrect. Then choline, which is connected to phospholipid, is a polar hydrophilic organic compound. Okay, so now choline, we have to come to choline. Then when we go to the lipids, when we discuss about the lipids, right? Okay, when we discuss about the lipids, so you have to understand now lipid is made up of what? A kind of a head and tail kind of an arrangement, no? So the head is made up of what? Head is made up of glycerol, right? Head is made up of glycerol, one glycerol molecule. Then these tails are made up of what? Fatty acids, no? Fatty acid chains. They are made up of fatty acid chains, right? Okay. Then, right? Then, then we come to glycerol. Glycerol is the head part, and it is kind of what? It is kind of hydrophilic, no? Hydrophilic. So this is hydrophilic. The head part is hydrophilic. Then tail part is hydrophobic. Hydrophobic. Then also the glycerol head part is hydrophilic. Because it is hydrophilic, it means it is polar. Polar hydrophilic. Then tail part is it hydrophobic. Then it is kind of, this tail is non-polar. Non-polar. It means therefore the polar substances are dissolving in water. No? So this is non-polar. Got it? Okay. Right, fine. Then, so therefore, you have to understand now. Then regarding the phospholipid, in phospholipid, now normal this is the lipid molecule, right? In phospholipid, what happened? Instead of this one fatty acid molecule, now there are two fatty acid chains. Then there will be a kind of a molecule known as phosphate, right? Phosphate. So then what is the composition of phospholipid? There's glycerol. There is two fatty acid instead of three. Now third one is cut off in what third one is not there in phospholipid. Then instead of that there is a phosphate molecule. Then phosphate gives a negative charge for the phospholipid. Then this negative charge molecule needs to be neutralized. No, it means it has to be neutralized. So therefore normally for molecules to be stay, they have to be neutral. No? They can't be there with negative or positive charges, or net positive or net negative charges, right? Okay. So therefore, to neutralize this, now this gives a kind of a negative, no, a kind of a net negative charge for the molecule. To neutralize this, we need to insert a positive molecule into the phospholipid. No, that positive molecule is the choline. So what happened? Choline is bound to the head, to the head. Now you have to argue, you have to logically think. Now choline is bound to the head. No, head is what? Head is polar. Head is polar. Polar means it is hydrophilic, right? So then like molecules are get attached. No, so therefore definitely choline needs to be polar. Then definitely choline needs to be hydrophilic, and also choline is positively charged. Therefore choline which is connected to phospholipid is a polar, okay, hydrophilic, got it. Then an organic compound, okay. Then you can furthermore add it is a positively charged molecule. So therefore you have to what you have to know about choline is it is a positively charged polar hydrophilic organic compound. Then third one is correct, okay, right. Overconsumption of cis unsaturated fatty acids, right? Not cis unsaturated. It has to be trans. It has to be trans. So therefore, this is incorrect. Fifth one, hydrocarbon chains of fatty acids contribute to their not to hydrophilic nature. Now we have already discussed that fatty acids are what? They are hydrophobic. They are hydrophobic. Head or either the glycerol molecule is the hydrophilic one. Okay, right? Then therefore, answer number 3. Okay, okay, right. Now we have already discussed two MCQs, right? So could you please kind enough to send me some kind of a feedback whether this is okay? Are you okay with the session? Are you okay with the discussion, or are you okay with the mode of discussion that I am doing? Because this is the first time I am doing a separate English medium session. So please be kind enough, students, to send a kind of a feedback. Just then I can cope with it, no, right? Then I can get an idea if there is any mistake or such kind of thing. Then I can go move along with that. Are you okay with the session? No, no. Point collection. You have to make the point collection according to the that session known as the end game, right? The end game. You have to make the point collection. Here you have to go through the MCQ only and note down the important facts here and there, which I discuss, right? Okay. So this is what for the MCQ 1 and 2. Okay. Uh, Okay, you are very comfortable now. You are okay with this? Oh, all right, all right, all right. Then, so there is no issue. So continue, please continue. Right? Don't move away from the session. If you feel like boring, don't mind with that thing. Right? Okay, continue with the session. So this is very important to go through the session. Right? Okay. Fine. Give me a second that I will change the PDF because I just have a PDF with space, more space available, right? So this will be difficult to mention the things, right? Okay. Right, okay, students. Now I will share the PDF with some kind of space available. Now, right, okay. Because it is easy for me to just draw things. Previous one, there were no spaces, right? Okay. Now we'll come to the third MCQ, right? Okay, we'll come to the third MCQ. No, I just changed the PDF. That's it. That's it, right? Now all okay. Now come to the third MCQ. We'll go quickly. Which of the following statement is correct regarding microscope? The resolution power of a microscope is inversely proportional to the wavelength. That is a correct statement. Now first one already is a correct one, right? It is correct. Now resolution power is what? Inversely proportional to the wavelength. Resolution power. These are the important facts you have to keep in mind. Resolution power inversely proportional to the wavelength, right? Okay, inversely proportional to the wavelength, right? So you have to keep in mind when once the wavelength is increasing, the resolution power is decreasing. So that's why you know the resolution power. I will mention resolution power. I will just put P. Power of electron microscope. I will put EM here. The resolution power of resolution power of what that compound light microscope. I will put compound light microscope as CLM. Then what is happen? Electron microscope is higher. What's the reason? In the electron microscope, what's the reason that wavelengths which are using in electron microscope, wavelengths which are used in electron microscope are very low compared to what is there in the compound light microscope. So that's why with the wavelength becoming decreasing, that resolution power tend to increase with the decrease in wavelength. Okay, so okay, right. Okay, right. Then that is correct. Transmission electron microscope is more suitable to observe the surface view in three-dimensional appearances. Now keep in mind, now this is a very important MCQ session. No? We are discussing 500 MCQ. So here all these MCQs are kind of guess questions because these MCQs are not previously tested, or there may be some here and there parts which are previously tested. But more than that means more than 95% of questions they are new, and they are just kind of targeted questions which we gave in the class. Okay, right? So therefore, you have to keep move on the question and understand the important points and what are the aspects that can be tested in the during the exam, right? Okay. And the other thing is now I don't go I will I won't try to explain furthermore. It is furthermore means that now entire now you have to if we regarding microscope, then there is a kind of detailed aspect regarding microscope. Now then you have to study of compound light microscope. Then you have to go of electron microscope. Electron microscope have two parts. That's the transmission one and the scanning electron microscope. So there are two. Then come to a compound light microscope, you have to go about the magnification, right? Then you have to move to the resolution power. Then around then come to electron microscope also, you have to go for the magnification, then also for the resolution power. Likewise, then there is a kind of a massive area under microscope. So if we take all the time to discuss all those things, then it will be kind of a difficulty when come to the session. It will be quite difficult to discuss the all the 500 MCQs. But keep in mind, now 9th of sorry 9th of November, then there will be end game session. No, end game seminar, then end game seminar. During that, I will discuss around the important facts like now microscope. Now during the today discussion, you will find several amount of MCQs here. You might find around 10 to 15 MCQs from microscope topic, right? Okay, so therefore microscope is kind of a what? Is kind of a common pattern point, right? Common pattern point. So therefore you have to understand what are the common pattern points. Then after the today's session, I will share with you that all the common pattern points of the entire syllabus, right? Common pattern points of the entire syllabus, right? So common patterns of entire syllabus, all the syllabus I will share with you. So for that, prior to that, you have to go through the just theory parts to recall theory parts via questions. That is what we are doing. So that's what I have to just share with you. So therefore, if someone just try to understand the entire theory with the MCQ, so I can do it, no issue. But we have to, we will ended up doing only 30 to 40 MCQs around 7 p.m. right? Because I can explain the entire microscope by taking this MCQ. But keep in mind, this is not such a time to discuss all those things, right? We'll discuss that certain particular that common pattern, those MCQs which are frequently tested during today's seminar. We'll discuss it on 9th of November at the end game, right? So then I will discuss around 52 odd kind of points which will be there definitely in new MCQ paper, and I will take around 12 to 15 hours to complete that session, right? That will be a prolonged session with prolonged detailed parts, right? Okay. Now for now we'll go through the MCQ, right? Then third one. Second one, transmission. So to observe the surface view in three-dimensional appearances, it's not the transmission electron microscope. We are using scanning electron microscope. Scanning electron microscope. Therefore, second one is incorrect. Heavy metals are used to stain the specimen in scanning electron microscope, right? Scanning electron microscope. Then regarding the scanning and transmission electron microscope, in scanning electron microscope, we are using gold to stain, right? Gold, right? Gold. Heavy metals are used in transmission electron microscope, right? So gold is kind of a heavy metal, right? Gold is kind of a heavy metal. It's okay, but what is different with gold and other heavy metals? Then gold versus other heavy metals, what is the difference? Other heavy metals, then gold is there, then other heavy metals, what is the difference? Gold is kind of what? It's kind of a shiny one, no? Shiny metal, no? Then it's kind of a shiny. There is a kind of shiny nature in gold. So that shiny nature helps into what? Helps into reflect the light, no? Reflect the light. So that's why now we are in scanning electron microscope. We are going through the surface, no? So to observe the surface, we need what? That shiny appearance just help in, just importantly scanning the surface. That's what the important thing, right? That gold is shiny. That's the thing, right? Okay, that's why we need gold for scanning electron microscope. Otherwise, for the transmission one, we need we use to stain what? Heavy metals, right? The maximum magnification of light microscope is not 1500 times. It is what is the largest magnification? It is the 1000 times. In the light microscope, visible light first passes through the glass lens and through the specimen. No, from the specimen to the glass lens. Now, if we take the microscope like this, now there is mirror is there, then this is the stage. Now with the stage there is diaphragm hole, then on the stage there is the specimen, then light from the coming from the now come to the mirror, then through the mirror there is diaphragm, and there is condensing lens and all those parts here. So then this light rays through the diaphragm and through go to the specimen. Then on the specimen there is what there is that objective lens, and there are three types of objective lens. Now through the objective lens it is transmitted through the then to the optical tube. Then in the optical tube there is what there is eyepiece here, no? Finally eyepiece. Then through the eyepiece there is the eye of the observer. Then it's moved to the eye. This is the eye. Now this is the eyepiece, right? This is the objective, right? This is the objective. Then on the stage this is the specimen. So then what is the pathway of light traveling through from the specimen to the objective lens to the eyepiece, then final to the eye? So therefore this is the pathway of traveling the light from the specimen to the eye, right? Okay, right? Okay, fine. Then therefore that's the fifth one is incorrect, because it states that from the eye to the specimen. That is incorrect, right? From the specimen it's moved, right? So therefore the fifth one passes through the glass lens and then through the specimen. No, from the to the glass lens is from the specimen to the glass lens. Then that is incorrect, fifth one, right? So therefore answer for the third one is first one, right? Okay, that is microscope. Now second one answer was that containing what third one answer was third one. Then first one answer was fifth one. We have discussed with the different PDFs there, right? This is the pathway, right? Okay, right. Then we'll move to the fourth question quickly. Which of the following statements is correct regarding the Golgi apparatus? So now doing the MCQ, while you are doing the MCQ, this is kind of now in the session also I just kind of doing the paper. No, it's the same way you have to do in the exam. So now once you are reading the paper, you have to highlight. You can highlight or you can circle kind of important things, because you in the paper you can do anything. In the answer sheet that you need to be kind of disciplined. No? But in your paper you can circle these things or you can underline these things. You can do anything you wish, right? Okay. So therefore, then those underlining things and those circling things may be highlighted, right? Okay. So then you focus on Golgi. Now I have circled this Golgi apparatus. Then what is happening now? My mind and my thinking process completely on Golgi apparatus, right? Then Golgi apparatus is kind of highlighted there. Then my mind it started to think of Golgi apparatus. Now, yeah, Golgi apparatus, these functions, these structures, likewise. Okay. Okay. Then come to the fourth one. Involves in detoxification. Detoxification is done by either SCR or peroxisomes, right? Peroxisomes is responsible for the detoxification. Therefore, first one incorrect. Then come to the exists as network of tubular sacs. Then it is also SCR, not Golgi. Produces peroxisome. No, they Golgi are producing what? They are producing lysosomes, not peroxisomes. Therefore, third one is incorrect. Then fourth one, cis phase is present near the ER. That is correct. That is correct. So therefore, fourth one is correct. Produce cellular components such as lignin. No, they not the lignin. They are producing cellular components like what? They are producing cellular components like cellulose. Then again, pectin kind of things are producing, right? Okay, that's the part. Then regarding Golgi, if we take the Golgi apparatus like this, right? Just I will draw this like into this kind of appearance, just simply. Then here the cis phase, here the trans phase. Now closer to the cis phase, they are the, I will just write down SCR and RER. It means endoplasmic reticules are near the what? Near the cis phase, not the trans phase. This is the kind of the arrangement, right? Okay. So that's what happening. So then what in the trans phase and the cis phase? In the cis phase, they are receiving what? They are receiving the transport vesicles which are coming from the SCR and RER. Transport vesicles are here coming for the cis phase, and from the trans phase, they are removing what that secretory vesicles, right? Secretory vesicles are passed away from through the trans phase, right? Okay, right? Okay. So that is what happening in the fourth statement, right? Okay. Then that's the answer. Now we'll come to the fifth MCQ, right? In the cell cycle, there are checkpoints in G1, S, and G2. That is incorrect because G1, G2, and M are the places where we can find the checkpoints. So therefore, not S, right? That is incorrect. Then the second one, synthesis of histone proteins occur in not in G2, it is occur in S phase. Spindle microtubules polymerize during the telophase. Mind you that that there are two procedures taking place. Now if we take spindle microtubules like this, now they are made up of what? They are made up of tubulin protein. No? They are made up of tubulin protein. Then what happens? This tubulin get connected to form what? They are get connected to form microtubules. I will just roughly draw like this. Then they are connected to form filaments, right? Okay. Then what happens is spindle fiber filaments are formed like this, and this is what arranged. Now these microtubules are just converted into what? They are converted into spindle fibers, right? Okay. Then spindle microtubules, right? These tubulin molecules. So what happened then? These tubulin molecules are get collected or get polymerized. This is known as the upper arrow is known as, this is kind of a polymerization, no? Polymerization, right? Then what happened? This structure can be once again can be broken down into these tubulin just tiny molecules, right? That is known as depolymerization. Depolymerization. Now this polymerization process, it is taking place in what? It is taking place in prophase. It is taking place in prophase, and this depolymerization phase is process is taken place in telophase in the cell cycle, right? Okay, this is what happening. So you have to keep in mind. So therefore, spindle microtubule polymerization is not taking place in telophase. It is taking place in what? It is taking place in prophase. So therefore, that is incorrect. Centrosomes reach opposite poles during the pro-metaphase. No, it's during the metaphase they are reaching the what? They are reaching the opposite poles. Then again, come to the fifth one. Cystochromatin separate all the centrosomes during anaphase. That is correct. So the fifth one is the answer. Right? Okay. Right. Okay. Fine. Then that is what you have to understand on the fifth one. Right? Fifth one. Right? Fine, right? So note down the important facts, right? Note down the important facts you need, right? Okay. Now see, now you can now come to the PDF. Now just kind of a recalling thing and just kind of a notice for you, right? Now here you come with the speed recovery or the kind of, this is the Podu Ratawatute, right? Okay. Now when you come to the Podu Ratawatute, right? Okay, the Podu Ratawatute, this is the important fact of the Podu Ratawatute. Now this is what we will be doing on now today, 4th, 5th, 6th and 7th of November, right? Now you can find here now kind of things that we have come across with these MCQs microscope now. No, so this is what you have to discuss. Now you see now in the question number 35 and 36, now it is asking a fine beam of electron is reflected from the surface of specimen, right? Now these kind of, now this is what they were in the MCQ, right? And these parts related to these structures. Now what glass lenses are used in what, heavy metals are used in what, which one, right? Likewise kind of a recall, no? Right? So then again here then that is for the microscope, no? This speed recovery is for microscope. So this will be a kind of a very important thing. I think this Poduratava tute or the common pattern tute is the most important thing. Now while you are doing the MCQs, you are getting into a point where how I can keep studying these things, no? Then for an particular points, now what will be tested in the MCQ? There are several organelles. Now here is about the Golgi apparatus in the question number 5. We have now what we have discussed. But what will be the question for our year? It means what will be tested in 2025. So therefore for that there is a complete analysis on organelles. Now we'll come to the, when come to the 37th one, it is about the organelle or the subcellular or the extracellular component that correspond to the following description or the following functions. Now here a lot of functions, no? Protein synthesis, synthesis of glycoproteins, photophosphoration in plants, then maintenance of cell shape, right? Then components of apoplasm pathway, stores calcium ions. So there are plenty, right? Okay, there are plenty of it. It means almost all the functions, all now producing lysosome, right? That was they are tested in that MCQ, no? All the available functions in the entire resource book are listed there in the this PDF. So it will cover the entire that Poduratava, kind of a Poduratava fact that the organelles MCQ is a must in your A-level exam paper. Therefore, that this practice or the 37th one will cover the entire organelles, right? Okay. Then when we come further, there is cell cycle and the process of cell division, right? Okay, you will get it. No? Now there are several kind of things. Now in the early MCQ, you will find about the several functions or the several processes. What is the phase that is taken place, or what is the phase responsible for those process? Now here we come. Now these are the processes given. These process from the entire resource book for the cell cycle. There are now I think around 25 processes here, no? Right, for the mitosis. Then we come to the meiosis part. Then meiosis will be tested. Then what is the phase? Now what is the phase belonging to nucleolus begins to disappear. Then genital non-identical haploid 4 daughter nucleus are formed. Then what is the phase belonging to? Likewise, that meiosis also will be revised there. So all the points the necessary or all the points required for the exam under the mitosis and meiosis are there in the what? They are in the Poduratava tute. Okay, got it. just drop me a message that did you understand the relationship between the doing papers and what is there in the Poduratav tute, and what is the difference there? It's just not only a tute, right? Poduratav is the whole thing, right? It's the whole thing. It's the entire. I think that's the entire point, or the entire tute that is a kind of a golden one, right? Golden one, very very important for your exam, right? Very very important for your exam. Entire exam paper will be there according to the Poduratav tute, right? Okay, Poduratav tute. So therefore, it's a must. So you have to take a printout there. We'll complete it on the 7th of right, 7th of November. That's the day. I think 7th or 8th. I think 8th, no? 8th, I think. 8th or 7th. I just missed the date, maybe. Maybe 7th or 8th, I think, according to the timetable, that will be discussed on the particular day. Right? Okay, so understand it's kind of a addition announcement, right? It's kind of a alerting thing, right? Okay. Right, okay. Then come to the 6th MCQ. Will come. Then 6th one. Which of the following is correct regarding enzyme inhibitors? Poisons are irreversible inhibitors, right? Yeah, they are irreversible inhibitors. Then first one is correct. Most competitive inhibitors are reversible. Then this one is also correct, right? So therefore, two answers here. First one, second one. Non-competitive inhibitors compete with substrate molecules. No, this what compete with substrate molecules are the competitive inhibitors, right? They are the competitive inhibitors, not non-competitive inhibitors. What are just kind of compete are the competitive inhibitors. Therefore, that is incorrect. The effect of non-competitive inhibitors can be reversed by increasing the substrate concentration. Now, keep in mind, the effect of non-competitive inhibitors cannot be reversed because they are permanently bound. Then what we can reverse is the effect of what competitive inhibitors by adding the substrate. Then by adding the increase in the substrate concentration, what we can just reverse is the competitive inhibitors behavior. Some inhibitors bind reversibly via covalent bonds. Mind you, now covalent bonds are kind of they are strong bonds, right? They are strong bonds, not they are weak bonds. They are strong. Therefore, if they, when there is covalent bonds, it means, right, those inhibitors are bind not reversibly. They are binding irreversibly. They are bind irreversibly. Now keep in mind what when there is covalent bonds, right? It means they are strong bonds. So it's strong means once some inhibitor is bindly strong, it means they are irreversible, not reversible. Fine, okay. So that's the point you have to keep in mind. Now there are two kind of inhibitors. Now this is the active site of the enzyme. Now these are two enzymes. Now one inhibitor, one kind of inhibitor is binding to this active site, no? They are binding the active site. Then the other kind of inhibitor, they are binding to the non-active sites and alter the shape of the active site, no? Now the active site will be kind of a alteration in shape like this. So that's what happening, right? Okay. Then when come to the reaction rate, likewise, then what happen now when you add, now we'll come to the, now the reaction rate is kind of this kind of a thing, no? Reaction rate is this with the concentration. Now when you add inhibitors, it will lower like that, but will come to an end with the similar thing. Now this is kind of the competitive inhibitor. This is the competitive inhibitor. Then this is the normal reaction without inhibitors, this one. Then what happen with the non-competitive inhibitors? Then it will no way come into the the maximum place. So this is known as this particular place is known as Vmax, or the maximum speed that that reaction can obtain. You know, you are not supposed to have that kind of a knowledge, but keep in mind now once it is non-competitive, they can't reach that maximum rate value. Okay, maximum rate, because the inhibitor is permanently bound. Then what happen now? This is x-axis is the concentration of the substrate concentration. Once the substrate concentration is increasing, what happen? You can reverse the activity of this competitive behavior. That is what discussed there, right? Okay. Right, now we'll come to the seventh one. Seventh one, okay. Right. Select the correct statement regarding C3 and C4 plants. This is a very important one. Maize and paddy are example for C3 plants, right? Okay. Right. Maize and paddy are examples for C3 plants. What you think? That is incorrect, no? Right? So that is incorrect. Then carbon dioxide fixation occurs first in bundle sheath cells in C4 plants, no? Carbon dioxide fixation occurs first in bundle sheath cells in C4 plants. No, that is incorrect. First, carbon dioxide fixation is taken place in what? That is taken place in mesophyll cells, right? Mesophyll cells, right? Not bundle sheath cells. Therefore, that is incorrect. Photosynthesis only occurs in mesophyll cells in C3 plants. That is correct because C3 plants in C3 and C3 and C4. What happened in C3 also? There are two types as there are mesophils and also bundle sheath cells, right? There are bundle sheaths also found in C3, but mind you, in C3 there is no cross structure. Now in C4, there are also mesophils and what? There are also bundle sheath cells, right? Bundle sheath cells found in C4. And what is that structure? We name that structure as trans tissue structure, but in C3 there is no such structure. But what's the important thing in C3 plants, bundle sheath, what they lack pigments. They lack pigments means they are not green in color. Therefore, what they are lack pigments means there is no photosynthesis. Only mesophils do photosynthesis. But in C4, both mesophils and both bundle sheath, they can perform photosynthesis. That is the important point, right? So photosynthesis only occur in mesophyll cells in C3 plants. That is a correct statement, right? Okay. The optimum temperature of C3 plants is 35 Celsius. No, that is incorrect. Optimum temperature for C3 plant is 15 to 25. Now keep in mind, this fact is now not furthermore in your resource book. This was just removed from the 2022 edition. Up there were in the updates from 2022. This point was removed from your resource book. So, but keep in mind they might they can test, no, because this was they are available in the resource book until 2022. So we don't know, but just keep in mind 15 to 25 is the optimum temperature for C3 plants, right? Carbon dioxide fixation is carried out in C4 plants by that this is incorrect. PP is what? It is a 3 carbon compound. Therefore, that is also incorrect. Answer number 3 is the answer. Okay, right. Right. Okay. Now it's correct, no? Right. Okay, that's the answer. Uh, and the first one, the reason is maize, right? Maize is a what? Maize is a C4 plant, right? Not a C3. That is a C3, right? That's why first one is incorrect. Okay. We'll come to the eighth one. Right. During the Paleozoic era, origin of mammals took place in Mesozoic era. Now keep in mind, this is a very important fact. Important fact, right? Now regarding the eras and eons, right? Now regarding eras and eons, now keep in mind, from 2022, that kind of what update in the resource book, right? They are they are saying that these eras and eons no need to be keep in mind. It means you need no need to study. Student not have should not study those things because those things won't be tested in exam. Like that kind of a idea is conveyed. Now you can go through your resource book in the unit 3 and go to the eras and eons part. Then there will be inside brackets such kind of a notice, right? But keep in mind, so I have to state that, no, but it means there are guys who are looking at this live and those are frustrated of this live and might say that guy is just teaching all syllabus. There are no eras now. Eras are not tested in exam like us. But once you go through the past paper questions, you will realize, right? Once you go through the past paper questions, you will realize now, yeah, eras and eons are tested. Yeah, right. eras and eons are tested. Oh, that's the thing, right? So therefore, we can't assure that this won't be appear because the resource books are made from NI, but the paper is made by Department of Examinations. Those are two different institutes, so you can't predict what they will do. But for our safe and for your sake, I have added MCQs regarding those eras and eons also. Got that point? Just drop a message, Namai. Right? Okay. Just drop a message. Did you get that point? That regarding the eras and eons, that is the truth. Okay, that's the true aspect regarding that. Okay, right. Then genus Homo. Now, first one that was taken place at Mesozoic, right? That was taken place at Mesozoic. Okay, genus Homo originated at Cenozoic. Okay, Cenozoic. Then third one, the origin of Earth took place at, right, Hadean neon, Hadean neon. Right? Okay, Hadean neon. Then the first tetrapods evolved. That was correct. That was in the Paleozoic era. Then dinosaurs were evolved at the Mesozoic era. Were at the Mesozoic era. Right? Okay. Then answer is the answer number 4. Okay. Answer number 4. Right. For the ninth one. Correct statement regarding the classification system of organisms. Now, when regarding the classification, there are two types of classifications. Now we come to the classification. There are two types of classification. One is the artificial classification, right? The other one is the natural classification, right? Okay, right? Natural and the artificial classification. Now the artificial one is the most oldest classification, right? This will be discussed in that Poduratawa session, right? Furthermore, the classification we'll discuss it taking 10 to 15 minutes regarding classification, because this kind of a guess part, right? Guess part, this classification. So when come to the natural classification, it is the newest one, means after the artificial classification. But the thing is, in the natural classification, that evolutionary relationships are considered, right? That evolutionary relationships are considered. Evolutionary relationships are considered in what? In the natural classification, but not in artificial classification. No evolutionary relationships. So that is the main differences. We'll go through the question. Natural classification is the only classification used before. Now, used before 18th century means the oldest one. So therefore, that is the artificial one, not the natural one. It is the artificial one, right? The oldest one, artificial classification. Artificial classification is difficult to use. No, it is very easy to use. Not difficult to use. It is very easy to use, because just the organisms are classified basis on their appearance, whether there is organisms with two legs, like that kind of difference, that kind of characteristics, with two legs, with four legs, right? Okay, with six legs, okay, or without legs, like that kind of, and right, then wings present, wings absent. Okay, so such kind of similar that simple things are considered when classifying organisms. That is very simple. Even we can classify regarding that artificial system, right? So therefore, no any evolutionary relationship or any genetically variations, or those things are not considered, right? So that is very easy to use. Very easy to use, right? Evolution relationships are considered. No, they are not considered, right? Not in artificial classification. More organisms can be added and enhanced under the natural classification system. Mind you, in the natural classification system, you can't add new organisms, because that organisms, when you need to add new organisms, right, that is difficult, or that is kind of impossible in natural classification, because it is already classified using the genetical variations and genetic composition. So what you can do, if you need to add the organisms, then that is in the artificial classification, because that is very easy to use, and you can add new organisms as you wish. Because now see, now we know 2-leg organisms, 4-leg organisms, 6-leg, and without legs. No, so that is the classification we have done in the artificial. Now mind you, there is a new organism found with what? 10 legs. Then what will we do? We will add it into a new group as 10 leg organisms, right? Okay. But In natural classification, from the time that they involve Nidaria, right? Okay. Then the time they found Platyhelminthes, then Nematoda, right? Then Arthropoda, then Mollusca, then Echinodermata, right? Then Chordata. These are the classes they are used. No, sorry, these are the phylums they are used. No. Now, apart from these phylums, they haven't used anything, right? Okay. They just rely on these phylums. Now, what they do, if they find another X kind of a new organism, they go to these phylums and check whether this organism goes with this trend. Then they don't add another particular phylum as X. No, that is not doing here, right? They just add the X organism into the existing phylum. But here what we do, 2 legs, 4 legs, 6 legs, and without legs are the groups available. Then we suddenly found a 10 leg organism. Then again we add a new group as 10 leg organisms, right? Okay. So that is the difference between artificial and natural classification. So therefore, more organisms can be added and enhanced not under natural, it's under the artificial one. Natural classification depend on several characteristics. Okay, several characteristics, that is correct, because it is kind of a complex one. Okay, complex one, different characteristics like morphological one, structural one, genetical one, molecular biological levels, and different of aspects are considered in regarding the natural classification, but not in artificial classification, right? Okay. Then come to the 10th one. Okay, this is regarding the kingdom Protista. Kingdom Protista. Okay, presence of contractile vacuoles and food vacuoles. That is, we can take amoeba and paramecium for that. No, amoeba and paramecium for this one. These are scientific names. I will underline this. Then highly diverse based on shape. That is the diatoms, right? That is the diatoms. Then multicellular thallus with floats. When you come to that, you have to take sargassum with that, right? Sargassum. Okay, sargassum. Then those are the organisms. So the organisms that show the above characters respectively. Now we can go through the answers. Now first one is incorrect because there is no amoeba or paramecium. Then we'll come to the third one where sargassum need to be there. Then we can cut out the second one because no sargassum here. There is no amoeba or paramecium here. Then 3, 4, and 5 are remaining. Then for the second one, it needs to be diatom. So therefore, this is incorrect. So there is no diatom here. It is incorrect. Then the answer is answer number 5 for the 10th one, right? Okay, fine. So that is the question number 10. Now we have now step by step. Now 10 out of 500 is kind of a milestone. Now we have completed around 2% of today's work. Just to kind of update you on that. Now we have completed 2% of today's work. Okay, right? Now students, just be kind enough. Now, okay, are you okay with the discussion? Now 10 questions are done, right? Not many, but by question we are completing the today's work. Now 10 done, right? Okay, drop me a kind of a comment or kind of a message like, are you okay with the discussion now? You are going along with the discussion nicely? So better to take a printout of this document. Okay, the document and all this PDF are available in the website. You can get them, right? Okay. So then with one comfortable with PDF, you can continue with the PDF, no issue, right? These are valuable MCQs, right? The same MCQ will be there in the question paper itself, right? So there are some guys I noticed that they just enter the live and just they go likewise, right? So what I need to tell them is you will just kind of regret, right? Shit, I have left the live. I just only enter the live and I just left, right? But the questions itself suggest now your colleagues might discuss in your exam hall that one guy who did those the end games, that questions were there in the paper, no? Likewise, then you will be frustrated, right? So don't be like that. Okay. Okay. Then come to the question number 11, right? Then the statement that can be accepted regarding the seed plants. All seed plants have sperms that do not bear flagellas, right? Not all seed plants have sperms, right? Angiosperms have sperms. Angios Angiosperms, right? have sperms, and some of the gymnosperms, right? Not all. Not gymnosperm is some seed, right? Now seeds are two types, no, gymnosperms and angiosperms. Now, sorry. Now some gymnosperms have flagella. So therefore, not all the seed plants, all angiosperms, that is correct. And but some gymnosperms. So therefore, seed plants are two types, no, angiosperms and gymnosperms. Then there are some gymnosperms with flagella, and it indicates that there are gymnosperms without flagella. No? Therefore, you can't say all seed plants. That is incorrect. No? Second one, Archegonia retains spores within the sporophyte cell. Yeah, Archegonia is the this is the what is the female part, right? So female part of the plant. Yeah, it retain with the it retain the spores. That is correct. Second one, correct one. Third one, the embryo is covered by a protective covering called the seed coat. No, the embryo is covered by, yeah, it's covered by a protective covering, but it's not the seed coat. It is the now which is saying protective no seed coat. No, so what is covered here by the seed coat is the now embryo is not covered by the seed coat. Embryo is covered by the embryo covering, right? Okay, right. So there's embryonic layers, and seed coat covers the what? It covers the endosperm, right? Endosperm is covered by the seed coat, right? Not the embryo, right? Not the embryo. The gametophyte is always microscopic. You can't say that gametophyte is always microscopic. It is mostly microscopic, right? It is mostly microscopic, not always microscopic. Got it? Not always microscopic, right? Mostly microscopic, because there are some situations where we can see the gametophyte. It means kind of macroscopic, no? The dependent sporophyte obtain nutrition from the gametophyte, right? Okay, now come to the question number fifth. Statement number fifth. The dependent sporophyte obtain nutrition from the gametophyte. Now, regarding seed plants, no. So when come to the seed plants, what is dominant in seed plants? What is dominant is the sporophyte. So therefore, not the dependent sporophyte. It has to be dependent gametophyte obtain nutrients from the sporophyte. So that statement is changed. Change means two phases are changed there. Okay, fine. So what happened now when goes through the seed plants? Now we'll kind of in seed plants there are what is the dominant and what is the recessive. Now dominant one is there, and also there is a recessive plant in seed plants also. Recessive one also there. So what is the dominant one? Dominant one is the sporophyte, and what is the recessive one? It is the gametophyte, right? It is the gametophyte. Okay, so keep in mind. Then come to the 12th one. Okay, right? Consider the following processes carried out by annelids, right? Okay. Then the structures that carry out the above functions respectively are now for locomotion. Now we'll go through the annelids. When we go through the annelids, for the locomotion, they have parapodia, right? They have parapodia. Now we'll write down those things. Parapodia for locomotion. Then also they have setae for locomotion. Setae for locomotion, and also they using suckers for locomotion, right? Then for external fertilization, they use clitellum for external fertilization, and for respiration, they are using parapodia, and for feeding, they use suckers. Now, keep in mind, now external fertilization, that external word is not now given in the resource book, so therefore better to cut down that external word. Just write fertilization. Now with the way they have given in the resource book, it suggests like that fertilization is internal kind of one, right? They say that the fertilization is occurs inside the kind of a sac, right? The fertilization is inside the sac. Inside the sac means kind of internal answer. Therefore, in the paper also cut down the phrase external, right? So better internal fertilization or the normal fertilization. These are the structures. Now, respectively, that is asked the structures are respectively asked in the question. Now, First one, it is incorrect because theta are in the final one. There is no theta. You have to final one need to be the definitely need to be the suckers. Therefore, second one is also incorrect. Then third, fourth, and fifth remain as the answers. Then for the second one, it is only the clitorum, so not setae. Then that is incorrect. Then not pseudopodia. That is also incorrect. Then what is the answer? That is the fourth one, right? So pseudopodia needs to be correct as parapodia, right? Pseudopodia need to be correct as what? Parapodia, not pseudopodia. Keep in mind, right? Parapodia, right? It's a kind of a typing error, right? Answer number 4. These are really targeted questions, right? So it's processes found in annelids. These are kind of high yield points, right? Cells of meristematic tissues now have a dense cytoplasm. First one itself is correct, right? They are having dense cytoplasm. Mostly living, not mostly, sorry, not mostly, that all are living, right? Have a central vacuole? No, they are having a central what? Central nucleus. They are having not vacuole. Are differentiated? No, they are not differentiated. They are non-differentiated. Meristematic itself say that they are not differentiated. No, undifferentiated or non-differentiated. Then not mostly isodiametric. They are all isodiametric. Not mostly. They are all isodiametric, right? It's a simple MCQ. Then come to the question number 14. In the root apical meristem, root cap is formed due to the cell differentiation of cells produced, not inwards. Root cap is as a result of outward cell production, right? Outward cell production. Root cells elongate up to 10 times their initial their initial is repeated here. 10 times the initial length due to their activity. That is correct. Okay, 10 times, right? Because of root. Now what is happening in the root apical meristem? Now this is the root. Now here the root apical meristem is like this. Now what is happening? Now we'll see. Now this is the first one. No? Now what happened with the time? With the time, that root is elongating. Now we can just draw the root like this. Okay, just draw the root like this. Now this is the root apical meristem now, and there is a covering like this also. There's a covering like this. Now what happened? Now this was the earlier level it was. Now and this is the earlier length actually. Earlier length I will denote it as simple L. Now what is the length of the root right now? That's the length is what? That's the capital L. Now you can see L is greater than simple L. Roughly they say now that L equals means you can't say that it is definitely that that approximately equals, but L is around, sorry, L is around 10 times simple L, right? That's what they are saying. That's the process is happening. Now what happened? Now this root apical meristem is cell division is in two direction. Inward cell direction also, cell division also. Then there is outward cell division also. Now what happened with the inward cell division? Inward cell division, that new cells are forming inward, no, in the interior side, interior aspect. Now with the outward cell production, that is by this outward cell production leads into formation of what? This structure known as root cap, kind of a protector for the root apical meristem. When the root going downwards through the soil, there is friction, there is damages. So that root cap protects, its guard, kind of a protection is given for the that root apical meristem. That is the importance of this root apical meristem, which is formed due to the outward cell growth from the root apical meristem, right? Okay. Contributes to increase the diameter, not the diameter, that's the length of the rooting. Contribute to increase the length of the roots. Then protected by leaf primordia. Leaf primordia protect the what? It's protect the shoot apical meristem, not root apical meristem, right? Leaf primordia protect the shoot apical meristem. So the root apical meristem is protected by what? It is protected by root cap, no? Root cap. So therefore, that is incorrect. Then cells with a great length are present near, right? Cells with great length are present near. No, great length, not elongation, right? So here what happened now regarding this part, this certain particular now associated with the that root apical meristem. This is the zone of cell division. Zone of cell division, right? Zone of cell division is here, right? Zone of cell division. So not the zone of elongation, right? Not the zone of elongation is here, right? Right. So keep in mind. Right, therefore, cells with a greater length are present here. No, the dividing cells are present near, right? Not greater length, cells, dividing cells, right? Cells which divide the present near. Cells which divide the present near. Now, answer for the 14th one is second one, right? Okay, kind of a deep theory basis on root apical meristem. These are definitely can be tested in your exam, right? So there is a must MCQ from tissues. No, there's a certain MCQ. There's a definitely MCQ may that under tissues from either there is one from plant tissue also. There is one from that animal tissues also. That plant tissue can be given from root apical meristem, right? Okay. Now the 15th one. Diffusion uses external forces to move molecules. No external force, right? No external forces are used here for the diffusion, right? Then 15, second one, right? Can occur through any membrane. That is incorrect. Occur if only the membrane is permeable, right? Keep in mind. Then occur during the transport of water and water soluble substances through the plasma membrane, not plasma membrane, right? Diffusion is not taken place in plasma membrane. In plasma membrane, through plasma membrane, what happened? That is the osmosis. So if this is diffusion, it is through the cell wall, right? Okay. Then if it is plasma membrane, it is the osmosis, right? Occurs because plasma membrane is what? Plasma membrane is kind of a what? Is a selective permeable, no? Selective permeable layer, right? Selective permeable. So therefore, that is incorrect. Occurs during the transport of oxygen and carbon dioxide through the cellulose cell wall. Now, it is taking place through cell wall. The diffusion take place across cell wall, but keep in mind, now oxygen and carbon dioxide are not moving through cell wall. They are moving through what? They are moving through plasma membrane. Plasma membrane. So therefore, if it is diffusion, then occurs during the transport of oxygen and carbon dioxide through not the cell wall, right? It has to be plasma membrane. Contributes to the movement of population of molecules along a particular direction. That is correct. That now we'll see. Now this is a kind of a stream view thing. Now this is a movement. Now one molecule here, right? Then one molecule this direction, another molecule this direction. So likewise, but collectively, all these molecules are moving in this resultant direction, no? Like that. Right? Okay. So therefore, fifth one is correct. Then we come to 16th. Okay. Right. 5. Then come to the 16th one. pressure potential, it is the physical pressure that is produced, not produced by the solution. No, it is the physical pressure that is produced, right, on a solution, right? Produced on a solution, not by a solution, produced on a solution, right? So that is why first one is incorrect. Always has a positive value compared to atmospheric pressure. Not always have positive value. It can be either positive or can be negative. Now, when come to negative, xylem vessel element, xylem vessels is a great examples for negative pressure, right? Negative pressure, right? It always positive within living cells. Now, this is a very important fact. Yeah, it is always positive within living cells. This is a very important fact. You might highlight that one also, because now come the xylem vessels. It is a what? It is a non-living one, no? Non-living cells. So therefore, even though they are negative in the pressure potential can be negative, they are negative in what? They are negative in non-living cells, not in living, right? Not in living. In all the living, in all the inside, within all the living cells, they are always positive. That is a very important point, right? Very important point, right? Okay, right. Okay, then okay, right, then it's positive in xylem vessels, right? No, it is negative in xylem vessels. Water potential of the cell reduces when turgor pressure increases. What happened? Water potential of the cell reduces when, no, with the turgor pressure increases, water potential of the cell is what? It is increases. It has to be increased. So now psi p is kind of a direct proportion with psi. No, we'll write this one, right? This manner. Now psi is directly proportional with the psi p. Psi p means the psi p is directly proportional with the pressure. No, the turgor pressure. Now this is the relationship. Once the pressure increases, what happen? Psi p is increases. With the psi p is increases, psi is increases. Psi means the water potential, no, right? So this is the relationship you have to take, right? So one student is asking of the previous question, can't we take third one as correct statement regarding the special diffusion? No, you can't argue like that, because the question itself is regarding diffusion. It is not given as the special diffusion. No. There is a special diffusion. You are correct. That is a facilitated diffusion. But the question is about diffusion. So therefore, you can't take the answer thinking, oh yeah, this can be true for the special diffusion. No, it's a different one. It's not the normal diffusion, right? So that's why that is incorrect. Okay. Then come to the 17th. Correct statement regarding the nutritional requirement of plants, right? Zinc activates the synthesis of chlorophyll in plants. Zinc activates the synthesis of chlorophyll in plants. That is correct. That's a function of zinc, right? So this nutritional function is another important fact, important part. So they are in the Podratava or that speed recovery pro one. That will cover the plant nutrition, and also not only plant nutrition, animal nutrition like vitamins, minerals, and all those things will be covered, right? Okay. So those are discussed there, right? Then second one, reduced internode length is a symptom of manganese deficiency, right? Reduced internodal length is because due to zinc deficiency, not manganese deficiency. Not manganese, it is zinc, right? Chlorophyll is an element that is required in large amount by plants, not by large, it is by kind of a small amount, right? Right. Micronutrients, right? Kind of a small means trace one. Sulfur is required for the transcription of DNA in plants, right? Uh, right, okay. Then standard growth of, now we'll come to the transcription of DNA in plants, right? Now sulfur. Just I feel like it's kind of guys are just only listening what I'm saying. Now what about the fourth one? Is it correct or wrong? Kind of a fan poll question, right? Just need kind of a quick reply from you. What is the element required for the transcription of DNA? Is it sulfur? Is it correct, or is it another one? Quickly, hurry up. So then what is the required one? That is incorrect, no? Right? So that is incorrect, false, right? Zinc is the one required for transcription of DNA, no? Right? Then standard growth of plant is symptom of magnesium deficiency. Then that is due to not magnesium, it is due to nitrogen deficiency, no? So here is not sulfur, it is zinc, no? Right? Okay. Very good. Then there is kind of MCQ which can be given from minerals and elements, right? Okay. Then come to the question number 18. Examples of plants that are bisexual and show homospory, right? Right? Bisexual and homospory, right? Uh, right. Then when come to you have to regarding you have to think of heterosporous and homosporous, right? So right, then then regarding that one, okay, then regarding that one, so first you have to keep in mind now regarding the homosporous and heterosporous one. So what you need to keep in mind now, all the that heterosporous, right? Heterosporous ones are, it means once we regarding heterospores, all the seed plants, right? All the seed plants are heterosporous, right? Then other one is some seedless plants, right? Some seedless plants are heterosporous also. So you need to keep in mind, right? So some seedless are heterosporous. So now key fact is when regarding the heterosporous, some seedless, one example is Selaginella. Selaginella is another one example, right? So then you need to keep in mind that lycopodium is a homosporous one, right? Lycopodium is a homosporous one. So you have to keep in mind that one also, right? Homosporous one. So then now we'll move into the question now with without considering this bisexual one, right? Without considering this bisexual point, you can go through the question with the homospory part only, right? So now Selaginella, incorrect. Cycus, incorrect, because these all are seed plants. Pinus, incorrect. Selaginella, once again incorrect, because it is a seedless, but it is heterosporous. But then what is the answer? Then seedless Neprolepis, seedless Lycopodium. So answer is answer number 2. Right. Then come to the question number 19. The plant hormones that contribute to the stimulation of seed germination out of the following, right? Seed germination, right? Then you have to keep in mind stimulation of seed germination. So then you have to circle these kind of things once you are doing the paper, right? Once you are doing the in the exam, you have to circle these kind of things. Then what happen, these kind of things are very important, right? Okay. Then come to seed germination in the question number 19, right? So what not oxine? Yeah, gibberellin helps, cytokinin help, not the abscisic acid. Then B and C, answer number 2, right? Answer number 2, right? Those are simple facts, but you need to keep in mind. So these plant growth substances, right? Then these plant hormones, these are very important. So you need to go through the Poduratawa tutor, that common pattern one. So that includes those kind of things are listed. A list is given, right? A kind of a list is given. Then you have to select what is the plant growth substance. Likewise, then that will practice you once you do it regularly. It will just keep helping you memorize those facts, right? Okay. Then come to 20th one. Select the process that is regulated by phytochrome receptors, right? Right. Then, okay. When come to phytochrome receptors, there are in plants there are two kind of receptors, that is cryptochrome, and then what is the other one? Phytochrome. Phytochrome means actually there are red color receptors. Cryptochrome means blue color receptors, right? So that is the point regarding when you come to receptors, right? When you come to receptors, that is the point. So with the phytochrome or the red color receptors, they help in actually seed germination, and they help in once again what? Shade avoidance, no? They are, it means that kind of they are directly given like that phytochrome are responsible for this. But keep in mind the other important fact is that phytochrome can regulate, it means almost not if it is given like that specifically seed germination and shade avoidance, but most of the activities can be regulated by phytochrome, right? You have to keep that in mind also, right? Okay. So out of this, then maybe phototropism, closing of stomata can also be regulated by phytochrome, but in the resource they have given these two, no? So you have to get the answer from these two. Then so what is the answer? Answer number 4. Seed germination is the answer, right? Okay. 20th one, fourth one is the answer. Right. Okay. Isi එකට එන්න 21st, right? Right. Okay. Deficiency of what vitamin B causes beriberi, pellagra, and anemia? Right. Okay. Right. Then beriberi, it is by thiamine. Then pellagra is due to deficiency of niacin. Then anemia can be because of B6, B9, B12. It means what? That pyridoxine, then folic acid, then cobalamin. Cobalamin. Those are the deficiencies, right? Result in those, right? Okay. So just keep in mind that anemia is a kind of a important fact, right? Important fact that those three vitamin deficiencies lead into anemia, right? Anemia, right? So then you can write here also B1 and B3 with that short form also, right? Then B1, B3, here B6, B9, B12. Okay. Then what is the answer? Answer number 1 for the 21st. Then 22nd. So this is also a kind of a what a common pattern, a kind of a Podurata one, right? That vitamin, that there's definitely an MCQ from vitamin or either from vitamin or minerals, right? They are definitely in question paper, vitamins or minerals, right? So you have to keep in mind now up to what we have discussed right now here. Now this one that 19th one, it is also kind of a what? This kind of a common pattern that plant growth substances, there is a definite MCQ from plant growth substances, right? Okay. Then when come to 18th, it is also a kind of a common pattern point or a kind of a Podurata point that regarding life cycles, that is more oftenly tested, right? That's definitely MCQ. Then come to the question number 17. Those are plant nutrients, right? Plant nutrients, there is definitely a MCQ from that aspect also, right? Okay, maybe kind of function or maybe deficiency symptoms, right? We'll discuss those on 9th of November with that end game session, right? So those are the common pattern. Then this 15th one also kind of a common pattern, that water transport systems are given but not each and every year, but most frequently they have been given. But I will give you the list after this MCQ regarding that common pattern parts you have to keep study on, and I will discuss those on 9th, okay, right? 9th means on Sunday, right? Okay, then come to 22nd. How hemoglobin exists when it's bind to O2, CO2 and carbon monoxide. When it is bind to O2, we name it as oxyhemoglobin. Oxyhemoglobin is what we say when it's bind to O2. When it is bind to carbon dioxide, we name it as carbaminohemoglobin. Carbaminohemoglobin, right? When it is bind to carbon monoxide, we name it as carboxyhemoglobin. So this you need to keep in mind these facts. These are very important. So what is the answer? Answer number 2, right? Answer number 2, right? Normally we speak of oxyhemoglobin, no? Oxyhemoglobin means HbO4, right? Oxyhemoglobin, we always discuss about oxyhemoglobin. So keep in mind. There are 4 binding sites in hemoglobin molecules. That's why O4, right? 4 binding sites in hemoglobin molecule, right? Then come to question number 23. The correct respiratory structure organism combination is given, right? So then skin. When come to skin, that is not earthworm. Those who have skin are the amphibians. This is very important point. Amphibians have skin. What earthworm have? They have the body surface, right? They have body surface for respiration, not skin, right? Right, not skin. Then tracheal system are found in they are found in insects, right? Tracheal systems are found in insects, right? That is found in insects. So what crocodile have? Crocodile have what? They have lungs, right? Lungs. They have lungs. Then third one, book lungs, right? Book lungs are present in spiders and scorpions, right? Book lungs are present in what? Spiders and scorpions. So note those things, these points, right? Scorpions have what? Book lungs. Spiders and scorpions have book lungs. Then cockroach is kind of an insect, right? So insects, they have what? Tracheal system, no? So therefore that is incorrect. Then shrimp having internal gills, that is correct. So fourth one is correct. Then shark have also what shark also have, that's incorrect, they have internal gills, not external gills, right? Internal gills. Those have external gills are the marine annelids, right? External gills can be seen in marine annelids, right? Okay. So that is for the question number 23rd one, right? Okay. You need to note down because these parts can also be tested in your exam, that respiratory structures, then excretory structures, no, then nervous system arrangements, how the nervous system differentiate with each phylums, no. So then once again they can give that circulatory structures, right? So those all these things are there in the that common pattern tube or the speed recovery pro one, right? So all these are there, then you have to fill them and I will discuss them, right? Okay. Then come to the question number 24. The organisms that respectively excrete ammonia, urea, and uric acid from their body are, right? Those who excrete ammonia, urea, and uric acid from their body. Now we'll come to the tadpole. What it excrete, that is ammonia. No, tadpole is living in water, no, right? Fresh water. So then crow excrete what? Crow excrete uric acid because they are in water. What kind of water shortage? Surroundings, no, that kind of serophytic surroundings. Now birds just, and also birds, they just they are not found in water rich areas, you know. Therefore that is uric acid, and also they need to serve water or just preserve their water content, no, right? Because they are flying long distance and their flying time is high, so therefore they are not able to get water as they will, no, right? Once they are flying, they can't hold for a while and just drink water in the sky and come, no, they need to land, no, they need to go to a water available place to have water, no. So the water is not freely available for them, no, that's why uric acid. Then monkey, it's not that like tadpole that always have available water, but not like birds, right? In the intermediate scenario, then it is urea. So the question, it is asking about ammonia for A, then uric acid for right, what, B, then sorry, urea for C, and uric acid for B. A, C, B in the answer number 2, right? Then 24th, right? It's asking that whether arthropods lack book lungs, right? Arthropods have book lungs. Spiders and scorpions are arthropods, right? They are arthropods, no, but they out in arthropods, they are just kind of special groups, right? Spiders and scorpions are specially mentioned in arthropods. They are arthropods, that is okay, but not all arthropods have book lungs. Out of arthropods, these scorpions and spiders have book lungs. That's the point, right? Okay. So Chandrika, I think Chandrika, no, Chandrika or Chandrika Lienege has asked that one. I think, are you okay with that? No, are you okay with that? Right? Yeah, that arachnids. Okay, yeah, that's correct. Arachnids, right? They are having the book lungs. No, I think you are now okay with that. I think I have explained. Okay, very good. Okay, right. Then come to the question number 25, right? Select the response that correctly shows the part of the human brain and its respective function. Now, act as a relay station for afferent and efferent nerve fibers. It is not the hypothalamus. It is the midbrain. No, the one responsible for this one is the midbrain. So therefore, first one is incorrect. Then second one carries out processes related to sexual behaviors. That is done by the hypothalamus, not by midbrain. So therefore, this one is also incorrect. It has to be hypothalamus. Then third one, then coordination of skeletal muscles is done by cerebellum, not by the medulla oblongata, right? It is done by the cerebellum. So therefore, this is incorrect. Cerebellum. It is done by the cerebellum. Right, okay. Then spinal cord coordinate reflexes, that is correct, no? It coordinate reflexes. Then cerebellum control reflex actions such as vomiting, swallowing, coughing, and sneezing. That is relative to, but not for the cerebellum. Those are done by the medulla oblongata. So then in the Poduratawa tute, if you have gone through, that Poduratawa tute, you can get it from the website. From the website, you can get them. All these tutes for all these free sessions, you can get them from our website, right? So if you have gone through that Poduratawa tute, or that common pattern tute, or that is Speed Recovery Pro one, the same tute, right? Those three names are used for the same one, right? Okay, then that those parts are included there. Okay, then that the relevant structure, and also in the first day, that on Friday, we have gone through some questions also there in that tute. We have discussed some also, right? Okay, so that is what we need to keep it, right? Okay, then 25th. Okay, then 26th. If they just send me a message that whether you have started doing that Poduratawa tute, guys. Uh, have you gone through that one, and have you taken a printout and go through the completing the tute? What about that? What about any update regarding that Poduratawa tute? Okay, have you gone through? It's very just kind of a vital one. Okay, very important and very vital tute. Okay, so I think it includes all the important ones. I think one who goes through that tute definitely right. Sorry, in 25th I have right, coordinates the skeletal muscles, no? That's cerebrum. Okay, sorry, I have missed that. No, cerebellum, that is cerebrum. Okay, thank you very much. Okay, that is cerebrum. Okay, yeah, you can just correct me if there are any mistakes in the discussion during the, because 500 MCQs are discussed, and okay, there can be mistakes taken place. No, it's your kind of a help actually if you pointed out if there are any mistakes from my end. Okay, thank you very much for that. Okay, right. Okay, so that Podurata audio, definitely if you can complete it prior to that session on Friday, sorry, not Friday, Saturday, no, right? Saturday, it will be a quite good, right? It will be quite good if you can complete it prior to Saturday, so then you can go through the discussion quickly, no? Okay, right. Then 26th, which of the following is correct regarding human eyes, no? The action of extrinsic eye muscles that are connected to the eyeball allows convergence. That is correct. No? First one is correct. That is correct. The convex nature of the eye lens is increased when observing near objects, right? Yeah, the near objects. Now convex nature means now eye is also always convex. Now when we looking far, eye is like this. Now when we looking near, what, right, then eye is like that. It means convexity is increased, no? So that is also correct, right? Second one is also correct. No? First one correct, second one is also correct. Third one, ciliary muscles contract when a far away object is observed, right? Ciliary muscles are not contract, right? They are relaxed, right? These are very important. Now what happened to ciliary muscles then? Okay, in near visions and in far vision. Now in the near vision, in the far vision, that ciliary muscles are, they are relaxed, right? They are not contract. Okay, they are not contract. Ciliary muscles are not contract. But in the far vision, right? Okay, in the far vision they are relaxed. Okay, they are relaxed in the far vision. Okay, right. Then what happened? Then in the right now, kind of when we come to the that lens, right? Okay, when you come to the eye lens, this kind of, this is the arrangement in the lens. No, now there you have that suspensory ligaments attached to the lens, and also with the suspensory ligaments attached, there is you can see that ciliary muscle is also here like this. Now this is the ciliary muscle, right? So if you name this is the ciliary muscle. Then in the green you can see suspensory ligaments. these are suspensory ligaments. Then this is the lens, no? This is the lens, right? This is the lens, okay? So this is the arrangement, right? So this is what there. Now, if you need to have a kind of a far away vision, then what is need to be done now? The lens need to be, now lens is in this kind of position, no? Right now you are trying to view far away point. Now what happened is the connexity needs to be decreased to observe the far vision. Then what needs to be done, the lens need to be kind of stretched like this, no? So to stretch that one, suspensory ligaments needs to be contract, no? They need to be contract. Then as a result of that, ciliary muscle is relaxed. That is what happening now. These suspensory ligaments, what happen now to that expand this lens, or the decrease the connexity. Now suspensory ligaments like this, they are just pulling the lens, no? Pulling the lens to increase that, that decrease the connexity, no? To get rays coming from far away, no? Parallel rays which are coming from far away, no? Now what happen in far vision, that light rays are coming like this parallel, from far away. Now if the lens is like this, now lens can't go through the lens, they just bypass the lens, no? That then you can't have the proper vision if the lens is like this. But in near vision, what happen, that light rays are coming like this, no, from the near point. So then connexity increasing is no issue for there, right? But if you are using a near point for this in near vision, just only get in the part of the lens, so that is also not needed. So then you need to increase the connexity like in this near vision, no? So if you need to have far vision, then rays are coming from just far locations like this, no? So if the lens is not convex in that length in if the lens is not covering that light rays coming from far, if it is unable to reduce it in convexity, then it is unable to catch those far objects properly, no? That's what happening in disorders, no? Right? Then okay. Right, then extension of suspensory ligaments increase during near vision, right? Extension of suspensory ligaments, they are decreased actually. This is now this is the near vision scenario, and this is the far vision near scenario, no? In the far, that you can see that green fibers are stretched, but in the near vision, that you can see green fibers are relaxed, no? So the extension of suspensory ligaments are decrease, not increase, right? They are decreases, right? Okay. Then the pupil contracts under low light conditions, right? So what happened to pupil? Then pupil actually not. Then there's another mistake. Also, pupil was not contracting. Actually, what is pupil doing is it's constricting and dilating, right? So pupil is kind of this kind of a structure. Now if we come to eye like this, right? Okay. Now pupil is a kind of a hole here, right? If the hole is increasing its size, we name it as not relaxing, right? We state it as what? We mention it as dilates, right? We mention it as dilates, right? Okay. So what happened? The size of the pupil increasing. Now, if the size of the pupil is reducing, right? If the size of the pupil is reducing, it means now the pupil is just going inside. Now we'll draw it in a separate one. We'll draw it separately. Now the lens is here like this, and the pupil is large here like this. Then what happened? Now pupil is that the size is decreasing. The size of the pupil is decreasing like that, right? Okay. Then what happened to the size of the pupil? Now it is decreases. Now this is known as what? Now this is dilates. Now this is known as constrict, not contract, right? This is constricts, right? This is constricts, right? Okay. Right. Right. Okay. Constricts, right? Okay. This is known as constricts. No. So there, what happened? That size is reducing. So that is what happening, dilates and constricts. So mind then kind of holes like this pupil or any kind of hole in body. So we don't use these phrases, contract and relax. We don't use these phrases for those things, right? So that is also incorrect. So keep in mind. Then could you please introduce the terms in intrinsic and extrinsic if possible? Intrinsic and extrinsic are now, imagine this is the eyeball. Now the eyeball is kind of now this is eyeball, right? This is eye, right? So eye is kind of connected internally and external. Now there are muscles known as inferior rectus, then superior rectus, medial rectus, lateral rectus. Likewise, muscles which are attached externally. Now external muscles are known as extrinsic muscles, right? Intrinsic muscles are known as those now if we suspensory ligaments and ciliary muscles, even though we draw like this, these muscles are arranged inside like this, no? There are some muscles circular rings. Now some muscles are like this, right? Okay, then this is inside the eye, right? This is inside the eye, right? This is inside the eye, right? Okay, then there are some muscles like this. They are arranged like kind of this radiating nature, right? That not circular, right? Okay, so these are once again inside the eye, right? Inside the eye. So therefore those are known as intrinsic muscle, which support the eyeball, actually, not eye means eyeball. They are externally found. They are known as extrinsic muscles. Okay, got it? Herath, did you got that point? Okay, right, fine. That is what regarding the extrinsic and intrinsic muscles. Okay, that is what. Okay. Now, nice. Then these are the things you need to be keep in mind regarding the dilates and constriction. Now the pupil actually it needs to be constricts maybe or under low light. In low light, what happened? Actually, pupil is dilate. No, in low light, pupil is dilate. No, in bright light, what happened? The pupil is constrict. No, in bright light, because if a torch is just directed into our eyes, what happened? That we just reduce the shape of the eye. Once in the darkness, we try to open the eye as much as possible we can. No? So like that's what's happening, right? Okay, right. Then come to the question number 27, right? Okay, that's what up to that question. Okay, it's a good question. Okay, definitely there can be a question from eye, so that can be regarding the eye movements and those controlling of the eye opening and closing, right? 27. Sympathetic nerves leave the base of the brain or the spinal cord, right? Keep in mind sympathetic nerves only from spinal cord, right? Only from spinal. Parasympathetic ones are leaving from the cranium and both spinal cord, from the brain and the spinal cord. The stimulation of the sympathetic part contracts bronchi in lungs, right? What happens? Stimulation, it means stimulating means bronchi needs to increase its increases diameter, no? So to pass to allow passage of more air, no, more air through it. So it needs to what? Not contract. It needs to actually dilate. Actually, it is also not contract. It needs to be constrict. So bronchi is kind of a hole also, no. So there is no contraction relaxation there, right? Bronchi is like this. When the bronchi diameter is increased like this, no, diameter is increased. Now if the diameter is decreased, this is like this, no. So kind of a hole. So then if the whole diameter is increased, we name it as dilate. If the whole diameter is decreased, we name it as constrict, no. Right, okay. Then stimulation of the parasympathetic division inhibits the emptying of bladder, right? So emptying of bladder is happening during the parasympathetic. So parasympathetic means that the normal condition, that we are not in a fight or flight mood, or we are not in a kind of a stress mood. Okay, we are relaxed. So then it leads to emptying of the bladder. But that which inhibits the emptying of bladder is the fight and flight condition, where that is stress, sudden stress condition kind of a that is the not parasympathetic, it is the sympathetic, right? So sympathetic division, right? Okay, it's what inhibits the emptying of the bladder. Acetylcholine is secreted from the parasympathetic part of the nervous system. That is correct. Acetylcholine is secreted from the parasympathetic part. Then adrenaline and noradrenaline, or the epinephrine or norepinephrine, is secreted by the sympathetic part. Okay, sympathetic part. So keep in mind now there are two, sympathetic and then other one is parasympathetic. So parasympathetic secrete ACH, it means acetylcholine. Then epinephrine and norepinephrine is secreted by what? Sympathetic system. So keep in mind, right? Okay. Controls the activity of skeletal muscles. Skeletal muscle activity cannot be controlled by the autonomic nervous system. What autonomic nervous system control is that control two muscles. Those are the smooth. It means involuntary muscles. Is involuntary muscles. Then smooth muscles, and also it controls cardiac muscles, right? So that autonomic nervous system control the smooth and cardiac muscles. Okay, that skeletal muscles are controlled by that motor division of our nervous system. Motor division, not by the that autonomic division, right? Motor division controls the skeletal muscles. Okay, got that point. So therefore 27th answer for the 27th is answer number 4, right? Then come to question number 28. Select the correct combination between hormones and functions, right? PRH is stimulation of secretion of prolactin hormones from the not from the posterior pituitary, it is from the anterior pituitary. It is from the anterior pituitary. Therefore, first one is incorrect. Then ACTH is stimulation of secretion of hormones from the not from the adrenal medulla. Sorry, right? Not from the wait. Wait. Okay, now okay, slight error I think in the share screen, that PDF just started dancing, right? That's what happened. Okay. Then ACTH stimulation of secretion of hormone from the not from the adrenal medulla. It's from the adrenal cortex, right? It is from the adrenal cortex. Therefore, that is incorrect. Then LH, the stimulation of spermatogenesis. Spermatogenesis is stimulated by not by LH. It is by FSH, right? FSH. So therefore, that is incorrect. Then stimulation of milk ejection due to contraction of skeletal muscle, not the skeletal muscles. Milk ejection is due to contraction of smooth muscles. Therefore, this is also incorrect. Then stimulate the protein synthesis, growth hormone that involved in growth. No, so what growth needs? Proteins. No, so proteins is given by the protein synthesis. That is the relationship, right? That's the relationship. Okay. So therefore, answer number 5. 29. Select the correct statement regarding the birth control methods. These are not tested up to now in A level, so they are there is a very possibility. There is a actually kind of a possibility, great amount of possibility to given in this time, right? Okay. Select the correct statement regarding the birth control methods. Okay. Some pills that are consumed by females have only synthetic estrogen in high concentration. Now, these are known as oral contraceptive pills, right? Oral oral contraceptive pills, right? OCP, right? So these are known as oral contraceptive pills. So what they includes, so actually progesterone, right? Not estrogen, they include progesterone, right? So here it's saying that estrogen high amount, that is incorrect. So that only synthetic, if there is stating that only hormone is correct, right? Okay, that is only if there is only certain particular hormone, then that has to be progesterone, not estrogen, right? Not progesterone, that has to be estrogen, right? Okay, so that has to be estrogen, not progesterone, right? So then that's why the first one is incorrect. Vasectomy prevents eggs from entering the uterus. Vasectomy is done for actually for the male, so therefore no relation with eggs or that uterus. So it has to be LRT. LRT is the surgery done for women, okay, for the females, which prevents the egg from entering the uterus. Therefore, second one is incorrect. Actually, vasectomy is done for what? For males, actually, right? It's done for males, right? IUD loop prevents fertilization and the implantation of a fertilized embryo. That is correct, right? That is correct. So it prevents the fertilization and implantation of fertilized embryos. Now if the uterus wall is like this, now endometrium is like this. Endometrium get kind of thicken for the implantation. So what IUD, once IUD is inserted, what happens? This thickening is inhibited, right? So endometrium is not properly get thickened and get prepared for the implantation. So then what happens? Once that ovum or the fertilized egg comes for the implantation, it's not taking place properly. So therefore, then the birth is not continued, right? Okay, right. So that the female is not getting pregnant. Synthetic estrogen is injected under regular intervals in the Depo-Provera. So again in Depo-Provera also synthetic progesterone is what injected in Depo-Provera. What is injected? Progesterone is injected, right? Okay. Then LRT surgery prevents the release of sperms. Once again, release of sperms are prevented by the vasectomy, right? So vasectomy, LRT is for what? For females, right? Vasectomy is for males, right? So keep in mind, right? So answer is third one for the 29th. Right. Then come to the 30th one. What is the correct statement regarding the arrangement of bones in the upper limb, right? Correct statement regarding the arrangement of bones in the upper limb. Now come to the 30th one. Radius move above the ulna during supination. That is correct actually, right? So radius move above the ulna during. Now if we come to our hand, right? So what is happening now, right? What's happening now if we take right kind of arm like this, then now like this, no, now radius is on this side, right? Ulna is on this side now, right? Okay, so this is kind of a supination position, that palm is facing upward. Now what happened in pronation, right? Right, so now we'll see now now we is saying about upper limb bones, no, right? We'll come to the now it's regarding supination, no, right? So they are just asking about the supination position, right? So radius move above the ulna during supination, that is actually incorrect, no? So during the pronation, that radius move above the ulna. So in the supination, the bones are like arranged like this. Now in pronation, what happened now? Now here the palm is facing upward. Now this is the palm, right? So this is the palm. Palm is facing upward, right? Palm is facing upward. Now what is happening in pronation? Now this side goes to like this, no? Now this is thumb, right? So palm is now downwards. Now what happened? Now this is the radius actually, right? Okay. This is the radius bone. Now this is the ulna, right? This is the ulna. Then what happened in the pronation, that radius actually moves through, like it's going upward that process, like actually radius just moves above the ulna. So that is during the not the supination, that is during the actually pronation. No? Pronation. Maybe you can say now during the that pronation. Now from the pronation to supination, that part is difference, right? Okay, so from supination, if we move to the pronation, that is what happening. Now radius is just now as the this is the radius, no? So radius is this one. So radius is going upward, going up from the ulna, no? Right? So ulna is as what is there. Radius will move just kind of above the ulna. No, it has to be pronation, I think, not supination. That is incorrect. The distal end of the radius articulate with four, right? Four carpal bones of the proximal row. So four carpal bones, that is incorrect once again. It has to be three carpal bones, right? Three carpal bones. Then distal ends of the metacarpal bones articulate with the carpal bones. Now distal ends of the metacarpal bones articulate with carpal bones. Now what happened? What happened that with the carpal bones, not the distal ends of metacarpals, it is the proximal ends of metacarpals what is articulates, right, with the carpal bones, right? Then the distal end of the humerus has three articulating surfaces. That is incorrect. Distal end of the humerus has, it has what? It has two articulating surfaces. The joint between the metacarpal bones and the phalanges allow power grip. Now the joint between metacarpal and phalanges allow the power grip. That is correct. No, the joint between the metacarpals and phalanges. Now metacarpal and phalanges, right, allow that power grip. That what precision grip is. There is another grip now. That is correct. That's the power grip. That kind of that grasping thing. That fist. No, fist is making like that. Now there's another grip known as precision grip. Precision grip involves what? It's the because of the thumb, right? Thumb is the main part in precision grip. Like we are holding kind of a that handkerchief, no, a kind of a dirty cloth that we use only the thumb along with the index finger. So that is known as a precision grip. Now here for the power grip, we need the entire palm for the palm to grasp tightly, no, anything like maybe a kind of that. Now we are going in a bus, right? So we need to hold on with the seat or maybe the that that bar in the bus. So we need power grip. So for that we need that junctions between the joint between metacarpal and phalanges, no, they need to be involved, right? That allow the power grip to the proper functioning of the fingers, no, that is correct. Then 30th one done. Then 31st, artificial breeding increase the quality of population. It's not increasing the quality. Actually artificial breeding kind of decrease, no, it is kind of just artificial one means not taking place with the natural parameters, no, right? So we are controlling it, so that quality actually decreasing, right? Second one, can show correlated negative responses. That is correct. Now artificial breeding is the breeding that actually we control it, right? We control it, means we as human being, we control it. So what happen, it's kind of artificial breeding is kind of breeding that's just kind of the same thing, no, the same thing once again is repeated, no, that the offsprings obtained are kind of similar to the parents, no, right? So therefore negative responses means now if they are that parent or that the breeders we used have some kind of negative things, maybe a disorder or something, it will transfer into the offsprings also. So negative responses correlatedly can be associated with artificial breeding. That is correct. Allows natural selection of species. Now, it is if the breeding is artificial, how it can allow natural selection? So therefore, this is incorrect. Produces heterogeneous plants and animals with traits that are desirable for humans. Now, artificial breeding, now we are controlling the breeding. Now, what is the normal breeding? It is the natural breeding. So, if then in the natural breed, sorry, not breeding, in the natural breeding, what happens? We are not controlling anything, right? So that parents are selected naturally, so they are not selected by us, or any similarity between parents, no, that is different. They have natural breeding. They have what? They have genetic variations, no. Genetic variations are allowed, no. Genetic variations take place. So because of genetic variations, right, so what happened? They lead into heterogeneous, no, heterogeneous heterogeneous offsprings, no, heterogeneous offsprings. Now what happens now in what happens in that artificial breeding? There are no, actually, in artificial breeding, there are no what genetic variations, no, no genetic variations in artificial breeding, no, no genetic variations. So no genetic variations mean it lead to homogeneous offsprings, no, homogeneous offsprings, no, right? No genetic variations, homogeneous offspring. So therefore, this is incorrect. Produces homogeneous plants and animals. Reduces the spread of inherited abnormalities. So not reducing. Actually, if there is any inherited abnormalities, now we are continuing with the same generation. No? So continuing with the same generation means we are not allowing this genetic variations to take place, right? So genetic variations are important if right, to reduce if there is any inherited, abnormalities, right, abnormal things. So then what happen? These genetic variations actually are very helpful to decrease or this lower down this inherited abnormalities, no, right? So but in artificial breeding, that we are skipping or we are bypassing these genetic variations. So then inherited abnormalities won't be corrected, they will be continued. So therefore it's not reducing, actually it kind of increase the spread of inherited abnormalities. So therefore, answer for the 31st is second one, right? So artificial breeding is kind of actually kind of, if we think about boilers, no, broilers chicken. So they are similar to each other, no, there are no variations. Everyone is shape similar with the shape, similar with the size, similar with the weight or the mass, right? Okay. Right. Okay. So that is the thing you have to keep in mind. Okay. This is also kind of an important part, might be tested in your exam. Artificial breeding, not given yet, right? In A levels, right? Okay. 30 seconds. What is an importance of DNA replication? Right? Production of daughter organisms that are identical to the mother during sexual reproduction. Now, genetic, the DNA replication is always taken place in asexual reproduction, not during sexual reproduction. Therefore, this is incorrect, right? Allows a species to maintain life, right? So that it means actually DNA replication is aiding in two manner, right? Two manners. Now if we take DNA replication like this, now DNA replication here, so DNA replication is helpful for kind of what, to an organism, I will say, and an individual, I will, right, and also for the species, right? Then for the species, it is helpful to maintain the life. So sorry, for the continuity of the life, right? Or the continuity of the species, no? So for the what happened for an individual, DNA replication is helpful to the maintaining the life, no, right? It is helpful for the individual to maintain the life, for the species to continuity of the life. So that is what important from in DNA replication. What is once again for the species to continuity of the life of the species, and for the individual to maintain the life. It means at that certain particular life, no, right? So allows the species to maintain life. That is incorrect. That is for the individual, right? For an individual to maintain the life. For the individual to maintain the life. Contributes to the continuous existence of a single organism. No, continuous existence of a what? Of a species. Now we have already write down that for a species that continuity is important. Replacing damaged and dead cells by new cells. That is correct, no? That is correct. That is an importance of that DNA replication occurs without any error. You can't say so. There can be errors, right? Errors can be there, right? Errors taken place, right? But it is rare, no, rare errors actually. Rare errors actually, right? If you take kind of rare error means error will be 1 over 10 to the power 10 occurrence, right? Okay. If occurring, if that 10 to the power 10 times occurrence there, then can be one error to 10 to the power 10 time occurrences. It means very rare actually, but errors can be even though it is 1 over 10 to the power 10, it means actually this is now 1 over 10 to the power 10 is not equal to 0, no, right? Not equal to 0. It means there are errors, but the error percentage is very, it means error taken place are very rare errors actually. Error percentage is very low. Error percentage is very low, right? Very low, right? Okay. So that is the point. So errors can be occur, but without any error, that is incorrect. Okay. Then that is the part regarding DNA replication. Once again, another important thing that DNA replication means DNA replication, right? DNA replication, sorry, wait, an error in the PDF, wait, okay. I think an error in the PDF, right? Anyway, okay. So what I have been explained, these are done, no? These are completed by you. Kind of error occur, right? Okay. So now DNA replication completely depend upon mitosis, no? So mitosis is helpful for what? Asexual reproduction. Asexual reproduction, no? If someone need that previous note from 30-second question, he or she can just pause the video in YouTube and go back and complete it, right? Because that was there. Anyway, my editings have left, right? Okay. So this is the concept of DNA replication. It's the mitosis is the what happening and is contribute for the asexual reproduction. So mitosis can lead into regenerations of parts, so maybe then formation of new cells, right? These kind of things, right? Okay. Okay. Then any kind of thing like wound healing. These are the wound healings. These are the importance, right? Okay. So okay, 33rd. Anyway, the answer for the 32nd is fourth one. 33rd. Agarose jelly electrophoresis, right? Okay. Uh, Larger molecules move faster compared to smaller molecules. No, larger molecules move slower, I think, actually, right? Actually, it's moving slower, right? It means that speed is inversely proportional with the size, no? Now, that speed or the speed of an molecule is inversely proportional to its what? Mass. Okay. Now this is mass. I write it mass. Then this is speed, right? This kind of a relationship. Okay. DNA molecules move through the gel towards the cathode. So then here, if we take like two poles like this and this one, and this is go to, then one is negative and one end is positive. Actually, positive one is the anode. The negative one is the cathode. Actually, right? Anode and the cathode. Okay, anode and the cathode. So then DNA, if we take DNA is negatively charged, then what happened? DNA moves towards the anode, right? Okay. So not only DNA, if it is RNA with the charge, they are move into their relative pole, right? Okay, positive or negative. This is the most commonly used technique to separate RNA molecules. No, that is incorrect. This is most used to separate what? DNA molecules. So therefore, third one is incorrect. A strand with a nucleotide that is specific to the strains used cannot be distinguished from others. A strand with a nucleotide that is specific to the strains used cannot be distinguished from others. Actually, that you cannot that is specific, no, with a nucleotide that is specific. So in agrosgel electrophoresis, actually, we can separate. Okay, so we can separate any certain particular DNAs. Now we'll see DNA A, then DNA B, right? Then DNA C. Likewise, we can separate based on their charge and their kind of mass like things. We can separate the DNA from regarding these aspects, but we can't. So in we can separate, right? We can separate, but in agarosteric electrophoresis, we can't specify any kind of what that kind of base order or like thing. We can't specify, right? We can't specify, right? Can't specify any base order kind of thing, right? For that, if we need to specify, we have to go for probes, no? We have to go for probes, right? DNA probes. We have to go for DNA probes, right? Okay. So that's what it's about electrophoresis. So electrophoresis is there to separate any DNA. If you take DNA, it can be RNA, any kind of that polar molecule, a charge molecule. So based on the charge and the mass means the size, right? Okay, mass means the size. Okay, the charge and the size. So that can't specify. If we need to specify any DNA, we need probe, right? So this is with the nucleotide cannot be distinguished, no? So that is correct. Large charge molecules are separated based on mobility within a magnetic field. That is incorrect. So there is a negative charge, there is a positive charge. It means that there is a electric field, not a magnetic field. There is a there is a electric field, right? Okay, so that is incorrect. Okay, got it. Then come to 34th. The biome with the largest temperature variation, no? The biome with the largest temperature variation is the actually deserts, right? Deserts that the desert temperature varies from -30 to +50. So -30 to +50 means there is a delta of 80 degrees Celsius, no? Temperature variation. 35th. That are invasive alien species. Invasive alien species are giant African land snail, water hyacinth, lantana, guinea tana. Right? Okay. Answer is 4. Answer 4. Right? These are very simple theory points. Right? This is a very important MCQ. Very, very important. I think uh, I still now I have gone through so many several question papers. It means provincial papers, school papers. But the thing is, none of the paper include this question. This is a hidden part in environmental biology. But in our papers, we always give this MCQ, right? That's why. Gas or gases that indirectly cause radiative forcing effect, right? Radiative forcing effects. Now keep in mind carbon monoxide, right? Then nitrogen dioxide, okay? Then sulfur dioxide, okay? Then nitrogen oxide, no? Right? Then what? Ozone, right? Ozone in the, especially it is given as ozone in the troposphere, right? Okay, then troposphere ozone. So these are the gases. So these are the gases that cause, right, radiative, indirect radiative force, right? Indirect radiative radiative force, right? Force effect. These things, right? These guys, right? Okay, therefore nitrogen dioxide correct, not methane, not hydrofluorocarbon. So therefore answer for the 36th is, right, answer 1. Got it? Okay, a kind of a very important point, right? Okay, very important point. You might I think so one, the students who going through the live might be the first time. Apart from our class students, they are familiar with these things. I think maybe the first time you are going through that this theory part. Okay, right. 37. 37. All moles have cross-wall called septa, right? Then regarding microorganisms, not all moles, actually many. So you can't say that all have septa, because there are some moles with asepte, right? Asepte, right? So they are not having septa, right? Okay, septa means if we take now if we take filament like this, so there are some divisions like this, no? uh, Between cells there are some divisions like this. There are some holes in the septa also, kind of these kind of holes which transmit substances from one compartment to other. So these separate as unit. This is unit 1, so this is unit 2, this is unit 3, this is unit 4, this is unit 5. Likewise, these are separated as units. No, there are 6 units. So through these pores, through this pores, what happened? Substances move along. No, right? Okay. Some cyanobacteria show sexual reproduction. Mind you, none of the cyanobacteria show sexual reproduction. They are asexual only. Right? Okay. They are asexual only. That's a very important point. Cyanobacteria reproduces asexual only. Nitrogenase enzyme in cyanobacteria is sensitive to, not to carbon dioxide. They are sensitive to oxygen. It is sensitive to oxygen. Mycoplasma and phytoplasma belong to domain, not archaebacteria, belong to domain bacteria. Some algae contribute to intermediate processes in organisms and lichens. That is correct. Some algae contribute to intermediate processes in organisms and lichens. That is correct. They are working close with those organisms, and they are this kind of intermediate in those kind of process, making kind of relationship among organisms in order for achieving in some particular nutrition relationships, right? Okay, so that is what answer number 5 for the 37, right? Then for the 38th, acceptable statement regarding toxigenicity. Exotoxins are thermostable lipopolysaccharides, right? So exotoxins are not thermostable lipopolysaccharide. Thermostable lipopolysaccharides are actually the endotoxins, right? Endotoxins are the thermostable lipopolysaccharides. So when we come to exotoxins, they are actually they are protein. They are made up of proteins, and they are heat liable actually, right? Not stable. They are heat liable, right? They are heat liable. So keep in mind regarding endotoxins versus exotoxins. What endotoxins are heat stable, and they are, it means thermostable, and they are lipopolysaccharides. Then exotoxins are protein compounds, and they are heat liable, right? Not heat stable or thermostable. Cytoxin stimulate the cells in the gastrointestinal tract abnormally. Then what stimulate the gastrointestinal tract abnormally are the enterotoxins, not cytotoxins, right? Enterotoxins are which responsible for this act, okay, or this behavior, right? not cytotoxins. Endotoxins give the same symptoms despite the species, right? Okay, right, that is correct. Then in doesn't mind what is the species, all the endotoxins give the same symptoms, right? Same symptoms. Endotoxins are inactivated by boiling. No, they are what they are heat stable, heat or the thermostable. So therefore, they cannot be inactivated by boiling. Endotoxins are mostly produced by gram negative bacteria. That mostly part is the they are misleading one. So endotoxins are only by negative, only by gram negative, right? So what is that? Endotoxins are produced only by gram negative bacteria, right? Okay, so that is why fifth one is incorrect. Then you come to question number 39. Correct statement regarding immunization vaccines, right? Okay, attenuated vaccines mimic an actual infection, right? Okay, uh, now correct statement will come. Now see, attenuated vaccines mimic an actual infection, right? So there is some there is actually a mistake, right? Okay, so there is, what is the mistake? They are missing that attenuated, that needs to be lively attenuated, right? Lively attenuated. So not lively attenuated part is present, not live phase. Live is not there. Therefore, that is incorrect. A booster dose is required for the vaccine against chickenpox, right? For the chickenpox vaccine, they don't need the booster dose because that is a lively or live attenuated vaccine. Chickenpox vaccine is a, what is that? It is a live attenuated vaccine. Those don't need booster or kind of booster dose or secondary uh, uh, protection, right? Subunit vaccines are used against diphtheria and tetanus. That is correct, no? Right? Okay. Then subunit vaccines contain live organisms whose pathogenicity is controlled. So live microorganisms are once again included in live attenuated vaccines, right? Live attenuated vaccines. Toxoid vaccines are used against rabies, right? Not in rabies, not toxoid vaccines. What is used in rabies? Rabies, they used what? That inactivated vaccines, right? Inactivated vaccines are used again rabies, not the toxoid vaccines, right? Toxoid vaccines are used again this tetanus, right? Okay, and diphtheria, like this, right? Okay, so then that is incorrect. So what is the answer? What's the answer? Answer number 3, no? Right. A vaccine is a very kind of a high yield area. I think maybe a kind of a strong guess for the MCQ, right? Vaccine part, right? A kind of a strong guess might be there in 2025. We'll discuss deeply under that Podurata, that point collection, that the endgame seminar on 9th before the biology MCQ day. No? Prior to that, we'll discuss that. Okay. When come to 40, organisms that cause meningitis in humans, right? Okay. Uh, Streptococcus pneumonia, that is correct. Okay. Then Haemophilus influenza, that is also correct. Then these guys are not contributing for the but meningitis. Then there is another one apart from A and B, that is the Neisseria meningitis, right? Okay, that is Neisseria, right? So very, be very alert on spellings. Neisseria, meninge, meningitis, meningitis, right? So this is the answer. So then A, B, and this. So Neisseria meningitis are not given here. So answer is A and B. Then 41st one. Now we are going to ABCD type questions, right? Select the characteristics that can be in supporting tissues which make up the endoskeleton of many vertebrates. Now keep in mind, endoskeleton is made up of two, no? Once we come to endoskeleton, it is made up of two tissues. That is, one is the bone and the other one is the cartilage, no? Cartilage and bone, no? Right? So endoskeleton is made up of bones and cartilage, bones tissue and cartilage tissue. So then many, so in many, what is the tissue? That is the bone. So you have to be careful. Now questions are in this manner. They are not giving straightforwardly what is the correct statement regarding bone tissue, right? What is the correct statement regarding cartilage tissue? They are not giving like that. They just indirectly give like this, no? Supporting tissue which makes the endoskeleton of many vertebrates. Now that is indirectly given as, so you have to understand, the question itself, it is about the bone tissue, right? It's not about the cartilage or anything, right? It's about the bone. So you have to understand that one and you have to think like that that manner, right? So that is the kind, or that is one kind of giving questions in now modern exam papers, right? So kind of develop one. So first we have to understand what is the tissue focused here. So this is how you have to separate. Now you have to just take pen or pencil in also during the exam. Now endoskeleton, two bones and cartilage. Then many vertebrates, that is the bones, right? So you have to pick that. So mineralized connective tissue, that is correct. Matrix is made up of collagen. Collagen fiber matrix is found in collagen, so therefore that is incorrect. Osteocytes are present in lacunae, that is correct. Each osteon consists of a monocentralized layer, that is correct. Provide support and flexibility. The bone is not provides flexibility, it provides support and strength, no? So therefore not the flexibility, that is incorrect. So this flexibility is given by the cartilage. So then B is for cartilage. Then E is also related with cartilage. So therefore answer is A, C and D, answer number 2, right? Okay. Then fine. Then 42nd, right? Select the option that only contain digestive enzymes secreted from the intestine. Okay. Nucleosidase and aminopeptidase that contain digestive enzymes secreted from the intestine, right? Secreted from the intestine. So therefore first one is correct. All are correct. It means it is asking about the what? It is asking about the intestinal enzymes, no? Intestinal enzymes, right? Intestinal enzymes they are asking. So you have to be keen what? You have to keen on stomach enzymes. You can't include, right? You can't include what? Pancreatic enzymes. You can't include those things. You can't include oral enzymes. It means in the oral cavity, no? So you have to keen on these three types of enzymes. So they can't be included there, right? Then in the B, disaccharide-dase, dipeptidase, phosphatase, all are intestinal enzymes. Chymotrypsin and you see chymotrypsin, dipeptidase, nuclease, all okay, but this chymotrypsin and nuclease, these are what? They are pancreatic enzymes, right? Nuclease and chymotrypsin, so therefore that is incorrect. The nucleotidase, carboxypeptidase and disaccharidase, that is correct. Then amylase, then you come here, amylase, it is a what? Amylase is a pancreatic or maybe oral, no? Pancreatic or maybe oral. Amylase found in both, no? Then that is incorrect, no? Then lipase, dipeptidase, then lipase is okay, right? Intestinal lipase is found, and also pancreatic lipase is also found, so therefore that is okay. Dipeptidase is found in intestine, but the reason is amylase, right? Amylase, it is found in pancreatic enzyme or in oral enzymes, right? Okay, fine. Okay, then answer is A, once again B, A, B, D, this time it is answer number 1. Then correct statement regarding the histological structure of heart, no? The endocardium is continuous with the inner lining of blood vessels. That is correct. Line in the internal structures. Myocardium is made up of cardiac muscles. That is correct. The pericardium is made up of not 3 sacs. It is made up of 2 sacs, right? That is the the serous pericardium and the fibrous pericardium. 3 sacs, it is not 2 sacs. It is the serous pericardium and the fibrous pericardium. Then endocardium is important in the transmission of electrical signals is done by the what? Is done by the myocardium, because myocardium includes all the what that electrical system. It means it includes the that conducting system of the heart, right? Conducting system of the heart, right? Then myocardium lines the chambers of the heart and its valves, right? Not myocardium, which lines the heart and its valves, means that is the interior, no? So interior part is lined by the what? It is lined by the endocardium, no? So therefore that is incorrect. Then A and B answer is 3. Then come to 44th, right? Autoimmune diseases affects female more than males. That is correct. Autoimmune are more prone in females, right, than males. Then diabetes 2 is an example. No, right? Yeah. Sorry. Affect females more than males. Affect females. That is correct. No, right? A is correct. Affect females more than males. Yeah, that's correct. Then diabetes 2 is an example. That is incorrect, because autoimmune disease is the, when we come to autoimmune disease, there are now we'll focus here. Autoimmune disease, there are few. Diabetes 1, diabetes type 1, right, not 2. Then here we come once again, what? Rheumatoid arthritis. Rheumatoid arthritis is there, right? It's a kind of another one, right? Then multiple sclerosis, right? Multiple sclerosis. These are the uh, autoimmune diseases. In diabetes type 1, that beta cells are damaged, which produce insulin, right? In rheumatoid arthritis, that joint capsule, or the joint, I will say joint capsule is damaged, right? Joint capsule is damaged. Then multiple sclerosis, that myelin, right? Or that myelinated axon, right? Axon is damaged, or the myelin sheath in the neuron is damaged. So this is what happened, that our own immune system, these beta cells, joint capsules, and myelin sheath also all are attacked by right, attacked by our own immunity system, right? Our own immunity system just attack these structures. Okay, so some body cells are destroyed due to the activation of cytotoxic T cells. That is correct, because cytotoxic T cells are part of the immunity system. No, now normally cytotoxic T cells or otherwise cytotoxic T, they are involved in invading pathogens, no, destroying pathogens. But here what happened? Cytotoxic T attacking on our own immunity system, no, that our own body, right? Our own body, right? Okay. Then in rheumatic arthritis, the immune system sends antibodies to the lining of joints by mistake. That is also correct, right? In multiple sclerosis, B cells invade the myelin sheath that surrounds neurons. That is not B cells, it is T cells, because T cytotoxic cells are which degenerate or kind of destroy that structures. It means especially beta cells, then joint capsule and the myelin sheath. So that is incorrect. It has to be T, right? Therefore, A, C, and D answer is answer number 2. Then come to 45th. Right, trimester scenario. Heartbeat begins during the 4th week. That is correct, right? Okay, that is correct. A high growth rate can be observed in the fetus during the third trimester. That is also correct. That's why when the fetus becoming too large and the movements becoming decrease in third trimester because the growth rate is high. Then the embryo can be referred to as a fetus during the 6th week. No, you can name that embryo as a fetus in the 8th week, right? In the 8th week, not in the 6th week. So that is incorrect. The length of the fetus during second and third trimesters are 30 and 50 centimeters respectively. That is correct. This is in the second trimester. 50 centimeters is around the third trimester. Got it? Right? Then the fetus is highly active during the third trimester. No, third trimester that the growth rate is highest and the fetus is growing rapidly. Because of that, what happened that inside that embryo sac, no, inside the embryo sac, that fetus is fully filled or fully occupied. That embryo sac is fully occupied by the fetus. Then what happened? Therefore, the space is fully occupied. Then fetus unable to move freely. That the movements of the fetus is restricted, right? Okay, so fetus is highly active, that actually what happened that movements are restricted. Movements are restricted. That is a phrase. Movements are restricted here in the third trimester, right? Okay, so what are the answers? A, then B, and D. Answer number 1. Right? Okay. Then 46th one. Correct statement regarding the joints of human skeletal system is A, right? The elbow joint and hip joint always allow a wide range of movements. Yeah, that is correct. No, elbow and hip joints, because these both are what they are kind of, they are the ball socket type of joints. No, ball socket joints. So socket joints, they show kind of wide range of movements, right? Wide range of movements. Right? Okay. Wide range of movements. They show kind of what they show. We can say that they show adduction, they show abduction, right? Then they show flexion, then they show extension, then they show rotation, then they show what, circumduction, right? Many more, right? Okay, then that is correct. Joints between phalanges allow limited movements, right? Joints between phalanges, yeah, they only actually allow what, flexion and extension only, right? Yeah, that is correct, because only flexion and extension, within phalanges means between fingers, right? So we on fingers only we can do, because of actually between phalanges, right? Joints between, it means interphalangeal joints, not that metacarpal and phalangeal joint. Because of interphalangeal joints, we only can do just flex and extend fingers, that's all. Ball and socket joints and pivot joints allow rotation, right? Yeah, ball and socket joints allow rotations. Then pivot joints also allow rotations. That is also correct. Right. Then pivot joints actually rotation of head actually, right? The head rotates due to the joints formed by the atlas within the vertical ligaments, right? Ligament ring and the odontoid process of the axis. Actually, not a vertical ligament ring, that is a horizontal ligament ring, because this kind of an arrangement. Now, here is the atlas. Atlas is like this, right? Okay. Then what happened in the atlas? There is a kind of articulate facet here, right? Okay. Then what happened? That this axis vertebrae is coming like this. It is like this, right? Okay. So then this is the odontoid process. Okay. This is the odontoid process. Okay, so this is kind of a ring, no, ring-like structure. This is the atlas. So this one is the axis vertebrae. Then what happened? So this kind of attachment, this allow a kind of what? This horizontal ligament, right? Not a vertical. Now this is the ligament, right? Horizontal ligament, right? This is the horizontal ligament. Horizontal, right? Horizontal ligament, right? Because of this horizontal ligament, what is happening? You can tilt head left and right. Okay, you can tilt head left and right. Okay, that is what's happening. Okay, so right. Okay, fine. Uh, okay, right. Then the elbow joints is a hinge joint, right? Sorry. I just show that okay. Sorry. Thank you very much. That elbow joints and hip joints, no? Yeah. I just I just thought that as that elbow as really sorry as shoulder joint, right? Okay. Thank you very much. Thank you very much. Okay. Thank you very much. Okay. Right. Okay. Here there's a mistaken that I took that ball and socket joint. No, that is actually shoulder and right hip. Okay, shoulder and hip. Really sorry. So it is okay for hip joint, right? But for elbow that is incorrect. Because of elbow that is limited. Okay, thank you very much. Some who is the one who has taken that one? I think that Herat, I think. Na? Now okay? Now okay, Herat? Okay, now okay. Just drop a message and everyone, right? Okay. Thank you very much. I misleadingly because of that. I just going fast through the question. I just got that as hip joint and the shoulder joint. Okay. But that what is there in the question? It is regarding the elbow and the hip. Okay. So elbow and the hip. Right, okay. In the pilot, it is saying about shoulder. Okay, anyway. Right, okay. Anyway, no issue. No issue. Don't mind that. Don't mind that because this is according to this that PDF that is here, no. So if it is shoulder now, I think it's actually good, no. Because you are now getting 2 points. Now, if it is shoulder, it is okay. But if it is elbow, it is kind of that limited movements, no. So limited movements can be seen in elbow. Elbow also kind of if we take elbow, then it is kind of a limited movements are shown in elbow, right? Limited movements are seen, right? Limited movements are seen, no? Right? Okay. Anyway, right. Right. Okay. Right. Then, so this is a horizontal ligament that is then incorrect. That D. Then ligaments play a major role in what? In pivot joints. That is correct. So then when comes to answers, B, C, and E are the answers. B, C, and E. Answer number 5. No? Answer number 5. Right. Answer number 5. B, C, E are the answers. Right. Then okay. Then we come to question number 47. Mendelian genetic patterns. Right. So now for that, there's only one thing you have to be noticed, that is a typical domine. You need to draw that particular person. That we do speak, then eyes are normal as a person, right? Nose is there. And I will draw the important parts actually with using a different color, okay? I will use different colors, okay? I will just complete the man like this first, and then I will uh, just include all those. Okay, now fine. Now this is the normal man. So I will include Mendelian factors. Now number one. This is number one. This is what widow speak. Now this is the Number 2, what is this? This is the free earlobes, right? Then number 3, this what is number 3? These are the dimples on cheek, right? Dimples on cheek, number 3. Then this is what happened. This is what is this? This is tongue rolling, no? This is tongue rolling. Then this is number 4, right? Then this, what is this? This is the number 5th, the straight thumb, right? Number 5, straight thumb. Okay, so this is what the typical dominant. This highlight means all these 1 to 3 5 are what they are the dominant characters, right? Dominant ones. So therefore you have to understand, apart from this, all are recessive. If it is given as attached earlobes, then it is recessive. If it is given as hitchhiker's thumb, or without straight thumb, that bending thumb, no, the bent thumb, no, now without straight, now we'll say now bent thumb, it is known as what? Recessive. If it is not having a widow's peak or the straight hairline, then that is what? It is a recessive one, no? So no dimples on cheek means recessive, likewise, right? Fused earlobes is a result of homogeneous dominant condition, right? Okay, yeah, that is a recessive, not a dominant, no? Fused earlobes, if it is say fused, now what is the dominant one? Dominant one is the that free, no? Fused earlobes, so it has to be what? It has to be not dominant, it has to be recessive one if it is fused earlobes. Individuals whose hairline is not directed downwards are all homogeneous recessive, right? So individual whose hairline is not directed downwards, right? Directed downwards are homogeneous recessive. That is correct. That is correct, because now widow's peak, if we come to widow's peak, there are 3 genotypes, no? Capital W, then capital W, simple W, then simple W, simple W, then capital W, and capital W, simple W means he's having widow's peak, no? Widow's peak percent, no? Widow's peak percent. Here simple W means no widow's peak, no? Right? No widow's peak, it means this is homogeneous and this is recessive. So therefore B is correct. We'll discuss this in during the 9th of November session, right? It means in the end game session, right? So dimples in cheek is controlled by a dominant trait. So that is also once again correct, no? C1 is also correct. The thumb falls when the dominant S allele is absent. Yeah, if the dominant S allele is present, thumb is straight, then if it is thumb falls when it is absent, that is correct. The ability to roll the tongue is controlled by not by a recessive trait, it is by a dominant trait. It is that given in the diagram, right? Okay, dominant trait. Okay, so answers are once again B, C, and D. So the answer is answer number 5. Okay, all fine, right? Right, we'll come to question number 48. Which of the following is a Roundup Ready crop or crops, right? Maize is a Roundup Ready, soya bean is a Roundup Ready, beetroot is a Roundup Ready, and canola is a Roundup Ready. So A, C, D, E, A, C, D, E, right? Then answer number, right? Answer number 5, right? These are the Roundup Ready. This is also a kind of a very, very important MCQ, right? So very, very important MCQ. Please go through the part Roundup Ready, right? Roundup Ready part. Will please go through that part, right? This is a kind of a exam target part, right? We'll discuss it in on Friday. Sorry, on Sunday. Prior to that, if you have time, you can go through this, right? Okay. Then when come to 49th, right? Question number 49th. Right. Question number 49. The energy processed by plants in this food web is 10 to the power 5. Now they say the plant have 10 to the power 5 joules. Consider the energy loss due to heat and respiration at each step. The group of organisms that have the same energy. Now for this kind of graphs, what we have to do in food chains, we have to number, no? Now starting from plant, this is 1, so this is 2, this is 3, we'll go this food chain this way, then this is 4, eagle get the 4. Now we'll move on this food chain. Plant to mouse, then 2, then snake 3, then once again eagle 4. Then we'll move towards this one. Plant, grasshopper, frog 3, then snake get 4, and eagle get 5, right? So this is the number. Then we'll move from, we have discussed it in the Poduratava day 1, right? Poduratava day 1, that last Friday session we have discussed it. Okay, we have discussed it, right? Right. Then move towards this one, plant, rabbit 2. Then once again eagle is 3. Then plant, rabbit, fox, fox 3. Plant, rabbit, leopard, leopard 3. Plant, deer 2. Right, okay. Then same energy means same numbers. Now grasshopper will see number 2. Rabbit have 2. That is correct. Snake have number 3 or 4. Then eagle have 3 or 4. No matter 3 or 3. Then 4 or 4. Okay, then it's correct. Mouse have number 2. Snake have number 3 and 4, so there is no 2, therefore that is incorrect. Deer have number 2, mouse have number 2, that is correct. Fox have number 3, right? Eagle have 3, 4 or 5, anything. So 3 and 3 matching, no? Then correct. Then answer for the 49th is fifth one. That answer is A, B, then D and E. Answer number 5, right? Answer number 5. Okay, right. Then come to the question number 50. Right, okay. So this is the step. Now we have to move towards each and every food chain and number from, start from the primary producer. This here the primary producer is the plant, no? So start from the primary producer. For the every food chain, plant is the primary producer. So start from 1 here, then go towards each and every food chain carefully. Goes through each and every food chains, okay? Right, don't miss anyone. So then one like eagle get 3, 4 and 5. Then he get 3 types of trophic levels. Then snake get 2 types of trophic levels, 3 and 4. All the other rest have only 1 trophic level, either 2, either 3, either 4. No? Right? Plant is always at number 1 because it's the beginning or the start of the food chain. No? Right? Then go to question number 50. Select the correct combinations or combination which show instances where microorganisms are used in the commercial production of enzymes, right? Amylase in Aspergillus niger, that is correct, right? Protease is by Aspergillus oryzae, not by Bacillus subtilis. So it is by Aspergillus oryzae, right? Okay. So it is by Aspergillus oryzae. Therefore, B is incorrect, right? C correct. Then once again D correct, E correct. All these are correct. Therefore, answer is A, C, D, E. Then answer number 5, right? So this is what up to question number 50. Okay, drop me a message in discussion and in the YouTube live. Are you okay with the question 50? Now we have discussed 50 questions out of 500. Now we have 50 discussed. It means now we have discussed 10% out of today's work, right? For the question 34, the desert to be biome with the largest temperature variation, doesn't it have to take both the cold desert and hot desert as a single biome? But actually it's not the same place. No. When regarding biomes, the hot desert and the cold desert all belongs to the biome cold desert, no? So what is the name? Halrek, I don't know, it's a username. Anyway, I don't know your name, so I'm answering you. So Halrek, so therefore I just refer your username in the live chat, right? So they are the cold and the that hot deserts or the warm deserts. We all consider them as a single one, right? Single one, right? Okay, single desert. We are considering all those things as uh, single biome, right? So therefore, that's why minus 30 and 50 are taking place, right? That's why we take the deserts with that maximum temperature variation. So anyway, to take a temperature variation, actually in any kind of a biome, there can be, if there is a slight variation or a kind of a heavy variation, there might be two sides. It means either cold side or the warm side, okay, right? In every desert, Now, if we know in Sahara, if you know about Sahara, if you can go through and search in that in the in the noon, in the afternoon, that is very very hot. Even people don't move at that time, right? Or even that those foreigners, they even don't go. But at night, that is very cool. The same desert, no, same desert at two particular instances, right? That's why. Okay. So that's what taken the desert as the answer. Okay. Okay, right. Right. Now one is asking, so what does Bacillus subtilis produce? I need the help from you guys. I think you are now preparing for the exam now. What about right? What about Bacillus subtilis? Quickly send the answer. What that Bacillus subtilis is producing? What does Bacillus subtilis is producing? What is the product or the output by the Bacillus subtilis? Quickly, hurry up. I need the answer. Right? You can drop the message there, right? In the discussion group and also in the YouTube. Just find, go through the resource book and find out what is there, because it might be the MCQ for this year, right? Okay. Amylase, no? Right. Amylase. Okay, then who has asked, right? Okay, Daro, fine. The one who has asked about Bacillus subtilis produce, it is the amylase. Okay, all right. You have to find it, right? It's given in the resource. Very simple. So no need to just hustle back, right? It's the amylase, so you have to go and just find out there. Very simple. It's all given in the resource, right? Okay. Page number 23. That is also given. One has given, right? Okay. Now move with question number 51. Okay. Right. PDF download වෙන කවුරු හරි ඉන්නවා නම් Chrome එකෙන් open කරගන්න. හරි. දැන් එන්න 51. හරි. මම ටග් ගාලා ඉක්මනට රෙඩි කියලා මැසේජ් එකක් දාගෙන යමු හැමෝම. හැමෝම ටග් ගාලා ඉක්මනට. Yeah, antibiotics also. Bacillus subtilis also producing antibiotics, right? That is also correct. Right. Ready එකක් දාගෙන ටග් ගාලා ඉක්මනට. Right? Okay. Quickly put ready. We'll start from 51st. We'll move somewhat quickly, right? Because it is taking somewhat of time, no? Right, okay. Right. Now, first, 51st. Robert Hooke, right, okay. Robert Hooke examined a cork using a compound microscope and used the term cell. Now, what is the error there? That Robert Hooke used a simple microscope, right? Not a compound microscope. He used a simple microscope. So that is the error there. Anton van Leeuwenhoek described unicellular organisms such as paramecium. No, he described about the euglena, no? Euglena and bacteria, right? Not paramecium, euglena and bacteria, right? Euglena and bacteria, right? So therefore, that is incorrect also. Sladen and Chavan put forward the cell theory. So you have to include not only Sladen and Chavan, you have to include Virchow also, right? Virchow also involved in the put in the right that cell theory, right? Then Schwann concluded that animal tissues are also made up of cells, right? Schwann concluded that animal tissues are also made up of cells. That is correct. So fourth one is correct. Then Virchow showed that all cells are right from free cells, existing cells by not by cell differentiation, by cell division, by cell division, not cell differentiation, right? Cell division. Therefore, that is incorrect. So answer for the 51st is fourth one, right? Okay. Then 52nd, the compound that doesn't adhere to the common formula of CNH H2N and ON. That's the deoxyribose because deoxyribose having C5H10 and one oxygen is short, then O4. So therefore, hydrogen to oxygen ratio is like 2.5 to 1, right? Okay, 2.5 to 1. So therefore, 2 is to 1 ratio is not there. So that answer is deoxyribose. All the other CNH, 2, sorry, CN is 2N and ON ratio is there. Okay. Then come to question number 53, the option that gives the resolution powers of human eye, compound light microscope and electron microscope. So first you have to understand about resolution powers of these, right? Compound eye, then compound light microscope, then electron microscope. I will put those values. Then of eye, that is 0.1 millimeters. Then compound light microscope, that is 0.2 micrometers. For electron microscope, 2 nanometers, right? Here in the answers, actually now you have to kind of that convert these things. Now 0.1 millimeter can be given as 100 micrometer. So now in the value, this can be given as 100 micrometer. No? So then this one is incorrect, not 0.001. Then not 10 micrometer, then not 1 meter. So only 1 and 3 can be taken as answers, right? So then come to compound light microscope, we can convert this into 200 nanometers, right? So therefore not 0.1 micrometer, then 200 nanometer, but then electron microscope, not 0.2 nanometer, it is 2 nanometer. Actually, there is a printing mistake. So it has to be 2 nanometers anyway. There is no answer. Then this one is also incorrect. Then answer for the question is actually all, right? But better you have to keep these values on just on knowledge. But mind, I don't study. Sorry. Right. I will just use another color here. Don't study that values inside these brackets, right? These are kind of conversions. These are physics, right? So millimeters into micrometers, then micrometers into nanometers. These are physics, right? This is not right. This is not biology, right? So therefore, these are kind of just creative questions. So you have to keep in mind now. I 0.1 millimeters, compound light microscope 0.2 micrometers, then electron microscope 2 nanometers. That's all, right? Okay, right. 54th. When you come to question number 54, right? Which of the following is correct regarding the cell wall found in right plants? It is an intracellular structure. No, cell wall is a what? It's a extracellular structure, right? Extracellular structure, right? Then component of the, not the symplast pathway, component of the apoplast pathway. Then middle lamella is present inner to the primary cell wall, right? Okay. Middle lamella is present outer to the primary cell wall. Now arrangement of the, if we take the arrangement of the cell wall like this, now I will take like this, that cell wall. Now middle lamella is here. Now if we take another color, I will take a primary cell wall like this. I will take a cell wall with secondary cell wall also. Then secondary cell wall is like this, and also will finally ending up with will end up with plasma membrane like this. Now, so this is the arrangement. Now if we name this, now this is the middle lamella. This is the middle lamella. Then blue color portion, so blue color portion is the it means this one, right? This one. This is the what? Primary cell wall. Primary cell wall is this, and this black color one is what? This is the secondary cell wall, right? Secondary cell wall. Then blue color one, this is the, sorry, we'll write it on other side once again. It will name it as this, right? This is the plasma membrane. So now this is the interior, this is the interior side of this. It means this is the, now this is the cytoplasm, right? Cytoplasm. So from this is the inner side, and this is the outer side. So from interior to exterior, this is the layers. These are the layers passing from plasma membrane to primary cell wall, sorry, secondary cell wall, then primary cell wall and middle lamella. So this is the order, right? So keen to draw this diagram, and this is a kind of a very important part, right? In MCQ, you can be given. Right? Okay. So that is what here, right? Okay. Fine. Any trouble or any issue with the volume, students, please? Any issue with the volume? No. Okay, it's okay. Right, all okay, no? Right. Maybe your device issue, anything like that. Okay, right. I just speak a bit little slow, right? Okay, less volume. Okay, I will increase my volume, right? Not the volume of the mic. I think it's okay. I will speak somewhat louder. Okay, anyway. Online sessions, I can't adjust the volume. No, this you have to say it, because in physical classes, along with the students there, you have to speak louder. But now if I realize that I can hear myself, then I think it's okay. Anyway. Then middle lamella is present inner to the primary cell wall. So then middle lamella is what? It's exterior, no? It's not inner. It is outer to the primary cell wall. Then that is incorrect, right? Okay. The primary cell wall is composed of cellulose fibers that are unevenly distributed throughout the cytoskeleton. That is correct, right? Okay. That sorry, through distributed, not cytoskeleton. That is the I didn't realize that not cytoskeleton. It has to be ACM, no, extracellular matrix, no. So the primary cell wall is composed of cellulose fibers that are unevenly distributed throughout the extracellular matrix, not cytoskeleton, because cytoskeleton is not the part, right, which where the cytoskeleton actually in relationship with the plasma membrane, right? Cytoskeleton is with relationship with the plasma membrane. So that's the part, right? Okay. Okay. Right. Then the secondary cell wall is laid between the plasma membrane and primary cell wall. We'll see. Secondary cell wall, the black color portion, yeah, it's between the blue color portion and the green line, no? It is, yeah, it's between the primary cell wall and the plasma membrane. So that is correct. Answer is fifth one. Right. That's it, right? Then we'll move on to the question number 55. Okay. Then consider the following process in the cell cycle. Once again, I'm telling, don't be frustrated if you take these questions and when you are trying to answer these questions, you will find, oh, what the hell? How can I answer? I don't know what is the phase, what these things are taking place for. But don't get disappointed. Now, there is that Poduratava tune, that common pattern tune. Once again and again, I am saying the same thing as a fool, right? I think sometimes the students who are working and going along with the with the Poduratava tute might get me as a fool because I'm telling the same thing. But I'm telling this same thing because there can be students who are hopeless at this time. So the only thing you can do is just take your resource book and complete that Poduratava, and I will also discuss that Poduratava tute, right, with you. So therefore, then you have to complete it by yourself prior to the discussion. So that will immensely boost your MCQ marks, right? Okay. So definitely, so that will be a great help for your MCQ marks. So please go through that, right? It's kind of a pleading, right? I'm pleading, right? Okay. It's not kind of warning, it's pleading because actually that will increase your marks. That's it, right? Okay. Then question number 55. Chromosomes get even more condensed. Now we have to find the phase relevant to this. Then this is pro-meta phase. No, this is happening in pro-meta phase. Then cystochromatically separate the centromere. This happen in anaphase, right? Okay. Then C, formation of matrix spindle begins at the this is at the prophase. Then chromosomes become less condensed to form chromatin. This is taking place. This is happening at the telophase, right? Okay. Then centrosomes reach the opposite poles. This is at sorry. This is at metaphase. Right? This is at metaphase. So those are the things. Then correct sequence from pro to then correct sequence means this had to be the number one, no? Prophase, then pro metaphase 2, then metaphase 3, then anaphase 4, then telophase 5. Then what will be the order? Order needs to be C, then A, then E, then B, then D. C, A, B, D. Then answer number 2, right? Answer number 2. So you have to find out like this in this kind of MCQ. What you have to do, you have to figure out the scenario or the situation and write down in front what is the phase belonging to a certain particular scenario, no, incident. Then take that out and then go through the question, no? Find the order, right? Which of the following is correct regarding the C4 pathway? No, that C4 is tested more frequently in our papers and in these 500 MCQs. The reason is that it's a kind of a target part, right? Target part, right? Okay. Then question number 56. CO2 fixation occurs first in bundle sheath cells, right? CO2 fixation occurs first in bundle sheath cells. That is incorrect. CO2 fixation occurs first in what? It is first in mesophyll cells, no? Then from mesophyll cells, it move into the bundle sheath cells, right? Malate is a more stable compound than oxaloacetate. Malate is, yes, more stable than oxaloacetate. That is correct, right? PP has a lower affinity for oxygen, right? Actually, PP has not lower affinity. PFE has no affinity, not lower even. It is no affinity. PS2 is absent in bundle sheath cells, right? PS2 we can't say that absent. We can directly say that PS2 is absent in bundle sheath cells, right? That kind of yeah, there can be instances where absence of PS2, but actually PS2 is that size is somewhat smaller, maybe say that size is smaller or that minimally available, right? Like that kind of a thing that regarding the PS2, right? Okay, that size is smaller is what given, no? PS2 size is smaller, no? Size is smaller, it means actually you can say that they are that kind of differentiation is less differentiated, no, less differentiated, no, less differentiated. Kind of, we can say once again on another phrase, we can say that they are that kind of limited, no, they are limited there, no, limited, no. So you can say that PS2 is absent even, that actually there are conditions that PS2 may be unavailable. There can be this kind of scenario also, right? But so now second one is most appropriate when considering with fourth one. Second one is most suitable, no? If second one is incorrect, then fourth one may be, sorry, that fourth one maybe they are just trying to get the that answer. Anyway, second one is there. PP reacts with CO2 faster than bicarbonate. No, PP reacts faster with then if it is, then there is PP, then there is bicarbonate, and there is carbon dioxide. PP goes towards bicarbonate more, right? Then just one type carbon dioxide likewise, right? But mind you, this bicarbonate and CO2 scenario was not there after 2022 update, right? So they are not included that now, not in your resource book maybe. So just keep in mind that PP has more affinity toward bicarbonate than CO2, but I think that is not available in your resource book right now. Okay. So therefore that is incorrect. Then we will move to question number 57, right? Pyruvate can break down further in absence of molecular oxygen, cannot be break down further, right? Cannot be break down, sorry, cannot be break down further. So therefore it leads to what, with the absence of molecular oxygen, it leads into what, is lead into two pathways. One is ethyl alcohol fermentation, right? Then one other one is lactic acid fermentation. No, all bacteria carry out ethanol fermentation. That is incorrect. Not all, actually many bacteria. No, limiting NAD plus allow the recycling of NADH and increase the ability to make use of NAD plus. Yeah, that is correct, right? Okay. Then what happened actually now? This is what happening now. Now glucose is there. Glucose is converted into pyruvate, right? Then in pyruvate then goes through what? Converted into acetyl CoA. Then acetyl CoA is transport into what? TCA cycle. No. In the TCA cycle, what happens? NADH produced. Then what happens? FADH2 produced. Then what happens? Once again, ATPs are produced, right? Then anaerobic, right? Anaerobic means all these processes not taking place, no, right? In anaerobic, right? In anaerobic condition, these are not taken place. It means what happened? NADH is minimized, no? NADH is less, right? NADH become decrease. You got it? Now that point, right? Okay. NADH become minimized, right? So what happened in this step? Sorry. In this step, what happened? That glucose into pyruvate, that NAD plus producing NADH, right? Okay, right. So to produce this what? Then energies. This is what happened. Then once again, this not stopped by this. Then TCA cycle move into what? Electron transport chain, no, ETC. In ETC, what's happening? In ETC, what's happening? You can further write down, right? In ETC, what's happening? This NADH is converted into NAD plus, right? Similarly, FADH2 is converted into FAD plus. Then likewise. So here what happened? NAD is increasing, no? NADS is increasing with the electron transport chain is taken place. Now for electron transport to be chain to be taken place, what need? Then in anaerobic conditions, then this one is also halted. What? In anaerobic condition, right? Got it? So then what happened now here? NADH production is decreasing. Meanwhile, what happened here? Because of Na, that electron transport chain is halted, NAD plus production is gradually decreasing. Now what happened? This produced actually. Now I will like this connect this produced NAD plus is transmit into the here in the that NAD plus produced in the electron transport chain with the oxidation of NADH, that NAD plus is moved into the that glycolysis, no, to produce NADH. Then what happened? NAD plus become limited because this process is halted, no, that electron transport is halted, therefore NAD plus is limited. Then cell need to find a way to produce NAD plus, right? Then that's the process. Then cell need to find a way to then cell need to find a way to produce NAD plus. Then that's why it's go to what? It's moving to the anaerobic pathway, right? Maybe I will write lactate here, lactate, or maybe ethyl alcohol. Now, if it is ethyl alcohol, there is intermediate process known as acetaldehyde. I just write lactate and ethyl alcohol. What is happening here? Now, what is happening here, right? That what is happening here now? NADH produced here will be given, and by this NADH, NAD plus is produced. So this NAD plus is transmitted here, right? So because of that NAD plus limiting as a result of halting of electron transport chain, so electron transport chain is not furthermore taken place, right? So as a result of that, what happened? NAD plus shortage arises. As a result of that NADH shortage, that pyruvate is converted into lactate or maybe ethyl alcohol in order to regenerate NAD plus. That is what happening. Those who have got the point, please drop a message quickly. Those who have got the point, please drop a message. Those who have got the point, please. Got it? A very important fact, right? Right, okay. That's a fine, nice point. Huh? Right. That is what happening, okay? Right. Then we'll move on to the question, right? So therefore, limiting NAD plus allows the recycling of NADH and increase the appeal to make use of NAD plus. That is correct, no? Third one is correct, right? So pyruvate can't break down in anaerobic conditions because pyruvate to convert into acetyl CoA and TCA, they need oxygen. That's the part. That's the thing. Without oxygen, they can't go through that pathway. That's it. So then this pathway, this entire pathway is halted from acetyl CoA to TCA to ETC halted. Then it's moving to this side. That's right. Okay. That's the point. Okay. Okay. Right. Then in lactic acid fermentation, pyruvate is directly oxidized to lactic acid. Now see what will happen. Now pyruvate to lactate, no, right? Now we'll see pyruvate to lactate what happened, right? So here that point on we discussed on Friday is important. Now here pyruvate is there, pyruvate, and it is transmitting to what? Lactate. I will say lactic acid here. Now what happened, right? Yeah, I just deliberately missed the just didn't write the acetaldehyde. Okay, I deliberately didn't write the conversion to acetaldehyde. Acetaldehyde is kind of an intermediate, no? It is not relative to that MCQ point, okay? we'll discuss this anaerobic respiration furthermore during that Sunday discussion, right? Okay, point discussion. So no need to just hustle things, right here. Then pyruvate to lactic acid means what happened here, that NADH is converted into NAD plus. This is what happening, no? Right? So if we write this as a single equation, then pyruvate plus NADH, then lactic acid plus NAD plus. What is happening here? Now NADH has a 0 oxidation number, then NAD plus have positive 1, no? Right? Then 0 to plus 1 means there is oxidation, right? It means if there is a oxidation, then there is a vice versa reduction. Then pyruvate to lactic acid is a reduction. This is what you have to keep understand, right? But this is what, if you write this as a reaction now, so NADH into NAD plus means it has oxidized, then pyruvate to lactic acid, then if that NADH has oxidized, then this other reactant, it is reduced, no? Right? Then we'll see, in lactic acid fermentation, pyruvate is directly not oxidized, it is reduced. This is what here mentioned, no? Pyruvate is reduced, not oxidized, right? Therefore that fourth statement is incorrect, right? Then in the first step of ethanol fermentation, pyruvate is converted into acetaldehyde by releasing 2 molecules of carbon dioxide. Now it is given here, right? What about that conversion? Now here what is happening in, I will write it here. Now this is what happening, glucose to pyruvate, this is the normal glycol, right? Normal glycolysis, right? Pyruvate, then it produces 2 pyruvate molecules actually, right? Then pyruvate is without the availability of absence, it means in the absence of O2, it can go to 2 pathways, right? One, it can convert into lactic acid, right? It means they get only 1 pyruvate molecule, right? So 1 pyruvate, if it is 2 pyruvate molecule produced, they only get from 1 pyruvate that lactic acid is producing, right? Okay, right? From 1 pyruvate only, not from 2 pyruvate, right? From only 1 pyruvate is used here, or whether it can convert in with the availability in or the type of organism, it can be converted into what, that ethyl alcohol, right? I will write ethanol. So this is very important. In lactic acid, it is pyruvate is converted directly into lactic acid. Okay, directly into lactic acid. Then there what happen? NADH is utilized and converted into NAD plus. Here what happen? Acetaldehyde into ethanol. Then there is an intermediate step. No, now acetaldehyde to ethanol procedure, that NADH is converted into NAD plus. Got it? Now prior to that, there is a removal of carbon dioxide molecule. Now pyruvate is carbon 3. Pyruvate is a carbon 3 compound. Acetaldehyde is a carbon 2 compound. Ethanol once again a carbon 2 compound. Then there is a removal of carbon dioxide, but it is 1 carbon dioxide. Okay, right? Not 2 carbon dioxide molecules. Therefore, that's why 15th statement is incorrect. Right? That's why 15th statement is incorrect, right? Not two carbon dioxide molecules, it is 1 carbon dioxide molecules. All right, okay, students? All right, okay, nice, right? That is what happening. You can note it down. You can pause the video and take a screenshot, or you can complete it with your notebook, right? No issue. This is the scenario. There is no then what you have to understand, there is no carbon dioxide release in lactic acid fermentation. When the carbon dioxide is releasing, we name it as decarboxylation, right? We name it as decarboxylation, that process. So therefore, no decarboxylation in lactic acid. There is decarboxylation in ethanol production or ethyl alcohol fermentation. Now I think, okay, that is one student who had asked earlier about acetaldehyde, what's happening. That's what happening here. Therefore, this is also incorrect. Answer is third one. Now we'll come to 58th. The most successful group of animals on earth with highest number of species, that is the arthropods, right? Arthropods. Those are very simple facts, right? Okay, very simple facts. Consider the following classes that belong to phylum Chordata, right? Then Chondrichthyes, Ostricthyes, Amphibia, and Reptilia, the organisms that show external fertilization. We'll see now. In Chondrichthyes, actually they show internal, right? Then Ostricthyes, they show internal and external. Then Amphibia, they show external. And Reptilia, they show internal. This is what is belong. Then the organisms show external fertilization now. Ostricthyes and Amphibia, it means B and C. There is no such, no? Right? There is no such answer. So therefore, actually that is all, no? Once again, that is all. Answers are B and C. Right. Okay, got it, students, right? So Chondrichthyes only show internal, right? Ostricthyes are those who show both internal and external. Then Amphibia external, Reptilia internal, right? So therefore answer is B and C. There is no such answer. Therefore answer is all, right? Okay. Got it? Why that's become all, but don't go through. Just depend on the answer of the question. Just get why that answer has become B and C. This is the theory part. This is what you have to be keen on. Who shows internal? Who shows external? Right? Right. Okay. In pilot paper, there is an answer and B and C. Okay, then you can get it. Now I am referring to this document. Okay, Daru, right? So the students who are there on the class, just go through with your pilot papers. No issue. I am just here going through with this document, right? This particular document. Okay. Okay. Nice. Okay, you are good. You only get the point. Okay, right? Answer maybe depend on the paper itself, because those papers in the pilot paper, I think in the corrections will be have been done in the exam, right? Okay, for your paper. Then 60. Right. The phylum of plants that is evolutionally closer to seed plants, right? So now when we write the order, now what we know, what we say that evolutionary order, when we write it, so we have to write it like this, no? We have to start from hepatophytes, right? So hepatophyta, then bryophyta, right? uh, right, then no, we can't consider that breaking down of pyruvate as that lactic, the conversion of pyruvate to ethanol and lactic acid is not a breaking down of pyruvate, right? The breaking down of pyruvate is the conversion of it into acetyl CoA and furthermore down to citric acid cycle and electron transport chain, right? Then bryophyta, hepatophyta, bryophyta, then anthocerophyta, then lycophyta, then pterophyta, then Cycadophyta, then Coniferophyta, then Neotophyta, then Anthophyta. This is the order. So these are kind of divisions actually. Now here this is another one division, here another is another division. Now these we can say this as what, this as we can say as what, non-vascular plants, no? And also we can divide this into once again one more. Now we can say completely these are seed, we can give right, all these as seed plants, but seed plants, sorry here, uh, right? I will write like this. Now, these are all seed plants. These are seedless plants, right? And we can furthermore divide. These are gymnosperms, and this is angiosperms, right? And further we can write these all as what? Vascular plants. Got it, right? This is the division. So closer to seed plants means this one. Closer to seed plants means we have to get this point. This is the transition between seed to seedless to seed plants. No, this is the transmission. Then closer to seed plants is the pterophyta. Then answer is uh, 5, right? Answer is 5, right? Okay, got it. Now keeping the order in mind and write it down anywhere, right? Because most students just don't get the order correctly. They just write bryophyta first, then pterophyta over the lycophyta. That is incorrect. This is the correct. This is the evolutionary order actually, right? So this is the evolutionary order from top to bottom, right? This is the correct evolutionary order. So this is a very good short note kind of thing, right? This is a evolutionary order, so don't miss this, right? This is the evolutionary order. This is very important, very important. If you don't know this order, you might not able to find out the correct answer. You might miss the correct answer, right? That is quite unfortunate, because just a small theory point, no? Just need to keep the that order in the mind. So if you forget the order, that's really devastating, right? Okay, right. Now just write the order, right? Okay, write down the order. Now 61. Consider the following features. Wall spores produced by sporangia, multicellular gametogenia, dependent embryo, presence of apical meristem. Features which are absent in green plants, green algae, and present in terrestrial plants, right? Then these all are specific or can be found in green plants, or can be found in terrestrial plants, actually not in green algae, right? Then none of these in green algae, actually. It means all are found in terrestrial plants. So therefore answer is A, B, C and D. Answer number 5, right? Very simple one, very simple one. But if it is asking a question that found in both green algae and that terrestrial plants, you can write presence of cellulose in cell wall, right? Starch as the stored food, right? Then what? Chlorophyll pigments presence, right? Then ability of photosynthesis, right? Those are common for both. Common for both, right? Common for both means that green algae and terrestrial plants, right? Common for both, that cellulose cell wall, starch as stored food, then chlorophyll pigments, then photosynthesis, right? Nice. The 62nd one, correct statement regarding vessel elements, right? 62nd, right? Okay. Slender long cells with tapered ends. Now vessel elements are like this. They are not slender. They are some short and containing that water, pose for water transport. Perforated plates are present. And when comes to tracheids, they are the tapered and elongated, right? Then long cells with tapered ends. These are tracheids, right? Tracheids are like this. Then this is vessel element. Vessel element is like this, right? They are short and that kind of thick and this broad, no, right? Then this is incorrect. Then second one, right? Process thin walls. Yeah, they possess thin walls in order to transmute. It means transport more water, no? Right? It means that more substance can be flown through this, no? Therefore, thin walls are present. That is correct. Found in all vascular plants. That is correct. Incorrect. Right? Not found in all vascular plants. That vessel element tracheids found in all vascular, right? All vascular tracheids are found, but vessel element is found in that all angiosperms. It means all flowering plants and Neophyta. Neatum. I will write the plant neatum, right? Okay. Pits are present in cross walls that binds with each other. Not pits, they are present. Perforated plates. Perforated plates are found, right? Pits are found in tracheids. These are known as perforated plates. These are the perforated plates, right? Okay. These are that kind of pores. Primary cell wall are thickened by lignin. No. Secondary cell wall is which is thickened. Not primary cell wall, secondary, right? Secondary one is which containing lignin. So that is incorrect. Then we'll move to 63rd, right? 63rd, right? Okay, fine. Question number 63. Which of the following statement is correct regarding the structure and function of stomata? Right? Structure and function of stomata. The inner cellulose wall has a lower thickness than the outer cellulose wall. Now regarding the stomata, it's like this, no? Now it's like this. Now inner wall is quite thick actually than the external wall, right? Inner wall is more thicker, right? It is more thick. It is more thick compared to external wall. Internal wall is thick, right? Compared with external, right? Internal wall is thicker. Then there are cellulose microfibrils found likewise, and there is also vacuole there inside. And there are chloroplasts are found in gland cells. Chloroplast can be seen likewise, right? Okay. Lower thickness, that is incorrect. It has more thickness. There are cellulose microfibrils that form inelastic hoops around guard cells, right? So that is correct, right? There are cellulose microfibrils that form inelastic hoops. That is correct because they are just allowing now if there are no now these are the cellulose microfibrils, right? These now I just drawing them. These are cellulose microfibrils. So then what they are doing now, if they are not cellulose microfibrils, that stomata will expand like this, no, with their liking like this. Now it's very ugly, no, right? Very ugly and very incomplete. With the cellulose microfibrils present, that elongation, the expansion is kind of a regular, no, that they are maintaining this length, that certain particular length of L is maintained throughout the expansion, right? Once again, that L is length is maintained. Now here what happened, L dash now kind of very irregular, no? It's very irregular kind of expansion, no? That is in, but these cellulose loops are they are inelastic, that allowing this kind of expansion. Okay, got it. Right. During the daytime, right. Okay. During the daytime, K plus actively enters guard cells from neighboring epidermal cells. During daytime, K plus actively enters guard cells from neighboring epidermal cells. Right, okay. Then what happened? Yeah, that is correct. Now in the daytime, what happened? Now there is stomata is here. Likewise, then neighboring epidermal cells are there. Then K plus they actively enters to the guard cells, right? That is correct. During daytime, K plus actively enters guard cells from neighboring epidermal cells, right? Stomata opens due to the decrease of carbon dioxide in the substomatal cavity. Yeah, substomatal cavity means that now if it is like this now plant leaf, now we draw the kind of this is the epithelium. Now this is the stomata. These are the adjacent epithelial epidermal cells, right? Epidermis is the epidermis actually. Then around the epidermis, there is a cavity like this. This is called as the substomatal cavity, right? Substomatal cavity. So when there is carbon dioxide is low, that it tends into opening. Then after the opening, what happen? Carbon dioxide will enter inside the stomatal opening. That will happen. That's what will happen. So then in the as is stated in the sentence, stomata opens due to decrease of carbon dioxide in the substomatal cavity. That is correct. The daily rhythm of opening and closing of stomata is governed by the internal clock of the neighboring epidermal cells. Keep in mind, there is a clock, actually kind of a clock, right? I will just note it like this. There's a kind of a clock inside. This is known as the internal clock. Internal clock actually sense whether that is daytime or whether it is night time. No? So it senses for the stomata whether it is daytime or whether it is night time. So that internal clock is found in the what? In the guard cells, right? Not in the epidermal cells, right? So therefore, that what is the mistake there? All are correct. So internal clock of the neighboring epidermal cells. I mean internal clock in the guard cells, right? Therefore, that is incorrect. Then answers there are 3 actually. 2, 3, 4, all are correct. Right. Then 64. Correct statement regarding secondary woody plants. The thickness of the secondary xylem in warm and wet growth period is higher than in normal, relatively in actually in warm wet growth periods actually. It's higher than the normal growth periods. Yeah, thickness is higher, right? Thickness is higher because they have to be adapted into that conditions than the normal, no? That is correct actually. First one almost correct. Xylem vessel in spring would have small pores and thick walls, right? What happened? Right? Large lumen and they consist of thick walls actually, right? So they xylem vessels in spring. Now spring wood, right? Okay. Okay. Xylem vessels in spring would have small pores and thick pores, right? Okay, then there are spring wood and right. Okay. Then right. Okay, spring wood. Then now we take to spring wood. Okay. Then what happened? They have small pores and thick pores. That is incorrect because actually they have thick walls, right? That is correct. They have thick walls. Then also they have large lumen, right? So large lumen. So that is what among spring wood, right? Okay. So therefore that is incorrect. Xylem vessel cannot be seen in hardwood. Xylem vessel is actually found in the hardwood, right? So they cannot be seen in softwood. So if we take hardwood and softwood, then hardwood and softwood. So how we categorize these two? Hardwood are the wood with they are with xylem vessel, no? With xylem vessel, right? Then softwood is the wood without xylem vessels. How that is how we categorize it, right? Wood without xylem vessel, right? Wood without xylem vessel is the softwood, right? So keep it in mind. So that is incorrect. Primary xylem tissue that is present outside the hardwood and still transport xylem sap is known as sapwood, right? Now, okay. Now, primary xylem tissue that is present outside hardwood and still transport xylem sap is known as sapwood. Now, remember now in a stem, right? If we take this stem, in the middle there is what? Now don't not just frustrate with hardwood and softwood, right? There are two combinations, right? One is hard and soft. That is based on the xylem vessel, right? Another one is that is hard and sapwood. That is based with the transportation of substances, right? That hardwood is that dark color one, and sapwood is the that light color xylem, right? Okay, light color xylem. So then the middle there is hardwood, and in the outer world there is what? There is sap. Now this is hard. This is hardwood. Now this is sapwood, right? That is the point, right? Hard and the sapwood, right? So hardwood is in the center. It's in the center. So primary xylem tissue that is present outside the hardwood and still transports xylem sap if known as sapwood. Now in the sapwood there are xylem sap, but in present, right? In hardwood there is no xylem sap like that. Okay? There is no xylem sap, right? Okay, right? Hardwood doesn't transport substances, right? Hardwood doesn't transport substances. It is thickened by pigments and thickened by different types of substances. Okay, so then it is not transporting anymore. But sapwood is transport xylem sap, right? Okay, fine. So then that is about heart and the sapwood. This hardwood is this dark color pigmented area. The sapwood is that non-pigmented, that pale color, the light color area, right? So it contains xylem sap. So but keep in mind that both the sap and the hardwood, they are not primary xylems, they are secondary xylems, right? Then they are what? They are secondary xylems, right? Not primary xylems. That is the point. Okay. Older as well as newer secondary phloem contribute to phloem translocation. Mind you that only the new secondary phloem is contribute to the phloem translocation, not older one, right? Only the new one, right? For the phloem translocation, actually, what is contributing? So for the phloem translocation, only new one is only new one, right? Only new one is contributing for the translocation, not the old one, because old one is now broken down and no functionality can be seen there. So that is what. Okay, got it. Okay, right. So that's a good MCQ. Might be given in the 2025 because it's not tested still in the A level, right? A level exam. So that's the kind of a vital fact there. So they are for 64. That please refer these parts, right? Please refer these parts in the resource, and I will go through that in the 9th of November in the that endgame seminar. Please refer these things, right? Springwood, then that spring period, right? Okay, springwood, then hard and sap wood, then hard and sorry, then hard and softwood, right? Okay, these things. Okay, so these keep in mind, right? Okay, keep in mind and go through these parts and just go through what is there. And actually I highlighted and just go analyze the main aspects around that hardwood and softwood. That hardwood is with xylem, softwood is without xylem. Heart and sap. Heart means dark color portion in the middle of the stem. Light color portion outside the middle is the sapwood, right? So sapwood still transport xylem sap. It means it is living, kind of, not living actually. It's functionally active. But hardwood is, there is no xylem sap transport. It's functionally actually active. It's harder. It's darker. All those things, right? Okay. Mature. Okay. Right. Then come to the 65. Nutritional requirements of the plant. Sodium is an essential element for plants. Actually, now sodium. When we come to sodium, sodium is not an essential, right? It's not an essential. It's non-essential, right? Non-essential for plants, right? Sodium is non-essential. Nickel is a component of middle lamella of plant cell wall. In the middle lamella, there is actually no nickel. It is calcium plus magnesium along with pectin, right? Then calcium, magnesium, pectate is the component. Crinkling of leaf is a symptom of not chlorine deficiency. It's a symptom of what? Zinc deficiency. Zinc. copper act as a component of some enzymes. That is correct, because copper act as coenzymes and cofactors. Sorry, cofactors, not coenzyme. Act as cofactors. That is correct. Zinc is a component of chlorophyll, right? No, components of chlorophyll, keep in mind CHON. I just give a phrase mage, CHON mage, likewise, right? CHON mage, it means CHON, right? And plus magnesium, right? That you can understand, you can keep in mind the CHON. Always we keep in mind of CHON, then mage means magnesium, right? Then what are the components? These are the components of chlorophyll. Don't frustrate or don't that's misunderstand that zinc is involved in chlorophyll synthesis, right? Chlorophyll synthesis, it's one thing, right? But if one is involved in the synthesis, it's not suggesting that it is a component, no, right? So components are CHON and Mg. So zinc, then again zinc and ferrous, they are kind of what? They are kind of chlorophyll synthesis. They are involved in chlorophyll synthesis, zinc and iron, right? But the CHON and Mg, they are the components of chlorophyll. They are two different ideas, right? Okay. So answer is 4. Then come to 66. Then come to 66. The spores produced during asexual and sexual reproduction in Aspergillus are respectively in sexual. In asexual, actually, they produces what? Right? They produces conidae, no? Conidae, and in sexual, they produces asco. Then asexual, conidae and asco. What is the answer? Answer number 3. Right? Then come to question number 67, right? Cells that can be considered as non-living, right? Cells that can be considered as non-living. Collenchyma are living. They are living, actually. We'll write it down. These are living, right? Sclerace, they are ones, sorry, these are non-living, right? Xylem vessel element, non-living, right? Then cork cells, they are non-living. Then B, C, and D. Answer number 5. Okay. Then 68. Correct epithelial tissue location combination out of the following is. Okay. Then simple squamous. They are not found in kidney views, right? Simple squamous are found in blood capillaries and alveoli. They are found in blood capillaries, alveoli. Kidney views, we have simple cuboid. Then simple columnar epithelium, intestinal lining, that is correct. Then trachea, not simple cuboidal epithelium. In trachea, what it has? Trachea has what? Pseudo-stratified squamous. Then simple cuboidal epithelium is found in what? Associated with glands, right? Likewise, okay. Sorry, right, in glands, and that glands means thyroid and salivary, thyroid, salivary, and can be also found in that kidney tubules also. But simple cuboid, kidney tubules, simple cuboid can be found there, right? So pseudo-stratified columnar epithelium alveoli, that is incorrect. In alveoli it is found what? Simple squamous. We have already given that. Then this also pseudo-stratified columnar epithelium, just given now. Pseudo-stratified columnar epithelium is found in places where that frequently, now we'll see, nasal cavity, that frequently goes with respiratory pathway, right? Okay, nasal cavity, and then trachea. Okay, trachea is a place there. Stratified squamous epithelium, right? stratified squamous nasal tract. No, that is incorrect. Stratified squamous is a complex epithelium that is found in outer skin. Then it is found in lining of mouth. Lining of mouth. Then again it is found in anus. It is found in anus, and also it is found in vagina. What is that? Simple. Sorry, it's not simple. Stratified squamous epithelium, right? Sorry, pseudostratified columnar here, not squamous. Sorry, really sorry that I write the squamous. No, pseudostratified columnar. The both nasal cavity and trachea both. Pseudostratified columnar, not squamous, right? Okay. Thank you very much. Pseudostratified columnar. It's a my mistake, right? It's a my mistake, right? Okay. Then these are the epitheliums and just kind of a rough analyze. Now once again, if we write about nasal cavity, then again nasal tract, pseudostratified column. Thank you very much, Latika, right? Latika, thank you very much. Pseudostratified column. Now all fine, no? Right, okay. Right. Then we'll move on to the question number 69. A component of pancreatic juice is, no? Component of pancreatic juice. Normally, what are the component of pancreatic juice? They are the bicarbonate, no? Bicarbonate is found. Then pancreatic amylase. You have to write down pancreatic amylase, right? Because amylase is found in where? Found in oral cavity even, no? Right? Pancreatic amylase. Then what else? Then pancreatic lipase. You have to write pancreatic lipase because lipase is found in intestine also. Intestinal lipase is also there. Then nucleus. Nucleus, but you have to write in pancreatic because not needed because it's in the pancreas only. Then trypsinogen and chymotrypsinogen. Then you have trypsinogen and you have chymotrypsinogen, chymotrypsinogen. These are the contents of what? Pancreatic juice. So out of this, not amino acids, not nucleosides, not yes, trypsinogen, not disaccharides, not phosphatases. Answer is answer number 3 for the 69th. Okay, fine. These are very important part. These are very important parts, right? You have to keep on going through, right? Okay. Then question number 17. A molecule that is passively absorbed. This is a very important point. Okay, passively absorbed through the epithelium of villi. That is the fructose. Fructose, right? Fructose. Then what about these amino acids? They all are active, right? Amino acids, small peptides, then vitamins, then folic acids and other thing. These all are right. What? Actively absorbed. Actively absorbed. Another one, keep in mind, actively absorbed, right? Many glucoses, sorry, right? Many glucoses are actively absorbed. Many glucose, but keep in mind, it means it suggests there are some glucoses which are passively absorbed also, no? Right? So fructose is passively absorbed. So keep in mind that it's given some glucoses are, that means not some, actually many glucose molecules are actively absorbed. It suggests that there can be, now it's not given in resource book, but I will write, there can be few glucose molecules which are passively absorbed, right? Which can be, there can be, I don't know, it's not, I don't know, actually it's not, I don't know, actually it's not given in the resource book, right? But it's this is the point, there can be few glucose molecules which are passively absorbed because they are clearly given that not all, that many glucose molecules are actively absorbed. Okay, got that point? Nice little point, right? Okay, right. Then we'll come to the question number 71. Okay, students, just drop a message how you feel of the session. Now just around 4 hours and 20 minutes, no? Just how you feel of the session. Just drop a message. Just somewhat boring you might feel, just to avoid that, just drop a message how you feel of the session and how the session is going on. Just a small message, just to break that monotony. Right? It's okay. No issue with the speed, no, right? Because I'm not that much fast, no, that you have enough time to note down, no, note down the important facts. Enough time to note down, no, right? Okay. Okay, right. Okay, very good. Then, right. So is it better to keep memorizing the part around the MCQ and go to the next question? Next question discussion, no, just go through sir's explanation only. No, you can just, if with the time you are available, you can further just begin with the part now. Here now question number 70 is regarding the absorption in the small intestine. No, if you have sometimes just maybe 5 minutes, right? Just go through and just roughly go through the point, and just you can write what is there important. No issue, but now it's depend on your level. If you are in the level of 60-70 marks on papers, when you write biology papers, then you can have that luxury to do, no. But for students who are at the lowest of their levels, so they don't have much time to that, or have that kind of what that courage to go through the resource and find facts. So you guys, it means those who guys at their lowest, just entirely completely depend on our discussion only, right? Discussion and the upcoming sessions, right? So those will be important. Actually, the I think the most vital, I think now provincial papers, students, provincial papers don't pay much at that kind of plenty of attention towards provincial papers, because I just include that as a part and parcel of this the seminar series. But I think that if I can take that time also to discuss any kind of MCQ points, I think that is more productive, because if we just go through the provincial papers, these are similar kind of questions, right? But they are a marking scheme on provincial papers. Then you can even, those who completed, you can just go through and check it themselves. We'll try what we'll just go through the provincial papers and grab the important points. But the most vital part is that this MCQ 500 is very good and very important. And the other part is that Poduratava or the common pattern tute is the most vital one, and that the points I'm going to discuss, right? I'm going to discuss with you on Sunday. That is the most important thing. And I think by the Sunday, you have to complete your works here and there and just get into that session. We'll try to actually figure out the A-level paper there, right? Try to figure out the A-level paper, right? Okay. Right. Okay. Right. Then 71st. The option that shows the respiratory pigments found in human body, right? Okay, we'll go through, right? The option that shows the respiratory pigments found in the human body is right, right. Then we'll see hemoglobin is present in actually the blood of human, and also other vertebrates and annelids also. Now it's asking the found in the human body, you know. So now you have to, now this that respiratory pigment is kind of a common pattern, right? So respiratory pigment, keep in mind, respiratory pigments, right? Respiratory pigments, it's a kind of a common point, right? Common point which is frequently tested in A levels as well, as then there's a more probability of continuing it on right, continuing it on the common pattern, right? Okay, and there's some large probability of coming it on 2025th also, maybe. Now human body, they have hemoglobin and they have myoglobin actually, right? So with both at same answer is the answer number 2. Therefore hemoglobin and myoglobin, that is the answer number 2. So 64 එකේ springwood එක large එහෙම නෙමෙයිද මං කිව්වේ. We'll see 64 springwood large really sorry, you have to correct it. In 64th it has to be large lumen and thank you Ponsisi, right? It have to be large lumen and thin wall, right? Thin wall, okay? Thin wall, right? Thin wall and large lumen, okay? Everyone please come to that part. Question number 64 regarding springwood. It has to be thin wall and large lumen, okay? Thin wall and large lumen, okay? Thin wall and large lumen in springwood. That's, I will just once again this remind that if the stem or that is something having right large lumen, it suggests in large lumen it suggests thin walls, no? If something has right kind thick walls, right? If something has thick walls, its wall is like this, then what about lumen? Lumen is very small, no? If the wall is very thin, then the lumen is large, right? If the wall is thick, then the lumen is very small. That is the difference, right? This is regarding spring, right? This is regarding spring, right? Okay, thank you very much for the that notice, right? Okay, right. Then I think now okay, thin wall and large lumen. Okay, now we are coming back into the place we were there. Now 671 done. 72, the effector forms of T and B. What are the effector form of T? It's the cytotoxic T cells and B is the plasma cells. Then answer is given in the first one itself, right? Answer is first one. Okay, right, answer is first one, right? Okay, first one, cytotoxic T cells and plasma cells. Okay, memory cells are not and the helper cells are not the effector forms, right? Right. Okay, then we'll move on to the 73rd. Unicellular excretory structure. Unicellular excretory structure is what? The flame cell. Keep in mind, if something is given as unicellular structure, unicellular excretory structure, the only possible one is that flame cells, right? Okay. 74th. The sleep and wake cycles of man are controlled by the what? Hypothalamus. Keep in mind that sleep and wake cycle, that what time you sleep and what time you wake up, that is controlled by hypothalamus. These are straight points, right? Okay. 75th, what is the structure of human ear that helps to get angular motion? These are straightforward points. If it is angular motion, keep in mind, if it is angular motions, right? If it is angular motion, then that is what? Semicircular canals. Semicircular canals, right? These are straight points, right? Straightforward points. Then answer number 5. Right, straightforward points. Semicircular canals, there is a kind, you can get this as a short method even now. Semicircular canals are arranged like this, no? They are arranged in different planes, one in here and one in this plane. Now if it is like this, then there is a kind of what? Circular movement, no? Or the angular movement, no? Right? Angular movement, then it has angular velocity, angular acceleration, likewise. Okay, then that's what this there's a relation with semicircular canals and angular motions. Okay, right. Then we'll come to 76. Right, a process regulated by positive feedback mechanism, right? Then a process, right? Normally process feedback mechanisms are there are with two types, right? Feedback with two types. One is positive feedbacks, other one is negative feedbacks, right? Okay, Positive feedback is associated with childbirth or parturition, right? Okay. Then again, positive feedback is associated with lactation. Then again, blood clotting has positive feedback, right? Okay. Then right. Then when we come to negative feedback, regulating body temperature, regulating body temperature, right? Then regulating blood glucose level, and then, right, osmoregulation. So osmotic balance, this is negative feedback, then lactation positive, this is negative and this is negative, and spermatogenesis actually it contain both positive and negative both, right? Okay. Spermatogenesis and as well as oogenesis, because it include both negative and positive feedback mechanisms. So therefore, what is the answer? Answer number 3, right? Right. Then 77. Correct function fetal membrane combination in question number 77. Are all homeostatic regulations considered negative feedback? Uh, most of the homeostatic, you can't say it's better to keep away from all, but what are the homeostatic regulation in A level syllabus are the body temperature, then blood glucose level, then osmoregulation, no? So then those three are which are there in the resource book, and all the that homeostatic regulation given in our resource book are negative feedback. Got it? That got it. The one who have asked the question, I think it's okay. Then come to 77. We'll see. Prevent the fetus from desiccation. It is not by the chorion. It is done by the amnion, right? Okay. Then produce SCG to maintain pregnancy. That is done by the chorion, not by the yolk sac. It is by the chorion. Then protects the fetus from the mother's immune system. That is done by the chorion, not the amnion. It is by the chorion, right? Then processes associated to development of urinary bladder, that is by allantois, that is correct. Producing the primordial germ cells that move towards testis or ovaries, that is done by the yolk sac, not by the allantois. It is by the yolk sac, right? Yolk sac. Okay. So that is what belongs in the question number 77. Right. Right. Question number 78. The hormone that is released by the placenta is an no, right? Hormones released by the placenta. Now HCG release, estradiol release, progesterone release, then all A, B, and C by placenta. All are correct, no? Right. Right. Then 79. Which of the following is correct regarding human skeletal system? Right. There are soft membranous regions called fontanelle between cranial bones in adults. No, fontanelles are not present in adults. They are found in infants about. Now, fontanelle are found in infants around 1 to 2 years. Now, fontanelle are found in infants around 1 to 2 years. No, infants around 1 to 2 years, right? So that is incorrect. Then there are sinuses in ethmoid, nasal, and sphenoid bones, right? Then sinuses are present in sphenoid, ethmoid. That's okay, sphenoid and ethmoid. But not in nasals, they are in maxillary and frontal. No, maxillary. And then when it comes to sinuses, sinuses are found in. Write down, these are very important points. Found in ethmoid, then in sphenoid, then in frontal, and maxillary, right? There are three processes in the temporal bone: zygomatic, mastoid, and coronoid, right? Okay. Temporal bone consists of zygomatic process, that is okay. Then mastoid process, that is okay. And the other process is not coronoid, it is the styloid process. Coronoid process is present in the in the mandible, right? So coronoid is not present in the temporal. So process in the temporal bones are what are they? Zygomatic, then mastoid, and then styloid. These are the process found in the temporal bone. Occipital condyle articulates with the temporal bone to form the temporal mandibular joint. Occipital condyle articulates with the temporal bone. No, right? So temporal mandibular joint is formed. It is between the mandible and the temporal, no, right? Mandible and the temporal bone, right? So that is the condyloid process, right? Condyloid process. So condyloid process is the articulates with the temporal bone, not the occipital. Occipital condyle articulates with the actually with what that atlas, no? Occipital condyle articulates with atlas. You have to write it down like this. Then occipital condyle articulates with actually atlas, right? Not with the temporal bone. So temporal mandibular joint is formed by the condyloid process and temporal bone. I will write condyloid process in mandible plus temporal bone. This is the formation of temporal mandibular joint. The head of a rib articulates with transverse process, right? While facet articulate with the vertebral body. No, that is the hindsight of that. The head of a rib articulates with actually vertebral body, no, right? Then while facets articulates with the transverse process. Now, if we take vertebra like this, now we see now rib is like this, no. Now this is the head part of the rib. Then the facet is like this, no. I will just mention facet. Then in the now facet is like this. Now the head is now this facet. Now head is articulate with the body of the vertebra like this. And there you go the transverse process like this, no. This is a transverse process. Those two transverse process. Then there is a articulate facet for head in the body, right? This is the articulation of head, right? Head of the rib, right? Head of this rib with the body of the vertebra. Now this is the head, no? Head bound with the body, right? This is the body of the vertebra, and this is the what you know as the facet, no? This is the facet in the rib. The facet articulate with the transverse process. This is the transverse process, right? This is the transverse process. So keep in mind, this is the arrangement like this, right? This kind of arrangement is seen here, right? Okay. So is it clear? Okay. Okay, Lama. This is a very important point. Now this is the rib. Now this is a rib. Okay. Now this is a rib. So in the rib, there is a facet on the way, and there's the head at the end. So head articulates with the body of the vertebra. Then the facet articulates with the transverse process. Just drop a message. Is it clear for you? Is it clear that rib articulation is very important, because rib MCQ is on the way, right? Rib MCQ is on the way for the 2025 A level. So therefore that can be tested in the A level, right? So therefore you need to have proper understanding, proper knowledge of what is the articulation type, how it articulates with the vertebra, what are the parts of the rib articulates with the vertebra, like this. Okay. So then that is also incorrect. So then all the statements of the here that MCQ are incorrect. So therefore this is a all one, right? This is all, right? This is all. There is no answer. Right? This is what you have to understand. So this MCQ carry very important points. Then what about fontanelle? When who have fontanelle, then you have a very important point that occipital condyle articulates with the atlas vertebrae. Then you have which we have sinuses, very important point, right? Then what are the processes found in the temporal bone, very important point, right? Okay. then you can also write down what are the processes in then you can write down if in the mandible I will write it also, right? So in the mandible also there are processes. What are the processes in mandible? They are the coronoid process and the condyloid process, right? Condyloid and coronoid are the process found in the mandible, right? Okay, so keep in touch. So then these are the processes, 5 processes in our skull we discussed. These are the process in the temporal bone. They are the zygomatic, mastoid and styloid. Then the process found in the mandible, 2 processes, coronoid and condyloid. Then forming the temporomandibular joint, what are contributes condyloid process in the mandible and the temporal bone. Okay, temporal bone. Then temporal bone along with the condyloid process in mandible form the temporal mandibular joint. Then when regarding a rib articulates with the vertebra, it means actually rib articulates with the vertebra. What is this type of vertebra, Lamai? What is this type of vertebra actually? These are what vertebra? These are thoracic vertebra, no, because ribs are found in the thorax region. Ribs are not found in the lumbar region. Ribs are not found in the cervical region, no. Ribs are not found in the what? Where? Ribs are not found in the sacrum or coccyx region. So ribs are found in the thoracic region. So therefore, this vertebra is a thoracic vertebra. Then in the thoracic vertebra, rib articulates with the head and the facet. So head and facet are found in the rib. So head articulates directly with the body of the vertebra. Then facet articulates with the transverse process of the rib. Okay. Those are the very important facts. Okay, we'll move on to the 18th. Even though the question is all, this is a heck of a question, right? Okay. Regarding the genetic code, if each nucleotide is converted into an amino acid, only 20 amino acid can be coded, no? Right? If each nucleotide is converted into amino acid, no? Right? Then how many nucleotides there? Right? Right? Okay. Then it will be adenine, guanine, right? Then RNA, no, uracil and cytosine. Only 4, not 20, right? Not 20, only 4 amino acids can be formed, right? Can be coded. That is incorrect. All amino acids have more than one codon. All amino acids have more than one codon, right? What do you think? No, not all. Not all, right? Some have more than one codon, right? Some have more than one codon, right? Some, not all. Words of 3 letters are or a triplet are read one after the other, right? Yeah, that's correct. Words of 3 letters or a triple are read one after the other. Then if you take AUG, right? Then we read A, then U, then G. That's the way of reading. That's correct. The third one is correct, right? Methionine is removed naturally after translation. No, we need to use enzymes, right? So can be removed by enzymes. So not removed naturally. No, that is incorrect. So then removed by enzymes, right? Removed by enzymes. Then all 64 codons provide codes for amino acids. No. Out of 64, only 61. Out of 64, only 61 provide codes. 61 provide codes, right? Because rest 3 out of 64, which are UAA, UGA, and UAG, these are known as what? They are known as stop codons. So they don't code, right? They don't code. So therefore that is incorrect. So answer is third one for the 80th. Then here we come to question number 81. Right, question number 81. Okay. Fine, no? Right. 81. A condition required for Hardy-Weinberg equilibrium. This is a very important point once again. This is a very important point. Hardy conditions, right? They can be given in 2025. So be careful. So there cannot be any mutations in for Hardy to be in Hardy-Weinberg equilibrium to be there. There cannot be any mutation. Then there should be random mating. That is correct. Random mating should be there, right? Presence of natural selection. No natural selection need to be happen. No, cannot be happen. Actually, no natural selection. Population has to be very large actually. Very population means to be extremely large actually, right? Not small. Extremely large population need to be there. So therefore that is incorrect. Immigration and immigration cannot be taken place. So no immigration and immigration, right? So actually for Hardy-Weinberg equilibrium to stay, so this need to be large. So no incoming, no outgoing, right? Actually. So then there is actually inside there is random mating need to be processed. Actually, then no natural selection, right? No natural selection. If it is natural selection, there is natural selection means no natural selection here. So there should not be natural selection. If there is natural selection means some organisms what they just leave the population by as a result of natural selection. What happens? The fittest stays. No, the fittest survive. Actually, the fittest organism survives, others fallen part. Then fallen part means they are dying. They are dying means they are emigrating from the population. So cannot be that cannot be let to do, so let to take place, right? That cannot be happen in to become this Hardy-Weinberg equilibrium true. So right, okay. So this need to be taken. So keep in mind mutations cannot be happen. So random mating needs to be happen. Random mating inside the population can be happen. Presence of natural selection is not available. So there should not be any natural selection. So the population need to be extremely large. So then the accuracy when the population is large, when what happens to the equilibrium, the accuracy is increasing. No, accuracy is increasing. Then immigration and immigration, what happened? There should not be any immigration or immigration. That the reason is that population needs to be remaining continuous. The population need to be consistent. No, consistent. No, need to be consistent. There should not be any increasing or decreasing in certain levels. No, right? Okay. Then that's for the 81. Then answer number is 2. 82. Right. We'll see now. This process that occur during protein synthesis. Now in Podurata also there are certain particular applications for these things. Now we'll see. We have to first understand where these things happen. Now A is taking place at the now A is at the actually beginning, no? Beginning of the initiation of translation. Then B is at the elongation in translation. Elongation in translation. Then C, termination of translation. Then D, beginning of transcription. Then E, beginning of the middle of translation. Beginning of middle of translation. That's what, right? Okay. So, right, okay. Right. These are the places where these things are taken place. So you need to understand what is the order. So first, what is happening? First is the transcription. No? After transcription, what is happening? Translation. No? So each transcription and translation are followed by 3 steps. That is the initiation. 3 steps. Now initiation is the number 1. Then second one is the elongation. Then third one is termination. No? These are the order. So now we have to understand. Now translation. Now transcription is here, right? Transcription is here. Now we have to understand an order. So therefore, definitely D need to be the first one, because all the rest of the things are translation. No, so therefore transcription had to be first. Then you can cut down this one. Then you can cut down this one. Then 1, 2, and 4 are remaining, right? Then in the translation, what is the final one? Final one has to be the termination. No? So these are very, very, very important points. So you can understand the answer. You can just use termination of translation. Now, what is the first one? Is the transcription. We have get the clue there. Then out of the translation, what is the last one? Is the termination. So termination of translation here, and then it is the last. Then C needs to be the last in the answer. So therefore here no C at last. Then 2 we can cut down. Then 1 and 4 remain same. Then we need to find whether B or A take place first, right? So B is at the initiation of translation, then sorry, A is at the initiation of translation, B is at the elongation of translation, right? Okay. Then E is at the beginning of the middle of translation, right? So therefore, what we have to say, then middle of the translation, right? Then translation middle means elongation, no? Right? So sorry, initiation means, no? Then the elongation in translation is the B, right? So it has to be now the initiation means beginning of middle of translation means initiation here. Then beginning of the initiation of translation, then beginning of the middle of the translation, it means initiation, the middle of the initiation. So therefore, this is the, I think the after the D, this is the best one. So the closest one, beginning of the initiation of the translation. Now, if we take initiation can also be taken as beginning, middle, and latter. No, now think initiation can be divided as beginning, then again we can divide it as middle, then as latter. No, what that certain initiation. Then even elongation can be divided as beginning, middle, and latter. Then termination also can be divided as beginning, middle, and latter. Then what happened here? Then A is the initiation beginning one. Then what is E? E is the initiation middle one. Got it? Then B comes with the elongation. It's the anyway last. So therefore, D first one, C last one. Prior to last one, it has to be what? B. You know? Then prior to C, there should be B. Then this is the answer. Then we'll see, is it correct? D right, A right, E right, B right, C. Fine. That's the final answer. Okay, Lamai, drop me a message whether you are clear of getting such kind of an answer or answer for such kind of an question. Quickly send me. To that you need to have that what you need to have the order. You need to have the order for that, right? You need to have knowledge of the order or where these processes are taking place, whether in the translation or transcription. You need to have that knowledge for that. But that knowledge will be given in during the right. Okay, that knowledge will be given during the Poduratawa discussion, right? Poduratawa discussion, that knowledge will be given. Okay, that knowledge will be completely given during the Poduratawa discussion. Okay, okay. Poduratawa discussion, that knowledge will be given. Okay, so that is the important part, right? Then right, right, then we come to the question number 83. The structural and functional unit of inheritance, that is the you have to keep in mind when they are given as structural and functional unit of inheritance. That is definitely need to be what? Definitely that is need to be what? It is the gene. They are referring to the gene. Nah? Keep in mind, they are referring to the gene, right? They are referring to the gene. So you need to go through ahead with referring to gene. Now, genes of eukaryotic cells exist individually. Now, keep in mind, now eukaryotes, they are what? They are two-end organisms. No? They are what? They are diploid organisms. So genes of eukaryotic cells exist as what? They exist as pairs. So therefore not individually, because we all have mother and a father. Then therefore there is no room of existing genes individually. They exist as what? As pairs. No, keep in mind that we are diploid organisms. The location of chromosomes on a gene is known as the gene locus. right? The location of a chromosome on a gene. No, that is incorrect. The location of a gene on a chromosome. So that has to be the order, right? The location of gene on a chromosome. Location of actually location of gene on a chromosome. Gene on a chromosome, you have to be, that is the incorrect one, right? So gene of prokaryotes are stored as circular DNA as split DNA groups. What is that? Genes of prokaryotes are stored as circular DNA as split DNA groups. Yeah, the genes of prokaryotic genes are found in their DNA. No, their DNA is circular DNA. That's good. That's correct. And they are found as split DNA groups. That is correct, right? Eukaryotic genes are arranged within the same region of the chromosome, one after the other as group. No, one after the other arrangement is found in circular DNA. Now we see this is a circular DNA. Now this is a circular DNA means this is a prokaryotic DNA. No? Now if we take like this, now I will take gene one by one. Now this is I will take G1 to gene 1. Then this is what? Gene 2, G2. Then this is gene 3 and this is gene 4 and this is gene 5, this is gene 6, this is gene 7, this is gene 8, this is gene 9, this is gene 10, this is gene 11. Now what happened? Gene 1, then gene 2, gene 3 after gene 2, gene 4 after gene 3, gene 5 after gene 4, gene 6 after gene 5, gene 7 after gene 6, gene 8 after gene 7, gene 9 after gene 8, gene 10 after gene 9, gene 11 after gene 10, then once again likewise one after the other, then one after the other as groups are found in not in eukaryotic, it's found in prokaryotic genes. Right? What in eukaryotes, there are gene 1 here, right? One gene here, one found here. Likewise, not one after another. They are just in linear arrangement. In circular arrangement, the genes are in a linear arrangement of chromosomes. Therefore, not one after the other. Therefore, that is incorrect. All DNA groups in eukaryotic chromosomes are active. No, not all DNA groups in eukaryotic chromosomes are active. Now, when comes towards the eukaryotic chromosome, you know that that in exons are active. Then what is the other thing? Introns, they are inactive. Then intergenic DNA, actually inactive, it means useless actually, right? Inactive. So that is the kind process. Then only active part is the exons. Right? Active part is the exons. Got it? Right. Nice. Then answer for the 83rd is third one. Then we come to 84. Select the correct statement regarding gene technology. Now this gene technology is a kind of a that definite target or definite point to look forward into, and the previous one, that gene question also, right? Okay, the split DNA means operon. Yeah, the split DNA means operon, so you can write it there for the in the third one. Actually third one is referring to the operons actually, right? It's referring to the operons, right? Okay, operons, right? So just keep in mind that is operon, right? So operons are the units found in prokaryotes, no? Right. Then 84th, cloning of DNA can be done in vitro, right? Cloning of DNA, right? Cloning of DNA, right? So then select the question regarding gene technology, right? DNA cloning, right? It is done in vitro, right? So cloning of DNA can be done in vitro. That is correct. No, inside the test tube. No. Oh, correct or wrong? Cloning of DNA is done in vivo or in vitro? In vivo, in vitro. DNA cloning, right? In vitro, no, right? So then the entire length of DNA molecule can be isolated. That is incorrect, no? It cannot be isolated, that entire length of DNA, because DNA length is very, very long, no? So therefore, you can't isolate the entire length, right? So because there are intron and exon parts, so you have to separate all those things, so can't be isolated. Bacterial cell walls can be broken down by grinding and homogenization, right? Bacteria cell walls can be uh, broken down by grinding and homogenization. Yeah, mechanical lysis of cells, know that taken place grinding and homogenization, right? So there, then okay. No, cloning. Sorry, that okay. Cloning of DNA in vitro, ne, here in vivo, no, in vitro here, no. So that normally in test tube condition that we know for the cloning we use PCR, no, polymerase chain reactions. Cloning is doing inside a living body, no. So therefore that is incorrect. Sorry, incorrect. Thank you. Right, I just mislead that with in vivo and in vitro. So in vitro means inside that test tube, no, right? So therefore cloning is in vivo, no, right? Right. Okay. Okay, Lama, right? That part is okay, no? That's I just missed that one, right? Otherwise, you would get that kind of misleading. I think in your resource book it is given as in vivo. I think, no, in vivo, no, right? In PCR, it's in vitro because right, cloning of DNA actually it is both. I think it can be done both in vitro and in vivo, but just going through your resource book, I think it is only given as in vivo. In vivo, no? Just slightly help me that in your resource book it is given as only vivo, no? Only in vivo, I think. But actual thing, cloning is in vitro and vivo. If we take cloning, the simple thing, cloning can be done inside the test tube and in a living body. It can be actually both in vivo. So can DNA cloning be done in both? Yeah, it can be done in both, in vivo and in vitro. Just go through your resource book and find out whether it is in vivo. I think only in vivo, no? In your resource book, it is given as in vivo, no? Just confirm whether that it is given as in vivo. I think that cloning section, the cloning is done in vivo. I think that's given as in vivo. So therefore, cloning of DNA can be done in vitro. Actually, that is also correct. That is not incorrect. So I will just put correct here. No issue, because it can be done in both. So, but just refer your resource books. So therefore, I will leave it there. Chill and find with what is there in your resource book. But it can be in vitro and vivo, right? Bacterial cell walls can be broken down by grinding and homogenization. No issue that cell wall components are broken down during the homogenization and grinding, because it just break the cell walls, no, right? I think in your resource book it is given as in vivo. In that aspect, that's why I just put a question mark. In the resource book, they have given only in vivo. No, I think in resource I will put it RB, right? In resource they have given as in vivo, right? So in in PCR it says that DNA copying can be done in vitro. Yeah, no issue. That in PCR it is in vitro. That is correct. It is in a test tube. No, that PCR is a mixture with doing inside the test tube. No, so therefore actually inside the test tube means in vitro. That is simple, right? But in vitro and even in PCR there is a kind of a cloning taken place. But here in the resource book, I think they just saying cloning in living body, no, inside living body. There's kind of that kind of a meaning they are giving, right? So in living body means in vivo, no, in vivo it means in an organism. So in vitro means test tube. So in a particular organism, like we put a nucleus here, if inside a certain particular organism, then we name it as in vivo. So in vitro means inside a test tube, no, in vitro means inside the test tube, in vivo means inside a living cell. This is a cell, right? Living cell. So cloning can be done in living body in vivo, actually in vitro also. But in resource book, I think in your resource book it is given as in vivo, but keep in mind it can be done in both, right? So okay, in this case, now third one is correct. Precipitated DNA with cold ethanol is dissolved in ethanol acid. No, ethanol acid, it is right. Okay, it is dissolved in a buffer, no, it is dissolved in a buffer, right? It is dissolved in a buffer. So therefore, not a ethanol acid, dissolved in not an ethanol acid. It is dissolved in a buffer, right, to maintain the pH, to regulate the pH values also. All the other DNA in a cell are considered as impurities. Yeah, in DNA technology, all the other DNA in a cell are considered as impurities. All other, actually, not all other DNA, all other substances in the cells are considered as impurities. All other there is some, I think in the fifth one there is a issue, no? All the other DNA in a cell are, right? Apart from DNA, all the other are actually the impurities, right? So there is a kind of a that kind of a that the idea is not conveying properly, right? So the idea is not going properly, right? Yeah, so isn't gene cloning not a gene technology? Yeah, gene cloning is also gene technology actually, right? In gene technology, we are doing gene cloning, no, right? Right. In normally gene technology, here in the resource book, we are discussing about the inside the living body, no? Inside the living body that gene technology is discussing. That's why they are given it as in vivo, because we use bacteria, all those living beings to for the process, no, right? Okay. Then normally cloning can be in vivo in vitro. Keep in mind, right? Cloning can be in vivo in vitro, but here it's discussed about the living system, right? Okay, living organisms, inside living organisms, right? So then fifth one, all the other DNA in a cell are considered as impurities. That is that idea is lacking. I think I just write this idea is lacking there. Idea is lacking there, right? Idea is lacking, right? So in the resource book, it is given that lysis of bacteria cell wall is done by enzymatically using lysozyme, right? Yeah, bacteria cell walls can be now normally the lysis of bacteria cell wall is done by enzyme. That the enzyme is the lysozyme, but here it says bacteria cell walls can be broken down, no, right? Can be broken down. Even grinding and homogenization can lead into the broken down of bacteria cell walls, right? Right. Yeah, right. So then so here then bacterial cell wall can be broken down by grinding and homogenization. It can be true, it is true, because but the main thing you can for the bacterial cell wall you can use the lysosome also, lysosome enzyme. It's the main thing that is using, but in the process of grinding and homogenization, that the cell wall is again breaking down, because breaking down into pieces, but it's not the final output. Got it? Now if we take like this, then bacteria cell wall is like this. If this is bacteria cell wall, right? Then homogenization and then we'll take grinding. Then what happened? It is broken down into pieces like this, right? It is for then with lysosome, it actually broken down to small tiny pieces. This is the final output. So lysosome is the major one, but homogenization and grinding can contribute into somewhat. Got the point? Got the point that who has Latik, I think Latik got the point that lysosome is the major one actually. When we talk about the bacteria cell wall, lysosome is the major one. There is no doubt of that, right? But the thing that homogenization and grinding can to a some extent can break down the cell wall, but it's not the final end product. It's not the final product, right? It's some sort of a degree they break down, but the final touch up is done, or the final process is done by lysozyme. Got it? Right? Got it? Ah, nice. Okay. Then, so therefore, that's why I take this as correct, but it's not given as any enzymatically breakdown or likewise. No, bacterial cell walls can be broken. No, it's not saying that it is the best way or kind of that thing, right? Can be broken down. Therefore, that's taken as correct. So then fourth one incorrect. Then fifth one is incorrect. But I think all the other DNA in a cell, all the other components in a cell are considered as impurities. That's the statement. Now I think for the fifth one, I think you understand why it is incorrect. Now if we take a cell, there are DNA, no? Then there are RNA, then there are what? There are proteins, right? Then there are ions like magnesium, all those things. Now these magnesium ions, proteins, then RNA, all these are what? all these, then proteins, magnesium, all these are what? They are impurities, guys, right? They are impurities. So then DNA, for the DNA, they are impurities. Then others are impurities for DNA. That's the point. Others are all the other things except DNA are impurities for DNAs. But here in the statement, it is given that the idea is not conveying properly. Idea is lacking actually. That's why I here tried that, write that. All the other substances in a cell are considered as impurities for DNA. That's what the statement have to be, right? Okay, got it? Got it. What about the fifth one? Did all got that point? Just drop a message. Did all got that point? Just drop a message. Just drop that message. Right, okay. Right. Then for the first one, right? First one, right. That someone has put the part of the resource book. Therefore, a large number of copies for the recombined DNA tech molecules are necessary. At least a few host cells will receive a copy. If the desired DNA fragment is a short one, in vitro multiplication can be done by technical DNA cloning, right? Okay. So then for the uh, cloning of DNA, I just mentioned that cloning can be in vivo and in vitro, no? Right? Okay. But anyway, in the resource book, it's given in vitro scenario is given, no? Right? Not in vivo. Sorry, wait. So then we can take that one as correct also, right? Okay, no issue. But they have already given. I think in single medium, I didn't remember the part, but I will surely update you. I think some part, some point, some extent, they given that in vivo for the cloning part, I think, right? But actually in vitro is correct. In vitro is correct, and also the gene technology, we just combine with the machines, test tube and all those things now. So therefore the gene technology can sometimes taken or doing apart from the living cells, right? Okay. Right, okay, that's the point. Okay, then all fine, no? Then first one you can take it as correct, right? Regarding the gene technology, correct. Then second one incorrect. Okay. Then bacteria, yeah, that's also can be partly correct, but it's not actually not 100% correct. Not 100%. Just mind the fact, and I have explained it in here that regarding the point, not 100% correct. Then buffer is incorrect. This is also incorrect, but then you can take first one or the third one as the answer. If we go to a final answer, then will be you can take the first one. No issue. Cloning DNA can be done in vitro. Can be done in vitro. That is also correct, right? Okay, it can be vivo also, right? Okay. So those are the things, right? Right. Then yeah, that's for the 84th, no? Right. I think that's okay. Then come to the 85th question. 85th. Then we'll come to the 85th. Then correct statement regarding DNA libraries, right? Mainly used as sources of DNA groups for sequencing. Now, what is a library? Now, DNA library means actually it carries different. Now, keep in mind different types of DNAs. No, now if we take this test tube, now we'll like this. Now, DNA library. No, library means a kind of a stall. No, so this contain A, B, C, and D sample of DNA. It might contain this kind of a DNA, right? So this might contain this kind of a DNA. Then C might contain another kind of a DNA like this. Then D like this, no? D like this. So these are the different types of DNA kinds. Now A, B, C, and D in the DNA library. No? Okay, got it. So these are the continuous. These are the manner. Then if we need A type of DNA, we can get it from this one. Then if we need B type, we can get it this. Then it's a kind of a different types of DNA mixtures. No? DNA mixtures present. So therefore, mainly used as source of DNA groups of sequencing. That is correct. Using RNA polymerase, DNA strand is replicated on the first DNA template. Now DNA replication. Now here DNA libraries, no. So therefore, no need to use RNA polymerase. So what is used here is the DNA polymerase, right? Okay. DNA polymerase is what here used. So therefore, not RNA polymerase. Therefore, that is incorrect. The sum of RNA in a cell is known as transcriptome, rRNA. right? Transcriptome is the sum of mRNA, not the rRNA, sum of mRNA. So mRNA, all the mRNA in the mRNA, in the number of mRNA in the cell is called the transcriptome. Keep in mind, right? Not the rRNA, right? mRNA. cDNA libraries do not represent the pattern of gene expression. No, they represent the pattern. They show the pattern. Actually, they show the pattern of gene expression. cDNA libraries. So what are cDNA libraries? Now, cDNA libraries means this kind of a thing. Now, keep in mind. Now we know now DNA is converted into what mRNA, no? Right? DNA is converted into mRNA, right? Clear? Once again, go on PDF. Okay, anyway, no issue. We'll discuss with here. Now, now we have first one is correct, right? Then first one is correct. Then second one is incorrect, right? Okay. Then third one is also incorrect, no? Right? I will write them. Then the reason here is it is what is the enzyme used? That is the DNA polymerase. So that's why it is incorrect. Then here sum of, it means total of mRNA is the transcriptome, right? Not rRNA. Transcriptome is by the total of mRNA, right? Keep in mind. Then cDNA scenario. Now DNA now come here, right? Now now DNA is translated into mRNA. Sorry, actually it is transcripted, no? This is transcription, no? Transcription, transcripted into mRNA. Then once again, we can transcript mRNA back into DNA, right? In that procedure, that DNA which is made in this scenario is known as cDNA because it is not the DNA from DNA, no? It is the DNA from RNA. This process is called the reverse of the transcription. Therefore, it is known as a reverse transcription. Right? So, these are known as cDNA. cDNA means the meaning is copy DNA because the DNA which have come from mRNA. Got the point? So, cDNA libraries do not represent the pattern of gene expression. That is incorrect because cDNA or the copy DNA contain all the traits which were there on this DNA. This is the similar. So, this copy DNA can express gene. They can be involved in gene expression. There is no issue in that. They are involved in gene expression. Got it? They are involved in gene expression, right? So DNA, the same DNA is converted to mRNA. That particular mRNA is once again converted back into DNA. So that's DNA is named as cDNA because that DNA is coming from mRNA, not from DNA, right? Not directly from DNA, no. Come directly from mRNA. That's why it is cDNA, the copy DNA. So that process is reverse transcription, and that copy DNA, no issue. They can involve in the gene expression, no issue, right? Okay. Then that is incorrect, no? They actually do not represent the pattern, no? They represent the pattern, right? They represent the pattern. This is a microbial culture medium that can transmit similar groups from the total DNA genome. This is a microbial culture medium that can transmit it. Just think, similar group from the total DNA genome. No, not similar groups, right? If you think of a library which have only the Madolduwa book, right? If you think of a library which have only the novel that Baddegamo like thing, or only having certain particular that we'll say the Sunaka Shape of Chandana Mendis, like that that Sherlock Holmes Sunaka Shape, right? Likewise, the library only has that book, has thousands of copies of particular book, or Madal do 1,000 copies. Is it a library? No, it's a Madal do store. So therefore, then in a microbial culture medium, that need to have not similar, no, similar group of total DNA, no, they need to have different forms of DNA, no. The library should have different forms of DNA, that's it, different forms of DNA. So that's why the fifth one is incorrect. That is the part, right? That's regarding the question number 85. Got that, everyone? Everyone got that? Very important one. DNA libraries, a kind of perfect guess for this year, right? Perfect guess, huh? Okay. Not a simple one, it's a perfect, right? Perfect guess, that's it. A perfect guess, that's all, right? Okay, nice. බත් ටිකක් කමු කියලා කියනවා කට්ටිය මොකද කරන්නේ? බයි ඕනද කට්ටියට කන්න? බයි ඕනද ළමයි? බයි කන්න ඕනද? මම බයි තියා උදේට කාලත් නෑ. මේක ඉවර කරන්න ඕනනේ වැඩේ දැන්. ෆ්‍රී කියලා දන්නවනේ අපි වැඩක් කරද්දි ෆ්‍රී කියලා නිකන් අර උඩින් පල්ලෙන් කරලා දාන්නේ නෑ. අර ඒක පේඩ් ක්ලාස් එකක දෙන උපරිමේ දෙනවා මේක ඇතුලේ. ෆ්‍රී කියලා නිකන් අර සමහර ෆ්‍රී කිව්වම බලාපොරොත්තු වෙන්නේ කරගන්නකො කොහොමද නිකන් කරන එකනේ මං කොහොම කරාම මොකෝ වගේ එකක් නේ. ඒ අපේ එහෙම නෑ ෆ්‍රී කියලා එහෙම නෑ. මේ හරි. ක්වොලිටි නම් ක්වොලිටි ෆ්‍රී කරත් හරිද පේඩ් කරත් මමම තමා සීන් එකක් තමා තියෙන්නේ අර කියන්නේ කතාවක් නේ ප්‍රසිද්ධ කතාවක් තියෙනවා නේද? පාර්ලිමේන්තුවේ කියපු කතාවක් තියෙන්නේ. එහෙම නම කියන එක හරි නෑනේ. කොතන හිටියත් මොකේ හිටියත් අර මමම තමා කියලා කියන්නේ අන්න ඒ වගේ තමයි. හරි ඒ නිසා ක්වොලිටි එක මේන්ටේන් කරනවා කොහොම කරත් රයිට්. මේ බත් බඩගිනිද කොහොමද කමුද එහෙනම්? කාලෙ එමුද වැඩේට? මේ රයිට්. ළමයි ඒක මේ අර ක්ලෝනින් එක රීකම්බයින් ටෙක්නොලොජි එකේ හරි ඉන් විට්‍රෝ තමා ගන්න ඉන් විට්‍රෝ කියලාම ගමු මේක කියන්නේ අපි මේ කතා කරපු ප්‍රශ්න මෙතන මේකේ 84 ෆස්ට් වන් මේ එක විවෝ නෙමෙයි ඉංග්‍රීසි මේක අප්ඩේට් එක කොතන හරි විවෝ එකක් තියෙනවා මම සිංහල එකේද කොහෙද රිසෝස් එකේනේ anyway මේ ඒක විට්‍රෝ තමයි හරි මේ ප්‍රශ්නයක් නෑ මම ඒකයි මම දැන් සම්පත් පොත බලලා කන්ෆර්ම් කරගත්තේ මේක දීලාම තියෙනවා හරි. ඒතර මම හිතුවේ සම්පත් පොතේ නිකන් විවෝ තිබ්බ වගේ මට මතකයි විවෝ තිබ්බ වගේ ඒකයි මම මේ විවෝ කිව්වේ මම සිංහල එකෙත් දන්නේ නැහැ විවෝ එකක් තිබ්බේ. මම ඒකයි ඔයාලට පැහැදිලි කළා මුලින් කිව්වේ මේ සාමාන්‍යයෙන් ක්ලෝනින් කියන්නේ නම් විට්‍රෝ කරන එකක් කියලාම කිව්වේ පස්සේ ඒකයි. රයිට්. එතකොට මේ විට්‍රෝ විවෝ දෙකම වෙන්න පුළුවන්. දැන් විවෝ මෙහෙමයි දැන් ක්ලෝනින් කියන එකක් කිව්වහම ලිවින් සෙල් එකක් ඇතුලේ ක්ලෝනින් නැචරලි වෙනවා එකක් වෙනවනේ. හරිද? ඉතින් ක්ලෝනින් කියන එක ඇත්තටම නැචරලි වෙනවනේ. ජීන් ටෙක්නොලොජි කිව්වහම ඉතින් ගොඩක් කතා කරනවා අපිට දැන් නිකන් අපි එක්ස්ටර්නලි අපි කන්ට්‍රෝල් එක අරන් කරනවා වගේ කියලා ගන්නත් පුළුවන් නේ. හරිද? පැහැදිලි නේද? එතකොට ඒ නිසා මේ ඒ ටික එතෙන්දි ගන්න. හරිද? රයිට් ඒ විදිහට. හරි එතකොට මේක හරි කියලා හරිය. මේක හරිය. ඒක හරි ප්‍රශ්නයක් නැහැ. මේ 84 එකේ හරි. එතකොට ඒක ප්‍රශ්නයක් නැහැ. එතකොට දැන් මේකට වඩා ඕන්න අපිට බෙස්ට් chance එක විදිහට ෆස්ට් වන් ගන්න පුළුවන්. හරි. ඒකත් මතක තියාගන්න. හරි නේද? ඔව් ඒක තමයි in page 32 it is given කොහේ හරි එක තැනක තිබ්බ කොහේ ඒක තමයි මම බැලුවේ මේ page 32 බලන්න. ඔව් ඒක තමයි right. එතන දීලා තියෙනවා නේ in vivo. yes. anyway අරක මම ලියුප් එකම තමයි ඒක. ඒතකොට ඒක perfect ඒක හරි. මම ලියුප් එක perfect ඒක ලියාගෙන ඒ කියන්නේ ඇත්තටම cloning කියන එකේ මේ මෙහෙම ලියා ගන්න හරි. මෙහෙම ලියා ගමු. cloning මේ අනෙතන යාන මෙතන දැන් බලන්න 32 ළමයි මේ පාර්ට් එක මම පොඩ්ඩක් බලන්න 32 පිටු එක තියෙනවා ලස්සනට right okay they are in the page number 32 right copying of DNA is possible in vivo using cloning and in vitro using polymerase chain එහෙම තියෙන්නේ then cloning වල so the cloning is given as in vivo no actually right then PCR given as in vitro this is what they are given then what they they have given is DNA copying, right? DNA copying. So DNA copying is given as two, cloning and PCR, cloning in vivo and PCR in vitro. And just make this short note, just make this short note, right? Then actually now cloning, now this is the final one, cloning with the way they are given. Now that's the reason in resource book, right? Actually cloning is in vitro, in vivo, both possible, right? That's why I just give that. But I just realized now in our with the way we are uh, just learning now, it's different with the A level. Now I'm in the university level and we just take cloning in different aspects. But in your side you have to just think of only the advanced level, no, right? So cloning, it is possible both in vivo and in vitro. That's it, right? That's it, huh? That's the, this is the final short, no, just make this. Okay, okay students, please drop a message now. All okay regarding that in vivo in vitro condition in cloning? All okay? This is the final one, nah? This is the final one. DNA copying can be two types, cloning and PCR. Then cloning in vivo, PCR in vitro. But hindsight, cloning can be either in vivo or in vitro. Just drop a message. Okay, right, Tamai? Now okay? Right, okay. So someone is asking, sir, can the answer one be incorrect? Question in asking about gene technology and it says that cloning is not a gene technology. No, actually recombined DNA technology is the gene technology. In the gene technology, actually it is continued as further cloning, no, right? So in DNA cloning, the DNA replication machinery of the host cell is used to copy the desired DNA. So actually that is kind of that vectors are used is the gene technology, right? So you can't say so that it's not a gene technology, right? Cloning is included in the part of DNA technology, right? Okay. So that is, you can't, in that aspect you can't argue that it's right, it's not a gene technology, right? Okay. Okay, fine. The one who have asked, right? I think it's Pala Credits. I don't know what's the real name. Anyway, you can't argue that it's not a gene technology. No, cloning is under gene technology, right? So that now is the final one, right? Okay, fine, nice. That's it regarding that question. Anyway, maybe better to look through, right? Look forward into gene technology and cloning. We have consumed a lot of time on that single particular MCQ. Maybe that one may be given in this time, right? In the A level, right? 2025. We don't know how the fate is there, right? Okay. Better to keep just highlight and just go through the session. Even in the ninth, the endgame seminar, I will discuss that part, right? Now I think now there might be a chance of getting MCQ from there. That's how fate works, right? Okay. Anyway, the time just consumes much more on that one, right? Uh, then question number 86, right? Question number 86. Savanna in Sri Lanka located in both dry and intermediate zones. Then answer number 2. These are very important points. Just go through that ecosystem table, right? Ecosystem table. I will give this in the that endgame seminar also discuss. But and also this is there in the that Podurata Vatute. But better to once again go through the ecosystem table. That's it, right? Then 87. The ecosystem in Sri Lanka where trees with small round-shaped crowns, leathery leaves, and twisted branch can be observed in where they can be observed in the tropical montane forest to resist against wind, no, to resist against windy conditions, right? Windy conditions that resist against windy conditions. That's the reason, right? This small round-shaped crowns means now not large. That if a plant is very tall in windy conditions, what will happen? Wind will blow like this, and what will happen? The plant will fall down, right? That will happen. So to avoid that, twisted branches that provide the strength, no, when the plant stem is kind of twisted like this, no, then the branches are twisted like then it provides a kind of a additional support, no, it provides a support. This twisting provide the support, right? That's what happening, right? Okay. The leathery leaves means that leathery leaves means when considered with other leaves, that their strength is much more higher. The strength is much more higher. No, when the leaf become the leathery, then it just when compared to another normal leaf, that it tends to fall far more late. It means it takes time to fall. No, it means a huge wind is required to the downfall of those leathery leaves. But normal leaves, they will detach easily, right? So that's why the tropical montane forest. So those plants need to have those adaptations in order to cope with that windy environment, right? Okay. 88. Not an effect of global warming. Not an effect. Okay. Not an effect of global warming. That is the reduction of insect population. Normally, global warming, right? Normal global warming lead into what? Global warming leads into increase in what? Insect population, right? Okay. Insect population. So therefore, reduction of insect population is not a effect of global warming. The occurrence of temperature extremes effect, loss of biodiversity is an effect, depletion of coral reef is an effect, then rising of sea level, all these are effects. So third one is the answer for the 88th. Then we come to 89th, the correct sequence of the steps in the lytic cycle of bacteriophage. This is a very important thing. So you might get it into the right as a SCQ maybe, but anyway, then regarding the steps, what is the how it is happened? Then first, that's the attachment. Now the bacteriophage is attached into the cell. Then after the attachment, it's what it's doing. It's going through the cell. It's penetrate actually penetrate going through the cell. Then what happen? It inside the cell with a collection of the proteins, then lipids is biosynthesized inside the cell. Then what happen? Then maturation and assembly, then protein parts, then membranes, all those are get together and form the what kind of the bacteriophage, the structure. Then finally after the maturation, then what happen? They again release. What happen? They release. Then again attached to another cell. That is what happening, no? Right? So this is the cycle. So it start from this 1, then 2, then 3, then 4, then this is 5. So what is answer that includes this one? This is the answer number 2, CEADB, no? That's the answer for the 89. This is the lytic cycle, no? Lytic cycle. Several cell arrangement of bacteria are given below. The arrangements of where cell division take place along one plane. Single plane cell division, right? Cell division in one plane. Coccus okay, Diplococcus okay, Streptococcus okay, right? Then Staphylococcus, that is incorrect, because Staphylococcus cell divide take place in multiple planes, right? In Staphylococcus, multiple planes. That's it. Multiple planes, right? So keep in mind this fact. Keep in mind coccus, then diplococcus, then streptococcus. Cell division in single plane. right? Single plane cell division. Then tetrad cell division in two planes, two planes. Then Sasine cell division in three planes, and then Staphylococcus multiple planes, right? This is the summary of this point. This is very important, right? Right, okay. So therefore answer is ABC, no? Answer is ABC, no? Right, okay. Right. Okay, right. Here we'll take a short interval. I think don't take hours for this time, right? Okay. Quickly go and take your lunch and come back, okay? With the lunch, come back with the lunch, okay? Then drop your message up to now 90 MCQs have been completed, right? 90 of 500, okay, right? Takes about 5:30 hours, no, right? Anyway, right, what you feel of the discussion? Okay with you are, I think you are getting so many MCQ points, I think, surely. So keep pushing, right? Keep pushing. There is no reason to go back or go right. So you need to go front always, because this is the time to uh, just get the boost in yourself, right? Okay. Then now 90 done, right? Okay, 90 done. I think yeah, lots of facts have been recalled, right? Okay. Then maybe I think I have done my best up till now and hope to do the very best after the interval also. Now go and have your lunch, and we'll be there on 1, right? We'll start on 1 p.m., right? Sharp at 1, okay? So there is no time to waste of hours, like giving an interval of 1 hour, 1.5 hour, likewise. Just a small interval of 22 minutes left. Then, yeah, come back at 1 p.m., sharp at 1 p.m., right? Sharp at 1 p.m. That's it, right? Okay. Right. Okay. Come back at 1 p.m. හරි ළමයි ඕකේ ඇහෙනවද කියලා දාන්න ටක් ගාලා ඉක්මන මැසේජ් එකක් ඇහෙනවද right ඇහෙනවද right හරි ඕකේ හරි right එහෙනම් වැඩේ පටන් ගමු ටක් ගාලා ඉක්මන රෙඩි කියලා මැසේජ් එකක් දාගෙන යන්න right okay so just drop a message saying ready okay Right. Question number 91. Correct statement regarding extracellular matrix, right? Extracellular matrix, right? Okay. So this question is from the unit 2, that's cellular basis of life, right? Okay. So you have to just go through the extracellular matrix, its function, its structure and all, right? Okay. Right. Then first one, main components are glycoproteins and other carbohydrate containing molecules that are secreted from cells, right? Okay. That is correct. That main components are glycoproteins, right? Glycoproteins means carbohydrate binds with proteins, right? Connects the cytoskeleton and extracellular matrix. So yeah, extracellular matrix bind with the cytoskeleton, then it makes a connection with that makes the connection that extracellular matrix and the cytoskeleton. No, itself it connects with the cytoskeleton. Forms a protective layer over the cell surface, not over the cell surface, right? That protective layer is that okay provided on the cell surface, on the cell surface, right? Collagen fibers are embedded in a network woven by proteoglycans secreted from cells. That is also correct. That proteoglycan means protein plus glycans means carbohydrate. Once again, glycoproteins, right? Collagen exists as the most common glycoprotein in all animal cells. The issue is not in all animal cells. In most animal cells, that point is correct, but you can't say that it is true for all the animal cells. Okay, right? That's why E is incorrect, right? Then come to the 92nd one. Common characteristics to both domains, archaea and eukarya. So need to be quite right, quite just kind of a cunning, right? Cunning that cunning thought process need to be there because now when it's asking about those domain similarities and differences, then there are sometimes now now when we come to RNA polymerase, right? The RNA polymerase, when we come to RNA polymerase, it says sometimes found in RNA polymerase, say one type, no? Then many types of RNA polymerase, likewise, right? Then, so anyway, if it is one type or it is many types, it means that certain particular organism or certain particular that the domain is present with RNA polymerase. But when we go deeper into the topic that RNA consists of RNA polymerase several types, then we have to consider of the type, how many types, one or two. Then we can differentiate it with this one containing only one type, this one containing several types, no? So then again, introns in genes, that is another one. Introns in genes, right? Introns in genes absent. Then introns in genes, some in some genes. Introns in genes found in every genes. Likewise, so then introns in gene is the main factor. Then when if it is not given as how many RNA polymerase types, so then how much of introns? No matter if it is intron present, if it is 1% or if it is one intron, or if it is 10 to the power 5 introns means introns, that's it. Okay, got the point. So you have to be somewhat just advanced in the thought process, right? Okay, need to be advanced because such kind of things are tested regularly. So here and there in the our papers also, in around this 500 MCQ, this will be tested, right? Okay, that now come to the question. Presence of histone proteins bound with DNA, both domain archaea and eukarya. So histone proteins are bound in DNA found in both archaea and eukarya. That is correct, right? So then presence of introns in many genes, no? Presence of introns in many genes, right? Uh, right, presence of introns in many genes. That is not correct for both, no? Then when we come to presence of many kinds of RNA polymerases, that is correct, many kinds of RNA polymerases. Growth is not inhibited when exposed to antibiotics. That is also correct. Bacteria get inhibited when exposed to antibodies, not archaea and eukarya. Then presence of branch hydrocarbons, then this is incorrect because this is relevant to, right? This is relevant to what is relevant to archaea, right? Archaea only, right? Got it? Archaea only. So therefore, the answer for the 92nd one is A, C, and D, no? Right? So answer is answer number 2, no? Right. Then, right. Right. What is say cell surface? Where that it says over the cell surface on the resource book. Forms a protective layer over the cell surface, no? First one, no? Right, over the cell surface. It's given that way, no? So, over the cell surface. On the cell surface, anyway. Right, I think in the single medium one, that's stated in the cell surface or kind of something like that in the anyway. Yeah, thank you. Yeah, then we can take this as a correct one, right? So over the cell surface, it is correct, no? Right, right. So over the cell surface, that is correct. Then A correct, B correct, C D. Yeah, then A B C D all correct, right? Okay. Again A B C D correct. Then E is not correct. So therefore 5 is the answer. A, B, C, D are the answers, right? Okay, thank you very much. Okay, yeah, over the cell surface, no, forms a protective layer over the cell surface, right? Yeah, in the functions, yeah, okay. Thank you very much. Then, yeah, that can be then taken as correct. Okay, sorry. Right. Then, move to the second, that 92nd one, right? Okay, that's okay. Okay, no, right? Then we'll move on to the 93rd, right? 93rd. Okay, make this correct, right? That C statement forms a protective layer over the cell surface. That is correct. Previously, I marked it as false, right? Then that is correct. Okay, so make it as correct. Right. Now, okay. When come to question number 93rd, deficiency of which of the following vitamins or minerals would cause reducing of immunity? No, immunity reducing is because of retinol and then what else? Then iron, no, right? Iron, because of retinol deficiency, it leads to immunity impairment, and also iron deficiency leads into immunity impairment. Retinol means actually vitamin A. Retinol is the active form of vitamin A. That's why it's retinol. So niacin, calcium, thiamine, those vitamins and minerals are not included for that. So then answer is C, D means answer 4, right? Then come to 94th one, right? Places where sodium chloride is actively reabsorbed in the human nephron. No, human nephron. So now sodium is actively reabsorbed, no, right? Sodium chloride is actively reabsorbed. You have to circle the question. Then proximal convoluted tubule, that is okay. Sodium chloride is actually reabsorbed. That is correct. Then now for this one, you have to have that diagrammatic representation of right. Okay. So hemo hemophilus is a virus, so therefore no need to uh, italic, right? Okay. right? It should not be italic. Okay, got it? Right? Then, right? Are you asking the second one, no? Hemophilus influenza. That influenza, they are for the eye. It means for the conjunctivitis. That is Hemophilus virus, right? Okay. Here, Hemophilus influenza is a bacteria, right? The Hemophilus influenza is a that gram-negative bacteria. So there you should italico. It means you have to underline. You have to use the scientific nomenclature there. Got it? Who has asked the question? No name anyway. So that's the thing, right? So Haemophilus influenza is a bacteria, okay, that causing the bacteria meningitis. Fine, right? Then for this one, for the question number 94, you need to have that diagram where that now proximal convoluted tubule there, then come in the descending loop, then come in the ascending loop, and then go the distal convoluted tubule, and then get the collecting duct. Likewise, then here what happened? Then reabsorb, then secreted, then here again reabsorb, they are secreted, then no secretion actually here, but we'll write it like this. When reabsorb, when here reabsorb, then secreted. Likewise, you have to have a keen understand, keen knowledge regarding the these ions and substances which are reabsorbed and secreted here throughout the pathway, right from the filtration, which is glomerular filtration, no, which is filtered by the glomerulus at the Bloman capsule. So therefore, you have to keep keen on this, and we'll discuss these scenarios and what will which one at the proximal, which has the distal during the that ninth ninth session, no, ninth seminar, right? Regarding that endgame seminar, we'll discuss it. Okay. So as proximal convertible, it is okay that the sodium chloride is actively reabsorbed. Then not in the descending limb of loop of Henle, that is not happening. So then ascending limb of loop of Henle, sodium chloride is actively reabsorbed. That is correct. Then distal convoluted tubule, once again sodium chloride is actively reabsorbed. And in the loop of in the collecting duct also sodium chloride is actively reabsorbed. But it is asking about the human nephron. So collecting duct is not a part of human nephron. Got it? That's why this is incorrect. But as a when we come to a as a part, as a segment, that is correct. But when come to the nephron, now human nephron, when we come to the nephron only contain these parts, right? Nephron only contain these part, Bowman's capsule, right? Then proximal connected tubule, then ascending limb of loop of Henle, the loop of Henle. Actually, the loop of Henle is ascending and descending all together. Then here descending loop of sorry. Here we come right, descending limb of loop of Henle, ascending limb of loop of Henle, right? Then we come to distal convoluted tubule, right? And then finally come to the collecting duct, but it is not included. So these are the parts of nephron. These are the included, right? Then like what about the glomerulus? Then that peritubular blood capillary network, PBN. It means peritubular blood capillary network, right? So then vasa recta, then all these are glomerulus, peritubular blood capillary network, vasa recta, all these are blood vessels, right? So therefore not part of nephron, they are blood vessels, right? Then what about the collecting duct? For the collecting duct, it is opening into several nephrons, not only one. Now if we take a collecting duct like this, 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 then one nephrons into here, right? Another nephrons drain into this, right? Then another nephron drains into this. Then another nephron drains here into this. Another nephron drains here into this. Likewise, now 1, 2, 3, 4, 5 nephrons are drained into the certain similar one single collecting duct. No, the nephron is drained into the single collecting duct. So therefore, there are several nephrons collecting. Therefore, you can't say that this particular collecting duct is included for this nephron, right? That's why parts of the nephron, they are only included these things: Bowman capsule, proximal tubule, descending loop of Henle, ascending limb of loop of Henle, and distal connected tubule. Only this, and these comes under blood vessels. And the reason that collecting duct is not included, because they are right, they are not the collecting duct is not specific for a certain particular nephron. That lots of nephrons are draining into the collecting duct. Okay, got it. That's the reason, right? So then that's why E is incorrect. Otherwise, E also correct. Then answers are A, C, D. Answer number 2, right? That's a good question, right? A good question means covering the parts of the nephron. But when in the exam, if you ask to write the nephron with associated blood capillary networks, like it was in 2023. In 2023, they asked to draw the structure of a nephron with associated blood capillary networks. Then you have to draw the glomerulus, pellotubular blood capillary network, vasa recta, and the right. Okay. So likewise, so that's the way to go. But if you ask only the nephron, then all those things part are not included. Only that five parts are included there. Then structures and all the structure that is are present in the dermis. When come to dermis, blood and lymph vessels are found in dermis. Sensitive nerve endings found in dermis. Sweat glands are found in dermis. Sebaceous glands are found in dermis. Hair erector muscles are found in dermis. Therefore, all these A, B, C, D, and E all are found in the dermis, right? When come to 96th, when come to question number 96th, right? We'll say, select the correct statement regarding the accessory glands of the male reproductive system. Accessory glands of the male reproductive system. In the proximal convoluted tubule, sodium ions are absorbed actively and chloride ions are absorbed passively, right? Yeah, sodium and chloride are separately given in the sometimes given as separately, but in the diagram you see in the diagram at the end of the selective reabsorption and all those things, they are in the proximal convoluted tubule. You can see that sodium chloride in a red arrow in the resource book, right? Yeah, actually what is happening, sodium plus sodium ions are absorbed, it means selective reabsorbed actively, and chlorine ions with the reabsorption of sodium ions happening to maintain the ion gradient, right? Actually, what is now, this is not the time to discuss those things. Now, what is happened now? Think here. Now, this is the proximal convoluted tubule. Now, actually, this is the outer environment. Now, when see sodium ions are reabsorbed there, then what happened? Outer side positivity, what happened? It is increased, right? To neutralize it, what happened? Now this is taken place with the use of ATP. It means this is an active process, right? Okay, but to neutralize this positivity, what happened? Chloride ions passively, this is happen passively, right? This is happen passively. Then passively chloride ions are reabsorbed. So then what happened? That ionic gradient, that positivity is balanced by this negative charge in the chloride ion. That is what happening. So, but altogether, when we write sodium chloride is actively with the use of ATP is reabsorbed. Got it? Right? Got it. Is reabsorbed. That's the point. Now, okay, the one who has asked the question. Okay. Then we come to 96, right? Accessory glands. Then A is correct. No, the seminal vesicles produce a thick yellowish fluid. Cooper's glands secrete a clear alkaline mucus. That is also correct, right? So you have to go through those glands and the secretion types. Anyway, we'll try to discuss this part also in that common pattern, or that that the seminar, that the endgame seminar, right? Okay. All of them are paired glands. No, only that prostate gland is a single gland. Apart from that, that seminal vesicles and Cooper glands, they are paired glands, right? So all of them are paired wrong. Prostate is a single gland. It's a single, right? It's a single gland. The fluid produced by seminal vesicle make up of the about of 60% of seminal fluid. That is correct. Seminal vesicles now of the fluid, if we take now the seminal fluid is what it is forming, no? For the seminal fluid. Now contributions coming from different aspects. Now here one and here another one, right? So 60% is from what seminal vesicles, then 30% from prostate glands, then the remaining 10% from the corpus glands or the bulbourethral glands, right? That is the seminal fluid composition, right? Okay, so better to keep it in mind. And so major contribution is by the seminal vesicle, then the least by the corpus gland, right? Okay, so all the seminal vesicle is a double paired glands. This is a single gland, and corpus is a double paired glands, right? Okay. Seminal fluid has an acidic nature, mind you, that not the seminal fluid or any other semen, that seminal fluid. Actually, the seminal fluid is the correction of that seminal vesicle fluid, prostate fluid, corpus fluid, and all those fluid make a fluid of what? An alkaline solution, right? It's an alkaline solution because in the vaginal tract, that vagina is acidic. Vaginal tract, the secretions are acidic. So when what happened that when if the seminal fluid also become acidic, that the acidity is increasing and the fertility is there question, no? Fertility may become lost or maybe reduced because of the acidic nature there. So therefore, to balance that acidity and to maintain a proper pH balance, that seminal fluid is alkaline in nature. When that is ejected into the female vaginal tract, that the alkaline nature pairs with the acidic nature in the vaginal tract. Okay, got it. So if they ask to draw the nephron with associated blood vessels, we still can't draw the collecting. Yeah, you still can't draw the collecting duct because it is not the part, no, right? So it is not a part of the nephron. Got it? Right. Then this is incorrect. It is actually alkaline, no? It is alkaline. It is incorrect. Then A correct, B correct, and D correct. Therefore, answer is answer number 1, no? Answer number 1, right? Then we'll come to question number 97. 97 is regarding the right now see that regarding the processes, no, that we have discussed, that 3 processes in temporal bone and 2 processes found in the mandible, no, right? So 97 will come. Zygomatic process forms the zygomatic process in the temporal bone. That is correct, no? That zygomatic process. Okay. Then styloid process provide the facet for muscle attachment. Yeah, that is correct. Condyloid process articulates with the temporal bone to form the temporomandibular joint, right? That is not the the articulates with the temporal bone to form the temporal mandibular joint. Select the correct one regarding the process in the facial region. No, it is asking, right? Okay. Then coronoid process, then see a process found in the temporal process, right? So in the C, what is the mistake there? Condyloid process, okay, articulates with the temporal bone, right, to form the temporal mandibular joint, right? So now we know zygomatic process, then styloid process, mastoid process, then coronoid process, and the condyloid process. Those are the processes, right? Those are the processes found in the both the mandible and the right temporal bone, right? So therefore, now regarding the condyloid one, condyloid is the latter part. Now if we take the mandible like this, there is a coronoid process, and in the latter part there is condyloid process. This is how the mandible is arranged. Now this is the condyloid. This is the condyloid, right? This is the condyloid, and this is the coronoid. Okay, those are the process, no? So articulates with the temporal bone. Yeah, it's articulate with the right. Okay. It's articulating with the temporal bone. It means with the zygomatic part, no, right? Okay. That is in the connects to the skull. So for the jaw movements, no. articulates with the temporal bone and to form the temporal mandibular joint. That is correct, I think. Condyloid process correct, no? Right, that is correct. So articulates with the temporal bone to form the temporal mandibular joint. Regarding the process in the facial region, that is correct, no? Right. Then right. Okay, we'll see. Now, coronoid process, a process found in the temporal process. That is correct. No, a coronoid process is a process found in the, not in the temporal process. It is found in the mandible. No, it is mandible. Therefore, that is incorrect. Mastoid process provides a phase for attachment of muscles and ligaments. No, mastoid process provides a phase for attachment of uh, muscle attachment and for ligaments, and mastoid process it is given, it is providing the attachment only for muscles, no ligaments given, right? In the resource book, no ligaments given, therefore that is incorrect. Then what happened, then it is asking about the processes in the facial region now, right? Facial region now means zygomatic process, facial, styloid, once again muscle attachment like found in the facial region, so therefore no issue with that. So with the processes found here, so with the processes found here, now A, B and C, three are correct, no? All those three are correct. So A, B, C answer is 5, no? Right, answer is 5. Here it is incorrect. That's not the temporal bone, it's the mandible. And here no ligament is mentioned there for the mastoid process. In the resource book only gives the provides a face for the muscle attachment. That's it, right? Okay, then 98 we come. Plant species found in tropical thorn scrubs. Guineandra found, Ranavara found, and Hirasa found, not Ebony or Val Kurundu, right? Okay. So therefore answer is 5 once again. A, B, C are the answers, right? Then come to question number 99. Example or examples for antiseptics. Antiseptics, right? Antiseptics is an R. What are the antiseptics? Now here, antiseptics and disinfectants, we have to understand about two, right? There are two. Taste them. Antiseptics. Antiseptics, right? Regarding antiseptics, there are two actually. One is ethanol. One is isopropanol. Sorry. Isopropanol. Then the other one is chloroxylenol, right? Right. Then we come to disinfectants. When we come to disinfectants, there you can find the phenol, right? Then hypochlorites like calcium and sodium, no? Hypochlorites, calcium and sodium hypochlorite, no? Right? These are the disinfectants and antiseptics. So antiseptics are ethanol, isopropanol, and chloroxylenol. Actually, chloroxylenol is what is given there. Chloroxylenol. Here you have to correct it as chloroxylenol, right? Anyway, C, D, E are correct. Phenols and hypochlorite, these are what they are? Disinfectants. No, they are disinfectants. That's the reason. So therefore answer is answer 5. Answers are C, D, E. Right. This is a kind of a note, very important. Very important, can be given in an MCQ, right? Then question number 100. In food-related infections, microorganisms enter the host body when spoiled food is consumed and release toxins that are characteristic for disease symptoms they cause, while they grow and multiply within the body. Examples for such microorganisms is an right. Okay, we'll see. Now, there are two types of actually diseases associated with food. No, we'll say food associated diseases. Food associated diseases, there are two types. One is food-borne infections. Other one is food intoxication. Right? Food intoxication is the other one, right? So food associated diseases are two types: food-borne infection and food intoxication. Now what is happening here? In foodborne infections, that we are taking the food, right? Okay. Right. So in the food, if we take the food as this, then actually pathogen is inside the food actually, right? Then pathogenic microorganisms are inside the food. So then that the food is somewhat spoiled, and food is actually spoiled. There's no issue of that. Food is spoiled. It means microbes are contained. Then what happens once it is consumed, right? Then once it consumes, right? So this is consumption. Now it is consumed. After the consumption, what happened? What happened now? This is, imagine this is a body. Then toxins are produced inside, no? Toxins are produced. Now, in food intoxication, what is happening? In food intoxication, what happening? That already, now this take a while, right? This takes a while. This takes a while means they take some time actually, right? Okay. So takes while means takes time. Takes time, right? Okay. Then it comes to food intoxication. What happened? Now food is there. What is included in food? It is toxics. Toxics because of what? The spoil, no? Right? Spoil. Not toxin means that someone is giving toxin to them. Right? Toxics are included because the food is spoiled. Toxins are because of microbes, right? Toxins are because of microbes actually, right? Because of microbes, that toxins are because of microbes. There is no issue regarding that one. And then what happened? Once the food is consumed, what happened? Intoxication take place. Right? Food intoxication take place. This is quickly taken place, right? Within a short period of time. Right? That the symptoms and all those things, short period of time. What is symptoms? Ne? Here, symptoms take while. Take a while, right? Okay, that is what is happening. Okay, so that is the difference between foodborne infections and food intoxication. So here, then Staphylococcus aureus and Clostridium botulinum, right? So they are responsible now. Salmonella typhi, Shigella, and Vibrio cholera. Now we come to food infections. We can write down the what types of food intoxications are. The diseases are typhoid fever, right? Because of Salmonella typhi, then dysentery, then finally cholera, right? Then we come to food intoxication, that is food poisoning, right? Then botulism, then aflatoxins, right? These comes under food intoxication. These are the diseases, right? So then what is asked here? It's regarding now carefully read the question, right? Now carefully read. In food-related infections, microorganisms enter the host body when spoiled food is consumed, right? Spoiled Very unfortunate now that everything has been gone, I think. Is there any way to recover? I think no. I think you have no. I think you can freeze the that YouTube video, right? You can freeze the YouTube video because that in the PDF, I think this is a massive PDF, right? So therefore, some stuck. The video sometimes stuck, right? So you can freeze the YouTube live some seconds prior, then you can take down note. Anyway, now when we come to the question, it is saying that now what is the condition that microorganisms enter the host body when spoiled food is consumed? That is right. Then release toxins that are characteristic for the disease symptoms they cause, right? Okay, while they grow and multiply within the body. Now that is what happening now. So therefore, this is what foodborne infection, definitely a foodborne infection, not a food intoxication, because toxins are not consumed, right? What has consumed is spoiled food and it causes toxins inside. No, this is a foodborne infection. What are the foodborne infections then what are the foodborne infections? Then it is Salmonella typhi causing typhoid fever. Then Shigella causing dysentery. Then notice Staphylococcus aureus, it's causing that food poisoning. Then Vibrio cholera causing cholera. And Clostridium botulinum, no, it's causing botulism. That is also food intoxication. So foodborne infections are A, B, and D. Answer number 1. Answers are A, B, D. Okay, right. Now 100 questions done, right? 100 out of 500. It means around 20% of today's work is done now, right? Are you okay, guys? Drop a message. Any issues or any mistakes or anything from my side that any explanation that you need further, any clarifications to be done? Now 100 done, 100 MCQs done. We have to move on, right? Just drop a message. All okay? All right? All right with you? Okay. Nice. Okay. Okay. All right. Right now we'll move towards the 101 from 101 onwards, right? Now, a characteristic shown by both living organisms as well as non-living things. What are they? The growth, crystal growth, and the what the other one is the movement of waves. Now here, what is shown by both living organisms as well as non-living, that is the growth. No, so therefore, third one is the answer. Really easy. Now, growth means crystal growth, that non-living character. Then again, wave movement. Those are the both non-living, found in both living and non-living. Right. Okay. So anyone you need to take the tute, you have to take it from the site, right? So no tute posting or anything in the Telegram. That's the reason now, because there should be a order tute here and there. So there is no order like that. Now we have maintained the website for the entire year, no, for the class now. So we have to continue it with the these sessions also. Even though these are free sessions, we have to continue we have to maintain that standard as a class, right? We are maintaining the standard, okay? So that's why we are not sending those PDF into the chat or into the Telegram channels, right? So you have to get it from the website as official, right? So that's what you have to do, okay? Then 102, and aldose and the ketose monosaccharides respectively are, right? So now we have discussed this point in the Friday, that last Friday, the Poduratava seminar. When we take our monosaccharides, we can divide, we can divide them into two, no? Right? We can divide them as we can divide those monosaccharides as with the availability of their carbonyl group. We can divide them as aldose. And other one is ketoses. Aldoses have that COO RH type, it means aldehyde type group. Then ketoses have that ketone type group, RC double bond O R, right? So for ketoses, we have two examples. Those are the fructose, then ribulose, right? Okay. Hari. Then what about aldoses? Then aldoses, there you have glyceraldehyde, then erythrose, then you see glyceraldehyde, erythrose, then you come now apart from these two, then you have ribose, then deoxyribose apart from ribulose, deoxyribose. Then you have what else? You have then glucose, then you have fructose, not fructose, you have galactose with you. So C3 done, C4 done, C5 done, C6 done, right? This is the classification. So you have to keep in mind. Now just quickly note down this one. You can divide them as aldoses and ketoses. This is the classification, right? Okay. One is mentioning that he or she is not having data to download, right? Normal data. Then what? How that one is uh, there in Telegram? I don't know whether there are Telegram packages. Are there? Are there? Aman? I don't think so. No Telegram packages even. No, Telegram is consuming normal data. No. Right. So how he or she is there on Telegram? I can't understand. Right. I think it's not consuming more data. Just 2 to 5, 10 to 5 to 10 MBs, right? For the download. I think at this time, one without data is in Telegram, kind of a ghost thing, no? Impossible, not impossible, that is impossible, right? Anyway, now don't give you attention for anyone in the discussion or anything like that, right? Just uh, just continue on the session, right? Don't get distracted by those guys, right? Then so aldose and ketose, no? Then come to aldose. Now ketose, you have to know erythrose is not a ketose, glucose is not a ketose, aldose not a ketose, ribulose, fructose, yeah, they are ketoses. The ribulose, fructose, both are ketoses. Answer is answer number 4 for the 102nd one. The combination of magnification from the eyepiece and the objective lens of a microscope to observe the maximum number of cells within the observable area is, now for the magnification, for the magnification of a microscope, magnification equal magnification by the eyepiece into magnification from the objective lens. This gives the total magnification, right? Okay. Now you have to understand there are two aspects, right? Two aspects, right? Two aspects, right? Then two aspect. One is to observe to observe maximum number of cells. The other one is to get full details of a selected cell or selected one, right? A selected one means selected cell, right? Selected cell. Okay. Now to observe maximum number of cells, what we have to do, right? To observe maximum number of cells, right? Maximum number of cells means that that magnification is now once we magnified, what happened? Now it is asking a microscope to observe the maximum number of cells. Now in a selfie, if we need to take the all the guys in the one frame, then what we use like 0.5. No, we are not going to 10 no, 10 no, 1 no, 5 like that. So we are using the smallest available that magnification, no, right? So here also to observe maximum number of cells, we need the what? We need the minimum magnification, but we don't get details of a certain particular one, but we get the all covered. We need the minimum magnification. To get full details of a selected one, we need to have maximum magnification, right? The minimum magnification and the maximum magnification. So here to observe the maximum number of cells, it means the minimum magnification. What is the magnifications here? Here 200, here 300, here 400, here 500, here 1000. What is the maximum? The minimum magnification is the first one, no? Right. So therefore, answer for the 130 is first one. This is a very important one, right? Very important one. Very important point that comes under the practical side of the microscope, right? Practical side of the microscope. Okay, practical side of the microscope. Okay. Anyway, then come to 104. A single membrane bounded organelle. Now nucleus is a double membrane. Golgi is a single membrane. Ribosome, there is no membrane. Chloroplast, once again double membrane. Mitochondria, double membrane. Then what is the answer? Answer is 2. This is non-membrane, right? Ribosomes. Then what are the non-membrane ones? If you go on through non-membrane bound are the ribosomes, then you can have what? You can have centrioles. They are non-membrane. Then no centrosomes. Cytoskeleton. They are non-membrane bound, right? They are non-membrane bound. So keep in mind others. You know now double membrane bound are the given ones: chloroplast, nucleus, and the mitochondria. Those three. And the rest are single membrane bounded. Then ribosomes, centrioles, and cytoskeleton. Those are membrane bounded. No, right. Then come to 105 question. The correct statement regarding cancers. This is also kind of a great guessing because cancers are not tested earlier, right? So therefore, there is a very good possibility of cancer MCQ from cancer, maybe SAO, maybe SCQ part. Anyway, so there's a great possibility anyhow. So you have to go, right? ප්‍රශ්නයක් දාතන බෝන් ටිෂු එක ලිවිං කියලා හිතනවා ඒත් ඕල්ඩ් ඒජ් අයගේ ආයේ ග්‍රෝ බැක් වෙන්නේ නැහැ කියලා හිතනවනේ chat GPT කරාම ඒක කිව්වේ both living non living දෙකම කියලා වගේ එකක් අපිට MCQ එක select living non living tissue කියලා දුන්නොත් bone tissue එක වඩා හොඳ living කියලාත් ගත්තොත් කියලා මට හිතෙන්නේ ඒක හරි නේද. Yeah, actually bone tissue become non living means actually it's doesn't continue the growth fact. It means growing, no, after some certain particular age that that there's a growth part known as epiphyseal plates, right? Those epiphyseal plates are not growing furthermore. So in that stage we are referring that the bone is not growing further. That's why that bones are getting weaker in all that means in old people, in grandparents, when the fracture taken place, that their recovery is actually that impossible. The impossible means recovery is very delayed, or maybe it's cannot be taken place. But in younger ages, in younger ages, in children's, maybe some fractures take place. They even don't know whether fracture take place. Sometimes it normally fuses. It means due to the growth of the bones, no, the growth, rapid growth taking place. So that's the reason. So therefore, but normally you take bone tissue as a living one. Okay, I just now saw the message. Okay, that is what is regarding the bone tissue. Okay, right, good question. Okay, then come to the 105. Normally cancer cells respond to the control mechanisms of the body. No, they don't respond, right? Shows no response, therefore that is incorrect. Cancer cells consider the singles that regulate the cell cycle. They are not considering, right? They are not considering, right? They are not considering. So, and for the first one, they shows no response, right? No response. Then third one, many malignant tumors are cause for dangerous issues, right? Yeah, many malignant tissues, actually tumors, uh, actually all the malignant tumors are actually dangerous. But just going through with the resource book, they say that many malignant, there can be times that even malignant tumors are not dangerous. Now, when we come to tumors are two types, right? Okay, tumors are two types. One is benign tumors, right? Benign. Other one is malignant tumors, right? These are the two types. Then malignant tumor, you name it as when someone having malignant tumor, we saying that one is having a cancer. This one, you are not saying that this guy is having a cancer, no? So this is the tumor classification: benign and the malignant. Malignant means cancer. Benign means there's no such cancer, right? Okay. Then many malignant tumors are causes for dangerous issues. Actually, that is correct. No, you have to take it as correct. So many praise is also given in the resource book itself. So therefore, you have to take it as correct. Benign tumors cannot be completely removed via surgery. No, benign can be removed via surgery. Right? Benign can be removed via surgery. Right? Okay. But we can't. Right? We can't remove malignant via surgery because they have been spread. They have been spread all over. They have been spread. No, spread. We name it as for the spread. We name it as what? Metastasis. No, metastasis. We name that is spreading as metastasis. Right? Okay. So benign tumors cannot be completely removed via surgery. That is incorrect. They can be completely removed by surgery. Spread of cancer cells to locations near the original site is known as, now metastasis is the spread of the cancer to the near the original site, where far from the original site, not near the original site, far away from the original site. So for the metastasis to take place, the cells have to be spread to far away from original site. Like now, if the cancer is there in the stomach, it has to deviate to duodenum, or maybe to the lungs, like maybe to the brain, likewise. So for a far away from original site, far away from original site, right? Okay, so metastasis had to be happen, not the near the original site. Okay, so that's why this one is incorrect now, right? Then 106th, right? Correct regarding the energy conversion combinations, ne? Right? Light used in bioluminescence, no, not in transmission of electrical impulses. Light is used in bioluminescence. So therefore, this is incorrect. Light is used in we write bioluminescence, no? Bioluminescence, right? Okay. So then electrical energy is used to maintain body temperature. No, body temperature is maintained by what? Heat energy actually. So then that electrical energy is used for the transmission of electrical impulses. Here it is electrical energy. Electrical energy. Now electrical energy is used to transport, but used in transmission of electrical impulses. Here, this one, right? Then heat for not for bioelements, it is for the maintain body temperature. That is incorrect. Then kinetic energy for the muscle contraction, that is correct. Chemical energy, synthesis of various compounds, that is also correct. Then there are two answers, fourth one and the fifth one for the 106, right? Then we come to the 107th question, number 107. Question number 107, right? Select the correct statement regarding the allosteric regulation, of enzymes. So you have to be keen on this. This one is also a kind of a great guess one, right? Okay. Then the molecules that naturally regulate enzymatic process act as non-competitive reversible. No, they are they are acting as non-competitive, then reversible inhibitors. Normally, non-competitive are irreversible because they can't again bind to the molecule. No? But what I mean, they are not detaching. No, they are permanently bound to the molecule. So therefore, they are irreversible. But here, keep in mind allosteric regulation, that is non-competitive and reversible. That is a very key phrase. That is a very key point. Allosteric molecules, or in the allosteric regulation, it is non-competitive and reversible. Normally non-competitive are irreversible. That's the point. Now because of that knowledge you have, you normally, now I will take this by side. Normally we say now competitive are reversible. No, we normally say competitive are reversible. No, we say that. No, reversible. And we say now then again we say non-competitive, we say non-competitive are irreversible. That is how we normally say. No, that is how we normally say. Non-competitive are reversible, then competitive are reversible. That's how we normally say, right? Okay, got it. That's how we normally say, right? That's how we normally say. But what is happening here in allosteric regulation, non-competitive and reversible. So that is kind of a mixing of these two, no? Non-competitive and reversible. This keep in mind. Like you, then therefore first one is correct. Regulatory molecules bind to the active site of the enzyme. No, they are binding into the regulatory site. Okay, they are binding into the regulatory site. Keep in mind, they are binding into regulatory site. Most enzymes that are regulators as such are made up of one or more subunits, right? So most enzymes that are regulated as such are made up of one or more subunits. They are saying no, they are made up of what? Two or more subunits. At least there should be two subunits. Now normally allosteric molecule like this, they needs to have at least two. Then this kind of a molecule made up of four subunits. This 1, 2, 3 and 4. So here the active site, here the regulatory site. Likewise, okay. So active site, and this is the regulatory site. Okay. So then not one or more. Actually, to be a allosterically regulated one, actually two. Two is the least. Okay. And two is the least. So two or more need to be there. Two or more, right? Two or more, right? Okay. Then ATP is an allosteric, not an activator. It is an inhibitor. Hemoglobin is an enzyme that hemoglobin shows cooperative. All fine, but it's not an enzyme. Hemoglobin is not an enzyme. Hemoglobin is a protein with quaternary structure. That's it. That's all, right? It's not an enzyme. Okay. That is what up with question number 107, allosteric regulation, a kind of a key. Very good question. Okay. Hundred and eight. Now, in the test related to non-reducing agents, right? Non-reducing agents, 1 cubic centimeter of sucrose solution is added to a test tube along with 2 cubic centimeter of HCl. There is a mistake. The error is now sucrose solution, that is 2 cubic centimeter, and HCl has to be 1 cubic centimeter. Therefore, this is incorrect, right? In the starch test, it means test for the starch, right? Okay, test for carbo, it means test for starch. Add 2 cubic centimeters of 5% starch solution is taken, and few drops of KiI2 is added into it. No, it is 2 cubic centimeter of 5% of starch. No, that has to be 1% of starch. That is incorrect. Okay. Then third one, 2 drops of 1% copper sulfate and 2 cubic centimeter of 5% KOH is mixed in the boiled solution. That is correct. That statement is correct, right? Okay. 2 gram of iodine crystals and, right, 1 gram of potassium iodide, 2 gram of iodine crystals and 1 gram of potassium iodide is dissolved in 300 cubic centimeter of distilled water to prepare the. When preparing the iodine solution, you have to add not 2 gram of iodine and not 1 gram of potassium iodide. So you have to add iodine, right? You have to add, right? Actually, I will say iodine, not iodine. You have to add, right? In iodine, you have to add how much? You have to add 1 gram, and potassium iodide you have to add 2 gram. That is the thing. So this is incorrect, right? Okay, so therefore this is incorrect. Fifth one, to prepare the macerating substance, plant specimen is immersed in HCl and heated for 5 minutes, right? To prepare the macerating solution, you have to add it not into the immersed in HCl, you have to immerse it in nitric, concentrated nitric, right? Okay, concentrated nitric. Right. Okay, that's the point, and this one is also incorrect to prepare the macerating substance. So there can be MCQ from practicals, maybe from the unit 2 itself, right? This MCQ is an advanced one. Now, when you go through this MCQ, right? Right. When you go through this MCQ, you find it difficult to answer. No, there are so many points, important points there. So therefore, this is very might be a very interesting part to test in the exam. So keep in touch with this, right? Okay. I will give some this kind of practical kind of parts which will be tested in the exam on 9th session, right, on Sunday session, right? So anyway, this is quite important points are covered here, right? Okay. Sir, is ribosome a subcellular component? Yeah. If for the organelles which are without membranes, we are named them as subcellular components, right? We name them as subcellular components. That is the point. So keep that in mind also. What is that? If a organelle without membranes are present, we name it as a subcellular components. It means organelles without membranes are known as subcellular components. Subcellular components, right? Okay. Subcellular components. Now we'll come to the question number 109. The kingdom system of classification was first put forwards by, right? Kingdom system classification. Now most will take Robert Whittaker as the answer, but it is incorrect. First kingdom classification was put forward by Carolus Linnaeus because he divided the organisms as plant kingdom and animals and plant, two kingdoms. He divided uh, organisms into two kingdoms as plants and animals. That was the first kingdom classification. That is the first kingdom classification, but it is not that much developed as what is put forward by that Robert Whittaker. But yet you can name it as a that kingdom classification because he used the phrase kingdom, use the kingdom, right? So the kingdom system classification was first put forwards by then who? By Carolus Linnaeus. Okay. So drop me a message those who have understand that. Those who understand that. In the fourth statement, what is the volume of water that potassium iodide did dissolve in? It is correct. It is correct. Huh? It is in 300 cubic centimeter of distilled water. That is correct. Huh? The error is with the iodine crystals and potassium iodide, right? Error is with the iodine crystals and potassium iodide. Rest is correct. Okay. Rest is correct. Then, okay. Right? Okay. Are you okay with the 109? 109th? It's a new point, right? New point for some students, maybe. We have taught this in class. Right. Then question number 110. An incident occurred during the Phanerozoic eon. Right. Okay. The 110, no? Right. Here, I think a statement is missing. No, anyway, no issue. I will give the fifth one, okay? Write down the fifth one in your paper. Increase in concentration of atmospheric oxygen. Right. Is the fifth one. Okay. Now we'll go through. Now, origin of Earth was taken place at Hardy-Weinberg neon, no? Hardy-Weinberg neon. Diversification of vascular plants. Now, when we come to eons and eras, there are Archean, then Hadean, Proterozoic, Phanerozoic. Now, very important thing, this Phanerozoic one is divided into Paleozoic, Mesozoic, and Cenozoic, right? Okay, keep in mind. So any of scenario take place in Paleozoic or Mesozoic or Cenozoic, then that is comes under Phanerozoic cure. That's the point. Now diversification of vascular plants, right? Diversification of vascular plants is under Paleozoic, no? Right? Then therefore it is considered as during the Phanerozoic eon. Otherwise, there are no any other scenarios or cases given in the resource book under Phanerozoic. It is under the Paleozoic, Mesozoic, and Cenozoic. Okay, that's the relationship you have to understand. It's a kind of a hidden relationship. Okay, right. Origin of oldest eukaryotic fossils, right? Then origin of the oldest fossils of eukaryotic cells. It takes place in Proterozoic eon. Proterozoic eon. Proterozoic. Origin of soft bodies invertebrates, that was in Archean. Right. Then increase in concentration of atmospheric oxygen. Sorry, origin of soft bodies not in Archean. It's in actually Proterozoic, right? Sorry, this is in Proterozoic. This is Archean. Increase in concentration of atmospheric oxygen, that is Archean. Right. Therefore, answer is answer number 2. Answer number 2. Right, okay. Hundred and 11. Difference between domain archaea and eukarya. Presence of DNA bound with histone proteins same similar for both. Inhabit different habitats. This is relevant for eukarya. Now the reason for this is now regarding the now we if we come across eukarya and archaea, archaea found in what? Extreme conditions, right? So maybe temperature very high, then pressure may be very high. Likewise, then salt concentration is very high. Likewise, those places where that unfavorable conditions or extreme conditions are available, then archaea are living there. So therefore, they are not finding everywhere. But when it comes to eukarya, we name them as finding everywhere. That's the point. Okay. You might feel now we archaea found it that extreme conditions, but not everywhere. No, it's limited. No, that's why. Then that difference between no, so therefore second one is correct. Methionine as the initiator amino acid for protein synthesis, that is similar for both. Intronsin genes similar for both. Many kinds of RNA polymerase similar for both. Right? What about the second statement? Did you got that, Lamai? Did you got that, Lamai? It's an important one. It's an important statement, right? That some sort of a hidden one with the idea that inhabits different habitats means found in everywhere. Now, our case limited, no, right? PDF link එකෙන් download වෙන්න නැති කට්ටිය Chrome එකෙන් ගිහිල්ලා download කරගන්න. Right, okay. Then here we come to question number 112, right? Body is dorsoventrally flat, right? Parasitic. Excretory system consists of protonephridia, unisexual. Actually, here it discussing about what platyhelminthes no. First one is itself suggests it is about platyhelminthes that dorsoventrally flat. Okay. Then then we have to go through the organism, no, right? So that we know here the tapeworm and the planaria are the two platyhelminthes. Hookworm is not a planaria. Roundworm no, earthworm no. So that roundworm and hookworm they are nematodes, no. Earthworm is annelids. Now these two are remaining. Now when we come to those both tapeworm and planaria, right? The point is now the tapeworm is unisexual, right? Tapeworm is unisexual, right? Then what's the point? Other platyhelminthes except tapeworm are bisexual, no? Therefore, answer is third one, right? As planaria is bisexual, this is incorrect, right? A kind of a very important fact to that separate tapeworm from other platyhelminthes, right? 130. Second, the vascular cambium produces the secondary xylem outside the primary xylem, right? Now, if we take vascular cambium like this, right? So it produces secondary xylem towards the primary xylem, right? Okay. So the vascular cambium produces the secondary xylem outside the primary xylem, that is incorrect, is produced towards the primary xylem. Produced towards the primary xylem, right? So that is incorrect. Both primary and secondary growth occur simultaneously in woody plants, right? Both primary and secondary growth occur simultaneously in woody plants. That is correct. Secondary vascular tissues are produced as a result of the action of co-cambium. Secondary vascular tissues are produced as a result of what? Vascular cambium, lamai, right? Okay. Secondary vascular tissues are because of what? Vascular cambium. Therefore, third one is incorrect. In a typical woody stem, the vascular cambium is present as a continuous cylinder, which is made up of a single layer of undifferentiated cells, right? In a typical woody stem, the vascular cambium is present as a continuous cylinder, which is made up of single layer of undifferentiated cells. Yeah. Now, vascular cambium like this, no? Vascular cambium is like this. So, it produces new xylem towards the xylem and then again phloem outwards. This is what happening. This is how it produces the secondary, that secondary phloem, that we say secondary xylem and secondary phloem. These are direction that is forming new layers, right? So fourth one is also correct, right? Then vascular cambium is present outer to the primary phloem, right? So vascular cambium is present outer. Now what happened? Now if we finalize the structure like this, now this is the arrangement. Now I will arrange these layers in this form, right? So this is now I will take several colors for here for this one. Now I will take this as the cambium, vascular cambium. Then it will be easy for you to understand, right? Then this is secondary xylem, right? Then this is primary xylem, right? Then here we come. This is secondary phloem, and this is primary phloem. Now I will name them. This is primary phloem, and this is secondary phloem, and this is vascular cambium, and this is then we come to the innermost one. It is the primary xylem, right? Okay, this is what happening after secondary growth. So therefore, then what it says, vascular cambium is present outer to the primary phloem. Now this is from inner to outer, right? So if we take arrow, this is inner, this is outer. What it says, vascular cambium is present outer to the primary phloem. Vascular cambium is present inner to the primary phloem. That is now, I think you are crystal clear, right? Okay. So this is very important. So just draw this down. Okay. After secondary growth, this is how it appears. No, this is how it appears. No, right. Okay. Right. Okay. Then we'll move on to the question number 113. No, sorry, 114. An organ that acts as a source during phloem translocation is right. Okay. So now this is very important. It's very important. Now in phloem translocation, there are which act as source, and there are organs which act as sink. Right. Source we know primary it is the leaves, right? And the sink, they are the growing roots, then stems, then buds. That's a very important point, that tubers and then bulbs. What? Tubers and then bulbs. These two, right? These two, right? Okay. These two act as both source and sink. They can act as with the occasion, they can act as either sink, either source, right? So therefore, an organ that act as source during, now tubers, yeah, okay, right? Growing root, no, always sink. Stems sink, buds sink, roots sink. Answer is first one, tubers, no, right? Okay. So why the first statement is incorrect? The vascular cambium produces the secondary xylem outside the primary xylem, no? It's toward the primary xylem, right? Now, production is toward, no? Production not outside. It's producing towards, no? Towards the primary xylem. That's why. Towards the primary xylem, right? That's why first statement is incorrect. Huh? Latika, okay. Now, with the location, primary xylem is inside, or that the secondary xylem is outside the primary xylem. It's according to the location wise, right? But with the production wise, or the cell production wise, that vascular cambium produces secondary xylem, right? Secondary xylem towards the primary xylem. Got it, Lasita? Right? Okay. That's the point. Okay. Now we'll come to the 115. A method of long distance transport is not diffusion, not imbibition, yes, backflow, not osmosis, and not the facilitated diffusion. Right, then 116. 116. Correct statement regarding the ascent of sap. Then this is also a kind of a guess one. Ascent of sap is not frequently tested in the A-level exam. So therefore, be alert on these questions. Right. Correct statement regarding the ascent of sap. It is described using the pressure flow hypothesis. No, it is described using cohesion tension hypothesis. What? Cohesion, then tension hypothesis. It is discussed using cohesion and tension hypothesis, right? Okay. So not the pressure flow hypothesis. That is pressure flow hypothesis is for what? For phloem translocation. Right. So that is not for the ascent of sap, right? Positive pressure helps water to flow upwards to the xylem. Now, you know, in the xylem, that pressure is negative, right? The pressure is negative. So, the not the positive pressure, that is the negative pressure, right? Negative pressure. Therefore, this is incorrect. Third one, xylem sap is transported within the xylem via diffusion. No, it is transported via bulk flow. It is transported via bulk flow. The upward pull for ascent of sap is provided by transpiration. Yeah, that is correct. That transpiration, now if we take a plant like this, now if we take a leaf like this out of the plant, now see what happened. Now transpiration pulls water out of, now transpiration pulls water out of the plant leaf. This out the this ensures a kind of a that space or gap where water is lack. Now this tension is carried out through the stem into the back into the roots, right? So this actually this water loss uh, actually act as a kind of a pull, no? So kind of a pull. Then what happened? That pull that apply on the root and towards from the leaf towards the root, and they just transport and pull water upward, right? That is called the ascent of sap. That is what happening, right? The transpiration is first water is loosed away, right? So water is removed outwards or removed to the external environment. Then as a result of that, a gap is occurs, no? Now once the leaf just now here like this, now water is arranging like this, no? Just imagine water is like this. Now this droplet is moving outward. Then what happened? So this droplets moving here, and this here, and this droplet here. Likewise, if it is kind of this kind of droplets, you just imagine from leaf to root. Now this is going away. Then what happened? This is moving here, this moving here, this moving here, this is moving here. So this that water movement create a upward pull, no? Upward pull. Actually it's indirectly happening as a result of transpiration. So anyway it leads into upward pull. That is what happened. Now this water molecule move here, this one here, this one here. This is lasting till the root to the till the leaves, right? That's it. That is what happening. Okay, right. Okay, nice. Then it's provided by transpiration. That is correct, no? That is correct. Both one is correct. Then the upward pull extends from shoot to root along the entire length of a xylem as a result of addition between. No, if it is water and water molecules, we know that is known as cohesion, not addition, right? It is the cohesion. That is incorrect. So that is incorrect. Okay. Did you understand the xylem ascent of sap or ascent of sap? That's a really good point, right? Really good part which has not tested frequently in past papers. Dear students, did you understand it well? Okay, these are the parts or these are the aspects which can be tested in your exam also, right? These are the aspects which can be tested even during your exam also, right? Okay. I need a bit help from your end. Normally, right? Right, okay. I need a bit help. I think it's not regarding this one. Normally, YouTube lives, they are lasting for 12 hours, right? They are lasting for 12 hours, okay. After the 12 hours, the live is, I think live is not recording, live is not staying, I think. So the parts which I'm teaching after the 12 hours, not, I think now it is around 7:20 around such kind. I think again now 4 hours and 30 minutes are remaining. So just just remind me when it is around how much? 2:30, 6:30, 7, no? Then when it is around 7, not 7 actually, just remind me of when it is around, we just start from 7, no? Just around 6:45, 6:50, 6:50 in the PM. Please remind me, right? Otherwise, this will be a wastage of doing live. It means live students will get that, but afterwards, none will able to watch the live stream. I think that part will be cut out. Okay, it's done by the YouTube, right? It's done by the YouTube because maximum duration of a YouTube live is, I think, 12 hours. I think that the previous part is not there after the 12 hours. Only 12 hours will be included. I think so. The previous parts will be, I think, cut down. That's what happening. Okay. So just remind me. Okay. Right. Then we'll come to the question number 117. Correct statement regarding the life cycle of cycles. Okay. We'll see. The female gametophyte is microscopic. Right. The female gametophyte is microscopic. Right. Then yeah, is microscopic and obtains nutrients from the endosperm. Right. The female gametophyte is microscopic and obtains nutrients from the endosperm. We'll see what is the problem. So that endosperm is arising from the actually from the that male gametophyte, no? So the female gametophyte is microscopic and obtain nutrients from the endosperm. Actually, endosperm provide nutrition to the embryo, no? So the female gametophyte is microscopic, that is okay, no issue, and obtains nutrients from the endosperm. No, actually, the endosperm provides nutrition to the embryo. Actually, no, the once the embryo is now like this, embryo is here. If this is embryo, it is covered by the endosperm, right? This is the embryo. This is the embryo, and this is the endosperm, right? Endosperm. So this provide nutrition to the embryo. That is what happening. So here say gametophyte is microscopic and not gametophyte is not taking nutrients from the endosperm because that endosperm arising after that arising after the fertilization taken place. No. It means that after the embryo is developed, then the fertilization has all taken place. So that is right. That is not correct actually. That endosperm, that gametophyte is not obtaining nutrients. Actually, for the statement to be correct, it has to be that embryo is taking nutrition from the endosperm, or otherwise we'll write endosperm provide nutrition to the embryo. This is somewhat incomplete and that meaningless statement. Okay. After fertilization, the remaining female gametophyte tissues together to produce the triploid endosperm. No, the triploid endosperm is found in where? It is found in angiosperms. Angiosperms, that triploid endosperm is found. But here in cycus, what what happened? The endosperm is in, not 3N, right? Not 3N. Okay. The male gametophyte consists of a branched pollen tube. That is correct. Huh? It's a kind of a very important point, a unique point for cycles, that they are it's kind of this kind of an arrangement now coming towards like, and this what is kind of a bifid, no, right? Kind of bifid. Then it is a branch, right? So it is branch that okay, it's branch that, but the pollen tube is branch. Okay. Branch pollen tube. Gametophytes consist of branch pollen tube. That is correct. Right. Okay. Then right. Okay. Just write down or draw down that pollen tube of cycus. That is the arrangement. Right. This is branch, huh? That bifid kind of appearance. Okay. Pollen grains become the male gametophyte at the microphyte. Pollen grains become the male gametophyte at the microphyte, no, not at the microphyte. They are becoming the male gametophyte in the pollen chamber. In cycles, that microphyte is like this, no? It's just provide an opening, and in the opening, that now this kind of area is there. This is not the perfect diagram anyway. This kind of an area is there. This is known as the pollen chamber. Right? Okay. So that the ovum, the egg, the nucleus, right, is there here, and it fuses with the what, that the sperm coming from that male gametophyte. Anyway, the pollen chamber is there, right? Okay. So pollen grains become the male gametophyte at the microfield, not at the microfield, at the pollen chamber, because this is the microfield. Microfield is just the opening. Okay. Produces biflagellated top shaped sperms, right? That sperms are actually, we name them as kind of arrangement is like this, no? They are kind of top shape, but it is not, we are saying it is ciliated, no? Ciliated top shape, no? Ciliated, right? That is biflagellated, no? It is ciliated. Right? So that is what regarding the sperms also, right? So that is some kind of a brief analysis on cycles, life cycle. So you need to keep those parts in your mind. What about the endosperm nature? That is not 3N, that is N. And endosperm protects the embryo and provides the nutrition. Then the gametophyte is consist of a pollen tube, and that pollen tube is branched in cycles. That's kind of a unique feature which we didn't see in any kind of phylums. And then that the sperm is kind of ciliated, no, rather than biflagellated. And also that pollen chamber helps for the what, for the pollen grain to become the male gametophyte, no, that allow the room or the space, but not the micropyle. Micropyle is just an opening, right? Okay, here, right? Rather than that, there is no such importance. They are providing for the what, that fertilization, no, kind of things, right? Okay. Then we'll come to 180. Okay. Right. Then we'll come to 118. Question number 118. Right. Inhibits the leaf abscission and promotes leaf abscission. The plant hormones that carry the above opposing functions are respectively plant hormones that carry out the above opposing functions, right? Opposing, it means inhibits leaf abscission and promotes the leaf abscission, no? Right? Then in 180, what inhibits the leaf abscission is actually auxin, right? Auxin. Then promotes the leaf abscission is ethylene, right? So answer is third one. Then 119. Correct statement regarding the nutritional requirement of plants. Now see, these questions are found in each and every paper. Now we are doing the third one. Now for the third time, we are having plant growth substances MCQ, then plant nutrition MCQ, right? Fluorine is an essential element for plants. fluorine is not essential. It's not essential. Fluorine is not essential. Copper involves in pollen tube growth, right? Not copper, which is important in pollen tube growth is boron, right? Right. Then purpling of leaf vein is a symptom of phosphorus deficiency. Oh, that is correct. Purpling is because of phosphorus deficiency. Chlorine is a macro element in plants. Chlorine is a macro, not a macro. It's a trace element. Trace element, right? Boron is absorbed into the plant body as H2B, H2BO4 minus, right? What is the form of absorbing boron is H2BO3, right? Minus is the way, not H2BO4 minus. Got it, right? H2BO4 minus is not the way, that the H2BO3 minus. Okay, right. Then that's the scenario we have to talk about. So then come to the point. So then you can just remind that you can calculate it with the oxidation number even, because H2 means 2.2 into 1, boron means 1 into 3, right? Okay. Then altogether these two gives 5 plus 5. Then O 3O means 3 into negative 2. It gives minus 6. Then minus 6 plus 5 means minus 1. So H2BO4 minus, that cannot be stated. No, H2 means 2 into 1. Borons means 1 into 3. Then this altogether give plus 5. O4 means how much? O4 means 4 into minus 2. It gives minus 8. If you don't remember the form, you can try these kind of things. So if it is H2BO4, it has to be 3 minus, right? Okay, therefore this is not correct, incorrect. But anyway, you have to give the correct form in mind. H2BO3 minus. Okay, fine. Did you get that, Lamai? Kind of tricky one, right? Not the correct way to take the answer, right? I just cope with oxidation number to show whether if you didn't find any clue, or if you just forgot what is there, you can just write that kind of a thing, right? Okay, right, right. You will get notice those things, right? Okay, we'll come to 120. So then question number 120, an anticoagulant of blood is, what is the anticoagulant of blood? That is the heparin, no? Heparin. These are very simple things. Now, platelet is a coagulating factor, calcium is a coagulating factor. Then vitamin K is a coagulating factor. Thrombin is formed by prothrombin into thrombin. Prothrombin into thrombin. Then heparin is a anticoagulant. Right. Okay. So then what is doing is control. It means that now prothrombin is converted into thrombin. No? Then what happened? Fibrinogen is converted into fibrin. This is done by the This is done by the thrombin. No, activated thrombin. So what heparin act on these places, right, and providing what kind of negativity, negative impact on the translations, that prothrombin to thrombin conversion and fibrinogen to fibrin conversion. It act as positive manner. So therefore it is a anticoagulant. But here these guys, it means what, right, like is thrombin, then vitamin K, calcium, platelets. Then thrombin is there a positive factor. Then platelets, calcium, vitamin K, these act as positive factors for these translations. Okay, these conversions. Okay, got it. That's the point regarding the blood clotting. That blood clotting may be a very important part. We'll discuss that on Sunday also, because it's a kind of a guessing part, right? Even maybe for MCQ or maybe for a SCQ part, kind of a chart, or maybe it can given as a part of essay, right? We don't know, but better to get prepared for that, right? Okay. Then we'll go to the 121. Right. Right. Then we come to 121. Correct statement regarding the structural features of the alimentary tract. Okay, right? Alimentary tract, it means the GI tract. Okay. Now, once you go through, right? The esophagus is a short tube. No, it's not a short tube. You know, it's around 25 centimeter. It is a long tube, right? So therefore, first one almost incorrect. The upper part of esophagus is made up of smooth muscles. No. So in esophagus, if we divide the esophagus into that kind of two, if like these two segments, right? Then that upper part is made up of what? It is made up of skeletal muscles. Then lower part is made up of smooth muscles, right? Okay. Then sphincters are made up of circular, not skeletal muscles. They are made up of circular smooth muscles, right? Smooth muscles, right? Smooth muscles. The jejunum is the terminal part of the small intestine. No, right? Uh, Small intestine is composed of three parts, right? Three parts. Okay. Starting from the duodenum, then it's the middle part is like this. I will just draw like this, and then the terminal part like this. There is this just small representation. Okay. So first part is this, like this kind of part, and the second part is this till this. Then what is this? Then the terminal part is this, right? Okay. So then if we go around, this is known as the duodenum, right? Then this is so here that jejunum is the middle part, and the ileum is the terminal part. So this is the arrangement of the small intestine, right? Okay, therefore this is incorrect. The internal surface of the stomach is highly convoluted. Yeah, the stomach is kind of this kind of arrangement is there, and inside the stomach you can see these kind of foldings. They name them as rugae foldings, right? Rugae foldings, right? Foldings. That name is not given in your resource book. They are given as what? They are given as circular foldings, right? Circular foldings. This helps in that kind of dilate the stomach when the food is ingested. No, when once the food comes, it increases the size. The stomach can be just kind of expand. No, due to this that foldings. By releasing those foldings, it can increase its size. Okay, so therefore fifth one is correct. So the 121st one answer is 5. Right, okay. Right, okay. I think so. All right, okay. Now we'll move on to the next question, right? Just if you're unable to write it with the time that I'm allowing, you can just pause the video, no issue. You can just complete and come along with the discussion, right? Right. Then we'll move on to question number 122. Okay. Now can you hear me properly? No issue or something with the noise, with the sound, a lack of sound or anything kind of that kind of restriction or any difficulty? Because just no any complaints today anyway. Hope things are really okay. No, things are going moving smoothly, right? I hope so. Just drop a message, okay? Okay, thank you. Right, okay. Then correct statement regarding the histological structure of pancreas. The endocrine part is made up of large number of lobules. Lobules are found in what? Exocrine part. Okay. Lobules are found in exocrine part. Lobules are found in exocrine part. Right. Icers of Langerhans are made up of, right? Made up of a collection of specialized cells, and they have ducts. No ducts in icers of Langerhans, right? So no ducts, it means ductless. Not Douglas, it is ductless, right? Icers of Langerhans is ductless. The exocrine part secretes pancreatic juice, that is correct. Okay. Then the hepatopancreatic ducts open into the duodenum at its, no, not at its end, it at the middle part. This is the largest gland of the body. The largest gland of the body is the what? Liver. It's incorrect. Now, when we move to the pancreas, it's kind of arrangement. No? So here there is the pancreatic duct moving around. Right. So this pancreatic duct uh, this pancreatic duct leads into what kind of right leads into just you see now we'll see these kind of structures, right? These are known as acini, right? Okay, these are known as acini, right? So these are connected with ducts. You can see in the acini there are inside acina cells, you can see like this. There are acina cells, right? Okay. Then here in the pancreas, here also everyone, each one, not only one, everyone is just divided as acini cells, acini. Then here once again acini and acina cells, right? These are acina cells, right? Then these acinar cells in the acini. Got it? Right, likewise. So this is called the exocrine part. So then pancreas is made up of two parts, no? So one part now, if we, when we come to the pancreas, there are two parts. One is what is known as exocrine part, exocrine. Then other part is known as endocrine part. Endocrine part is made up of eye cells of Langerhans, eye cells of Langerhans, where they have alpha and beta cells. Alpha cells secrete glucagon, and meanwhile beta cells secrete insulin, right? Then exocrine part, there it contain acini, right? Okay, that is the arrangement. And the reason and the other important part is exocrine part is present with ducts, ducts present. Endocrine part, it is ductless. That is very important. Okay. Then in the here in the right between this acini, there you can see collections of parts cells. These are known as alpha and beta cells. This is the blue color portion is the that endocrine part. Endocrine part, right? Okay. So main function of endocrine part is to secrete that insulin and glucagon, no? Insulin and glucagon, right? So then the function of this exocrine part is to is the exocrine part. Its function is secreting pancreatic juice, no? Pancreatic juice is secreted in the exocrine part. So pancreatic juice is containing several enzymes. Then we'll see enzymes plus bicarbonate is included there, right? Okay. So here this duct is known as the pancreatic duct, no? This is known as pancreatic duct, right? Pancreatic duct. Then right, okay. Then okay, right. Then what happened? Then they are with the right. There comes the hepatic duct along with carry belly. Sorry, carry bile in with okay. Then it forms the what? They get fused, and here they forms the what? This the hepatopancreatic duct. Hepatopancreatic duct, and it is what released at the midway of the duodenum. Now duodenum is like this. If the duodenum is like this, and then it is released, the bile is really that the pancreatic juice along with the bile is released at the what, the midpoint of the duodenum, midpoint of the duodenum, right? That's the point. That is what just tested here, right? Tested here in this MCQ. Okay, if you need something to draw or to note down, just you can note down these things, right? These are the MCQ points. right, okay. හරි නේද? එහෙනම් අපි ඊළඟට යමු. එහෙනම් ඊළඟට යමු. හරි, right. 123. Right, okay. We'll move on to the 123. Okay. That this is what all. Right. Now you have to keep in mind that duct and ductless or the acini and the isle of Langerhans parts and what is secreted and likewise. Okay. Exocrine, endocrine. 123. The ion that is subject to passive reabsorption in the proximal convoluted tubule and to active secretion within the distal convoluted tubule, right? What is it is asking about? Now, if we take now proximal convoluted tubule and down to loop of Henle, and here we go with distal convoluted tubule, right? So this is the proximal convoluted tubule. This is the distal convoluted, distal convoluted tubule. Okay. Now what is asking here? That's subject to passive reabsorption in the proximal convoluted tubule. So this kind of movement, no? That is passively reabsorbed. We don't know what is that. And active secretion within the distal convoluted tubule. So secreted active. Now red color denotes the active secretion and the blue color denotes the passive reabsorption, right? Okay. So what is the ion? Now here the ion responsible for this one is potassium ion. So potassium here is passively reabsorbed and here is actively secreted. So keep in mind this point. This is very important. Potassium ion it is uh, secreted and reabsorbed differently at two places. One at proximal convoluted tubule, one at the distal convoluted tubule. So these are then K plus is kind of a very important fact. Then bicarbonate also similar process like that. We'll discuss the entire arrangement or the this kind of special. Now K plus is a very important point because this can be tested in your exam also, right? Okay. Because this is a kind of an important point because it's acting or that involving in the that urine production in two different ways at two different places, no? Okay. Right, then move on to the 124, question 124. The autoimmune disease that is caused by the invasion of myelin sheath that surrounds neurons by T cells, that is called the multiple sclerosis, right? Multiple sclerosis, then that's okay. Those are very simple, right? Okay. Right, then we'll move to 125. Right. The disease that is caused due to the gradual degradation of neurons that dopamine neurotransmitters in the brain, right? So this is that now we note about the that there's an important fact regarding these it means two disorders. One is schizophrenia. I will write it down so then you will understand. This is very important point. Actually, these 500 MCQs they are very important, right? I think now for someone who has done past papers might understand these are new points, huh? These are not never tested. Some of these are never. I think majority are never tested in past papers, right? Because we have analyzed the past papers really well in our class by doing guest sessions and all those things. And these papers they entirely based on new questions, right? So schizophrenia. Now, what is the point regarding schizophrenia? Schizophrenia affects neural pathways that use dopamine as neurotransmitter. Now then Parkinson. what is important in Parkinson? It is a progressive motor disorder, progressive motor disorder associated with gradual degeneration of dopamine neurotransmitter releasing neurons in the brain. Right, okay. So now what we have to understand here, now understand, right, the difference, right? So in the Parkinson, it is a disorder associated, right? Disorder associated with the dopamine neurotransmitter. Disorder associated with dopamine neurotransmitter. Actually, the issue is in the dopamine neurotransmitter. That's the cause. Actually, this is the cause. Now you understand. This is the cause, right? This is the cause for the disease. This is the cause. Okay, this is the cause, right? This is the cause. This is the cause for the disease. And here, as a result, that dopamine pathways are, then as a result, dopamine pathways are affecting. This is as a result. Now, this is as a result, no? Result of schizophrenia. Now, this is as a cause for the, this is the cause for the Parkinson, and this is a result of schizophrenia. So here, cause due to gradual degradation of neuron that release, cause due to, what is that? Then it is has to be, definitely it need to be Parkinson. Very, very important, because you feel like both are same but not same, right? One is duck, one is what? Right? Okay, that bourbon and chocolate biscuit, that like that. Okay, right, then that's the reason. Okay, Parkinson and schizophrenia, understand the difference. Okay, really important, very important point, very important point. 126. Consider the following restatement parts of the human brain responsible, no? Reflex, sight and auditory response coordination, that is the midbrain. Controlling the autonomic nervous system, that is hypothalamus. Transmission of information between the forebrain, midbrain and the peripheral nervous system, that is Pons Varoli. Then answer third one. Very simple things, right? Okay. 127. The structure of the human ear that helps maintain the pressure around both sides of the tympanic membrane and atmospheric pressure, right? Now this is ear, right? This is auditory canal like this. Now here the tympanic membrane and middle ear. Then this is the opening, right? Opening of the mouth. Now this is open. Now this is once again open to the external environment, right? Here. Then here also it is open for the external environment, right? At the end of the auditory canal and at the that oropharynx and nasopharynx and all those things, right? So we have to maintain right in the middle ear and here what is happening, right? So if the pressure at here is now 5 is the atmospheric pressure, it is similar, and here it is 5. Now you have to maintain 5 pressure around, it means around the tympanic membrane on the right side also. It means the middle part of the ear, now external part of the ear. the middle part of the ear 5, because if there is a pressure gradient, then the tympanic membrane will be damaged, right? So to maintain the 5 there, that external environment is connected again. So to connect that, what is the structure that is very important? That is the Eustachian tube. Disconnect the, right, disconnect the that middle ear to the that that pharynx, right? Okay, to the pharynx, right? Actually for the oropharynx, right? Also oropharynx and esophagus, and both oropharynx connect the pharynx, right? Okay, then connect with the external environment, then there is no pressure gradient. Okay, fine. So what is the what is the structure? That is the Eustachian tube. Okay, Eustachian tube, very important structure that maintain the pressure at to maintain the pressure at either ends of the tympanic membrane. If one of the side increases, then tympanic membrane will be ruptured. Okay, so that's why if a large sound, or kind of a large vibration of sound wave is arise, it means a high noise, so high sound maybe, then if the pressure of this side is increasing, just imagine if the pressure is increasing than the 5, right, what happen, then this will blast this, it means this will rupture the tympanic membrane. Okay, that's what will happen. Okay, got it. So Eustachian tube is playing a very crucial role, very crucial role, that connecting the middle ear to the mouth, no, with the mouth, no, with mouth means with the external environment, right? Okay, right. 128. Correct statement regarding the, once again, it's regarding the accessory glands of the male reproductive system, right? Corpus glands secrete a clear alkaline mucus, right? That is correct. First one almost correct, right? It means first one is correct. Seminal vesicles secrete a thin milky. No, they secrete what? They secrete actually a that kind of a strong. No, it's kind of a yellowish fluid. No, yellowish color fluid. So that seminal vesicles are not secreting the thin milky fluid directly to the urinary tract through a short duct. So this is this kind of what fluid is secreted by the prostate gland. So this is related for the prostate gland. So second one, that thin milky fluid directly into the urinary tract, it is under what? Prostate. No, prostate. Seminal fluid takes an acidic nature. That is again incorrect. It is alkaline. We earlier also explained. 60% of seminal fluid is composed of the fluid secreted by the prostate gland. Prostate gland only accommodates for what? 30%, right? Ascorbic acid provides, not strength to the sperms are provided by fructose. Okay, it's provided by fructose, right? Okay, so therefore that is incorrect. Then the answer is first one. Right, then we'll come to the 129. The stage at which secondary egg cells are arrested before fertilization. Secondary egg cells, no, right? Secondary egg cells are they are arrested at metaphase of meiosis 2. Metaphase of meiosis 2. Answer number 4. These are very simple, and you need to go through the steps of oogenesis, right? Oogenesis, this is a very good common point, and we'll discuss this on Sunday, so don't worry. Oogenesis and spermatogenesis I will uh, discuss into the deepest of theories, right? It means deeply we'll discuss these two, because there will be surely a MCQ from either oogenesis or from spermatogenesis. This is really important passage of found in the resource book. Okay, right? So this is metaphase of meiosis 2. It is holded at the metaphase of meiosis 2. It's asking about the secondary egg cells, right? Secondary egg cell, not the primary one, right? Okay, right. Then moving into 130th. Correct regarding the ribs. So ribs is kind of a guess or a targeted part where ribs can be tested, right? Okay. So better to keen on that knowledge on ribs, right? Yeah, there are 13 pair of ribs. Okay, 13 pair of ribs. No, that is incorrect. There are 24 ribs. 24 divided by 2 means 12 pairs of ribs, right? Okay, only 12 pairs of ribs. There are no 13 pair of ribs, right? Second one, a kind of curved short, not short. It is a long bone, but it's not long as, right? Actually, it is not that long as other bones, like now we'll see femur, humerus, but relative with the other majority of the bones like phalangeus, right? Then vertebra, right? Like like those bones, when compared with those bones, this is a long bone, right? Okay, that's how it's classified as a long bone. Okay, not the longest, right? Okay, 1 to 8 ribs ventrally articulate with the sternum directly. Now what is happening? Now if we take sternum like this, then 1 2 3 4 5 6 7, right? These ribs 1 2 3 4 5 6 7, they are attached directly, right? Then what happened? 8 9 and 10 are attached with the seventh, not directly to the sternum, right? They are not attached directly to the sternum, they are attached to the seventh rib, right? 8 9 and 10. Then what happened? 11 and 12, they are not attaching to anyone, they are just floating, right? Okay, got it. They are floating, right? Floating ribs. Okay, that is the arrangement. That is how 12 are arranged. Now 1 2 3 4 6 to 1 to 7 direct attachment, 8 9 to 8 to 10 they are attached with the seventh one, 11 and 12 they are not attaching, they are just floating. Okay, so 1 to 8 ribs ventrally articulates with the sternum, incorrect. 1 to, not 8, 1 to 7, 1 to 7. Since the last two pairs of ribs are not attached to the sternum, they are known as floating ribs. That is correct. Fourth one is correct, right? The first two pairs of ribs, that is incomplete, no? So therefore, no need to just go through. That is incomplete actually. The first two pairs of ribs move during inspiration. That is the question. Anyway, you just add it. First two pairs of ribs move during inspiration. That is a very important point. Anyway, this is wrong. Now, if this is the sternum, now first pair of ribs are these, no? Now, first pair of ribs are just kind of static. They are static, fixed. They are at a fixed, no? They are fixed. Now, these are fixed, right? They are fixed and they are static. It means they are not moving, right? What is happening with the inspiration taking place, right? Rest of the ribs they are moving towards the first pair of ribs. It means rest move towards first pair of ribs. That is the point, right? Rest move towards the first pair of ribs, that because first pair of ribs are fixed. That is what happening, right? Okay, so this is the arrangement of ribs. I think you all got the understanding about that. 24 ribs, 12 pairs, then how these ribs are attached. 1 to 7 directly attached, 8 9 to 10 indirect attachment to the seventh rib, then 11 and 12 no attachment, they are floating ribs, right? They are floating ribs, right? Then that is it. And then during the inspiration, first pair of ribs are not moving, they are fixed, then rest of the ribs move towards the first pair of ribs. That is what you have to understand. Okay, then that is regarding the ribs. Then 131. Okay. Polyploidy, very, very important part. Must ask part, okay, kind of a straight guessing, right? Okay. Definitely this need to be looked forward into, right? Okay. Occurs due to the presence of set of homologous chromosomes in each cellular nucleus. This is because this take place because of presence of, right, more than one pair of sets. Presence of more than presence of more than presence of more than one pair of sets of chromosomes. It means, now what is the normal? Now normal, right? Normal arrangement is 2n. So apart from that, if it is 3n, 4n, 6n, 8n, it means all these are, right? All these are, right? They are more than 2n. No? They are more than 2n. So that's the more than one pair of set of chromosomes. That is the reason. Okay. So that is incorrect. If it is presence of a set of homologous chromosome in each cellular nucleus, that is not polyploidity. It is the normal ploidity. If like this, it's normal ploidity. This is used commonly in, not in animal breeding. It is used in what? In plant breeding. Commonly used in plant breeding. Can be induced not naturally. Antimitotic colchicine induce the polyploidity artificially. It's an artificial agent. Artificial, right? Increases homozygosity, right? So it's not increasing homozygosity, increasing what? Heterozygosity, right? Not homozygosity, right? Decreases fertility due to errors that occur during mitotic division. That is correct, because now 2n is the what? 2n is what we known as fertile. Apart from when deviating there, then these kind of things are, these are mostly actually infertile, right? Can be rarely fertile, but infertile mostly, right? That's the thing. So therefore polyploidity, actually the reason, now polyploidity, the main advantage is economical benefit, right? So main advantage is economical benefit here. The economical benefit is the main advantage of polyploidity, not any other thing. In economical benefits to obtain, right? Right, okay, right. Then the economical benefit are the important things to obtain the that. Now if you think now normal banana compared with that, so some banana they are available in supermarkets like that Cavendish, those are really large, no, really freshy. So polyploidity is targeting on those things. That is why mostly used in plant breedings. That is not applicable for animal breedings, right? Not yet, not yet. We'll see what will happen in future anyway. So this is the polyploidity. Now the ploidity levels are beyond the normal. Normal is the 2n. So beyond the 2n, it means 3n, 4n, 6n, 8n. Likewise, then that is called the polyploidity. It is called the polyploidity. They are infertile. Right? They are infertile. That is also need to be understand. Then antimitotic colchicine. This is very important point. Antimitotic colchicine induce this polyploidity, but it is induction is a kind of a artificial one, because we artificially induced it, no, not naturally, right? Okay. Then moving to 132, mental disorder that occurs due to genetic factors, right? So hypertension is not a that mental disorder. So due to genetic factors, what is that? That's the schizophrenia. Schizophrenia is the that mental disorder occurs due to genetic factors, right? Genetic factors, right? Okay. That's the reason. Okay, genetic factors. Now okay, right, this can result into the what that the dopamine harm, no, harm in the, or the degeneration of dopamine, no. So the reason is the genetic factors. Genetic factors, right? Is schizophrenia. Okay, fine. 133 types of RNA out of the above that directly function as the final product of gene. The final product of gene are transfer RNA and rRNA. Apart from mRNA, right? So other two can act as the what a kind of final production of a gene, right? Okay. Then you need to keep in mind now final product of a gene can be three, right? Can be three items. Now final product of a gene, it's mainly first one it can be a polypeptide. Then afterwards it can be a what, it can be a tRNA or can be a rRNA. Keep in mind these things, right? These three, not mRNA, right? Not mRNA, right? That's the point. These are very, very valuable points. Really valuable, huh? For molecular and genetics. I think some students tends to they are trying to skip molecular biology. Why you should skip? Now doing these 500 MCQs. Now it's the 133rd. See how many new points you have obtained. See how many new points you have learned in genetics and molecular biology. So that's the positivity, right? So don't no need to just skip those questions or leave them behind, right? Just going through these points will be there in the exam. Okay, right. Then 134, the largest biome on the earth is the what, northern coniferous forest, right? The largest biome on the earth is northern coniferous forest. So they therefore answer is answer number one. These are direct MCQs, right? Okay. Uh, okay. Wait, what is there a mistake in 132? Yeah, wait, now 132, I think there is, the question is mess up. Wait, you can complete up to that one. What has happened here? A mental disorder that occurs due to genetic factors. That occurs, no? Mental disorder that does not occur. No. Question itself asks occurs due to genetic factors, no? That is there. I think in the previously in the, I think, pilot papers, that was that asking of not a genetical disorder, I think, right? Maybe. We'll see. So now coming to schizophrenia. So it's due to mental disorder, no? Genetic factors, right? Then in the depression, when come to depression, genetic factors cause, no? Right? Depression, it is okay. Schizophrenia, it occurs due to, now, yeah, I think I have totally misunderstand or just mislead the question. So now again start. A mental disorder that occurs due to genetic factors, right? Okay, keep understanding, keep in alert, right? Occurs due to genetic factors, right? Okay, occurs due to genetic factors. Okay, then that is very that's the point. Okay, So then, when come to, right, okay. Okay, so when come to hypertension, what is the reason hypertension is not included? Because it is not a mental disorder, right? It is not a mental disorder, not a mental disorder. That's why first one is incorrect. Then depression, right, occurs due to genetic factors. Yes, depression occurs. Schizophrenia, no, it is schizophrenia. Harry, right? Okay, that's a kind of which have a that which experience now some voices which they can hear that others can't hear. No, that then also then think that others are like to harm them. Okay, so this actually affects that neural pathways which use dopamine. No, right? So this kind of the effect is for the dopamine pathways, but there is no actually no genetic factor is stated there, no, right? No genetic factor is there found as a reason, right? Occurs due to genetic factors. What is that in the question, right? So Alzheimer disease, in Alzheimer disease, right? In Alzheimer disease, genetic factors involved. It's a reason. Then Parkinson disease, genetic factors may cause for the disease. So a mental disorder that occurs due to genetic factors. Then it comes now in the question. It occurs due to genetic factors. Now students understand in the pilot papers that was there. This question was there as the that doesn't occur due to genetic factors. The question was there actually. Then the schizophrenia was the answer. I just read it with that memory and just didn't focus on that the question is about the occurs due to, right? In the pilot papers, it is given as, now I think you can check in your paper that it is given as mental disorder that does not occur due to genetic factors, right? Then it comes with does not occur due to genetic factors. Then it comes with hypertension and schizophrenia, but hypertension is not a mental disorder, therefore it is not, it cannot be taken, and schizophrenia is the answer. That's with that memory that I just that make hypertension that is incorrect and take the answer is schizophrenia. But here it is asked occurs due to genetic factors. Therefore, answer 2, 4, 5 all are correct, right? So there are 3 answers for the 132, right? Got the point, students? Please, I need a message for this, right? I need a message for this. Did you got the point? Did you got the point, right? Did you got the point? Just to confirm, right? Yeah, okay. Yeah, I just mislead that one because of that reason, right? Okay. Okay. Right. Then we'll come to the question. Thank you very much. I think uh, it was said by Herath, right? Thank you very much. Just letting me know that one. Okay, very good. Yeah, then 135 we come here, right? The amount of energy that should be possessed by a primary producer in a food chain of 5 trophic levels. Now, 5 trophic levels, no? We'll start 1, then 2, then 3, then 4, then 5. These are the trophic levels. Now, so food chain moving here from 1 to 2, 2 to 3, 3 to 4, 4 to 5. Then such that the last organism chain possesses an energy of 10 joules. He is getting 10 joules means fourth one having 100 joules, third one having 1000 joules, second one having 10,000 joules, then first one having 100,000 joules. Don't ask me how I find the answer here. Just go through the that Podurata. There are 2 live sessions in YouTube which were conducted on 31st of October, right? They are known as Podurata 1 and Podurata 2. There I discussed 10 points, right? There I discussed 10 points. So in that 10 points, this food chain factor or the short method was explained, right? Okay. So then such that the last organism in the chain is amount of energy that should be possessed by a primary producer. Then what is the amount? That's the 100,000 joules or 1 lakh. 135 answer is fifth one. Okay, because from 1 to 2 when it is transferred only 10% of energy is going through, and from here also 10% out of that the energy which have, and 10%, and during the process that 90% of energy is lost, right? Okay, 90% of energy is lost likewise. Okay, so that's how the transmission take place. 136. The present this is a very important point, right? Very important point actually. So keep in mind, I think normally in papers these points are not touched, right? Normally in papers like even in provincial papers these questions are not available, right? These are hidden parts, right? Hidden parts in the syllabus. Okay. The present extension of Earth and the time period where the last extension, present extension of the Earth, it is in the, right? It is in the sixth extension, and the last extension include the extension of what? Extension of dinosaurs occur. Extension of dinosaurs. Okay. Dinosaurs was taken place at last extinction, that was 65 million years ago. Answer is answer number 1, right? Then 1 for the, then Earth is currently at 6th, and it is the last one was 65 million years ago, right? That's the answer. Okay, right. Then moving to 137. Correct statement regarding microorganisms. Majority of microorganisms are pathogenic, right? No, majority are not pathogenic. Majority are either harmless, or it may be advantageous or harmless, right? Majority are, right, advantageous or right, harmless. They can be advantageous, but if there is no advantage either, they can at least be harmless, right? So majority is this, not pathogens, right? Therefore this is incorrect. The generation time of microorganisms is usually not higher. Generation time is very low, right? Actually not low, actually very low, right? Okay. Growth rate and reproductive rate are low. No, growth rate and reproductive, those rates are very high actually, not high. They are very high, right? Okay. Freshwater and marine microorganisms are the basis of food chains in freshwater reservoirs and oceans. That is correct, no? They just begin the food chains, no? Food chains in freshwater reservoirs and oceans, because there are no plants available, no? So therefore fourth one is correct. Have a relative low metabolic rate. No, they have high metabolic rate. Their BMR is really high. In microbes, BMR is really high. So therefore this is incorrect. Fine. Can someone let me know where can I get the PDF? You can get the PDF from, right? Get the PDF from our website www.bio.lk and go there, and there is a you have to enroll, register if you are a new student. Then there is a in the store there is a class known as that the endgame, right? The endgame, you can enroll there and you can take the PDF, right? Okay. Okay. Then right. Then when come to 138, right? Okay. The majority of soil microorganisms belong to the group. The majority of soil microorganisms are belong to the group of bacteria, right? They are belonging to the bacteria, right? Majority of soil microorganisms are belonging to the bacteria, right? So keep in mind, they are belonging to bacteria. These are very simple points, right? Simple points, but they are very important. Very important, but they are very simple points, right? Okay. Right. Okay. Then come to 139, right? Correct statement regarding the microorganisms that is used to check the quality of drinking water in Sri Lanka, right? So what is the microorganism? The microorganism used here is the coliform, right? So microorganism used here is the coliform. So first of all, you have to understand who is the microorganism. The microorganism is the coliform bacteria, right? Coliform bacteria, right? Okay. Now we'll see. Now, so then its metabolic mode is obligate anaerobic. That is incorrect. That coliform bacteria is facultative anaerobic. Facultative anaerobic, right? Okay. So then they are gram negative, right? So not gram positive, they are gram negative, gram negative, gram negative, and they are once again rod shaped. They are rod shaped, so not conical shaped, right? Okay. So okay. So they are non-pathogenic line in the intestine. So they are pathogenic. No, they are non-pathogenic when they are at the gut. They are non-pathogenic at intestine. No, they are non-pathogenic at intestine. So keep in mind, and also they are not producing endospores. So therefore, second one is correct. Second one is correct. Okay. Right. So then if it is if coliform bacteria, if I take a water sample and there you find coliform, right? Then what you understand? So coliform bacteria is found in the what? Small intestine, no? In the gut, no? Gut. Then through the gut we have feces, no? Then feces are get collected into the water. So that coliform in the water indicates there is a there is a contamination, no? Contamination by feces or some kind of like thing, no? There's a contamination in the water. Coliform, if coliform found in the water, it is a contamination because normally coliform found in the gut, human gut, no? Gut is found in the gut, no? Right, nice, very good. This is a very, very important point. Coliform bacteria. So keen on this. Actually these are the points which will be if it is given in the A-level exam. So this is the entire content or will be tested in your exam too. Right? Okay. Then we'll move to 140th. Right? A step that should be implemented monthly when maintaining domestic aquarium. Right? Okay. Right, domestic aquarium, no? Right. Right. Okay. Right. We'll see. Which have to be implement monthly, no? Monthly. What is that? uh, Monthly we have to know, we have to check, right? So we have to check in the terminals of the light source and we have to uh, clean in the dim, no? Right. So we have to clean in the rim. No? So turning off the aeration is done fortnightly. It means within 2 weeks of time. So then allowing decayed materials to settle. Then allowing decayed materials to settle. That is right. That was also again a fortnightly behavior. Fortnightly one. Then scraping of algae deposited in the glass surface, right? So scraping those things also fortnightly. Then checking the terminals of the light source and cleaning them, it is a monthly one. Then collecting the fish carefully using a using a not basin. That's the incorrect one here. This is also correct. Collecting of fish is also done monthly, but not using a basin, right? They are collected using a hand net and introduce them to basin, right? They are collected using hand net, right? That is the point. They are collected using hand net. Then they are right. Then they are what is doing? Then they are introduced into the basin. They are then introduced to the basin. Not they are collecting by a basin, right? Okay, not collecting by a basin. That is kind of a heck of a task, no? Right? So therefore, collected using the hand net and then introduced to the basin. So here it say collecting the using basin. That is incorrect. It is totally incorrect, right? Okay, therefore answer is fourth one. Then 141st. Examples for saturated fats, right? Saturated fats, then butter is a saturated fat, steric, palmitic, oleic. All these are, mind you, they are not in the resource book. They are just given to the fulfillment of the answer. These all are fatty acids. You just keep in mind that's enough. You don't need to understand whether saturated fatty acid or unsaturated. Actually, palmitic and steric are saturated fatty acids. Then oleic is a unsaturated fatty acid. I just uh, will inform there. These are saturated, right? Then they are saturated, then oleic is the unsaturated, then unsaturated fatty acid, right? So no need to just keep them. I just write them as they are given in the answer. Vegetable oil, once again, it's a unsaturated fat. That's why it's a fat. It's okay, but it's a unsaturated fat. It's an unsaturated fat. That's the reason. So the only answer is A. So A, only answer is A. Then answer is 5, right? Then 142. Photosynthetic plants or plants out of the following. Photosynthetic plant or plants out of the following. Normally, now you know Nepenthes, then Loranthus and Utricularia. These are what? These are that these plants are okay. Sorry, sorry, not that. I think not Loranthus, Nepenthes, Drosera and Utricularia, these are actually carnivorous plants. Actually, then carnivorous plants, right? Carnivorous plants, they photosynthetic. They are photosynthetic, right? So that's why, they are photosynthetic, right? They are photosynthetic. That's why we can take A, then B and D as answers, right? Okay. Then what about Loranthus and Cuscuta? Loranthus is semi-parasitic, okay? Semi-parasitic. Then when we come to Cuscuta, it is total parasitic, right? Then Cuscuta is total parasitic, okay? Cuscuta is total parasitic. Then Loranthus is semi-parasitic. Then Cuscuta is total parasitic. So then what is the point there? So total parasitic means they don't have the ability of photosynthetic. Ne? So no photosynthesis is here. So when it is semi-parasitic means, right? So absorb water and minerals from the host, right? So its aerial part is photosynthetic, no? Right? So semi-parasitic means it is photosynthetic, that take the water and minerals from the host plant, right? But do the photosynthesis, right? So they are photosynthetic. Then that's why Loranthus is also taken as the answer. Cuscuta cannot. It's a total parasitic. Then the answers are A, B, C, D. Answer number 5, right? Did you got that point, Lamai? It's a very, very important point. Just drop a message. Really very, very important point, interesting point. Normally, those are hidden points. Most of the time, these are hidden points, right? Mostly hidden points, right? Okay. Did you understand that? Okay. Nice. Then we'll move on to 143. Mineral elements that contribute to nerve function. Calcium okay, potassium okay, then sodium sorry, හිටපු ගමන් එක නටනවා පීඩි. Calcium, potassium, sodium, then chlorine, right? Okay. So phosphorus is not included in nerve function. So answer is A, B, D, E, right? Answer number 5, right? Answer number 5. Right. Then we'll move on to 144. Factors that may cause hypertension. factors that may cause hypertension, right? Right. So stress cause hypertension, prolonged hunger cause hypertension, right? Actually, it's hypertension too. It means it lead to low blood pressure, leading to hypertension. Obesity, hypertension, diabetes, hypertension, malnutrition once again result in hypotension. It means you can't say that prolonged hunger means staying hunger for one single day will lead into hypertension, but by doing it regularly, it can lead into hypertension, right? Okay. So A, C, and D answers A, C, D. It means answer number 2, right? Okay. So that the question asks that may cause. So they are not definitely causing hypertension, right? So that these are actually this A, then C, and this D. Actually, the best word to give them as risk factors for hypertension. Risk factors for hypertension. This is the exact wording. Risk factors for hypertension. This is the exact wording, yeah? Right? Exact wording, because you can't say that stress lead to hypertension. It may lead to increase in temporal pressure, but if someone is not stressing always, then you can't say that guy is having hypertension, right? Okay, got it. So that's the reason. Okay, that's the reason. So therefore, understand it, right? Then right, then understand. Okay, right. Then we'll see, right? Okay. Then 145. Select the correct hormone function combination, right? Okay. TSH, stimulated protein synthesis. The protein synthesis stimulation is not done by TSH, it is done by GH, right? Okay. It is done by GH, therefore that this is incorrect. Then stimulate secretion of adrenal medullary hormone, not adrenal medullary hormones. It has to be adrenal cortical hormones, right? So ACTH stimulate the secretion of adrenal cortical hormones, right? Adrenal cortical or adrenocorticotrophic hormones, right? Adrenal cortical means adrenocorticotrophic hormones, right? Okay. Adrenocortico trophic hormones, not medullary hormones, right? Okay. Then C, GH regulates metabolism, that is correct. So stimulate the secretion of testosterone, it is done by LH, not by FSH, right? Okay, then this is incorrect also. Spermatogenesis is stimulated by FSH, not by LH. So then the only answer is C. So answer number 5. Right. Okay, then we'll move on to 146. Symptoms of AIDS: appetite, yeah. Weight loss, that is also correct. Irritable itching around genital areas, that is herpes, right? Genital herpes. So that is incorrect. Then rashes on the skin, it is due to syphilis. Then infertility, that is due to gonorrhea. So answers I and B, then answer is 3. Very important parts, very important points, right? Okay. 147. Techniques related to recombined DNA technology. Appetite. Is given as appetite, no? You are asking of 146 or 145? I think you're asking of which one? 46 or 45? Yeah, yeah, it is yeah, that's it's appetite loss, so it's not mentioned, no? Right? Loss of appetite. Yeah, appetite. The increase or loss is not mentioned, no? Ne? Then you can't take actually the A, no? Appetite, appetite. Yeah, loss of appetite and loss of weight, no? Mm, yes. Yeah. Yes, I just went through there, no? Right? Didn't notice that actually. Just go through and just appetite loss. Thank you very much. So yeah, for appetite, the only appetite you can't say whether it is increased appetite or decreased appetite. You have to know. So what is happening in symptoms of weight is the loss of appetite, no? So in AIDS, what is happen? Loss of appetite. Yeah, that has to be there. Otherwise, you can't say it is correct or wrong, no? There are no clues, no? So loss of appetite, no? This is incorrect. So then answer is yeah, answer is then 5, no? Answer is 5. Only B. Right. Pilot papers are the papers given by us, right? Not the Mora pilot papers. We are not doing that kind of silly things, right? Just giving you the Mora papers and okay. So pilot papers are our pilot papers. All the MCQ, each and every MCQ is made by me. So not giving any sort of Mora or any Western province. So any such province. If I do provincial papers, I clearly mention provincial papers. That's it, right? So therefore, these entirely consist of our questions, right? The questions which are made by myself and which were given in the class. Okay. Then 147. Can yeah, someone can just reply that no issue, no need to even reply though. Someone has posted in the discussion asking whether provincial people so kind of anything. Right. Then 147. Isolation of DNA from other sources, technique related to recombinant DNA technology. Yeah, that is correct. No? Identification of the required nucleotide sequence using probes. That is also a technique, right? Separation of DNA groups using gel electrophoresis. That is also a technique, right? Right. Okay. Then D. Restricted digestion of DNA to be isolated using uh, restriction enzymes, right? Restriction enzymes. Okay. Then the other one, okay. Connecting DNA strand from various sources using not DNA polymerase. So connection is done by using DNA ligase, right? DNA ligase, right? So therefore this is incorrect. So then 147 A, B, C, D, no? A, B, C, D, answer 5. Right. 148. Invasive alien species actually a very important part I already mentioned and I stated, just mentioned in the previous paper also, just keep attention on invasive alien species, right? So invasive alien species, Lantana, yeah, and invasive alien species, Guinea okay, yeah, water hyacinth, giant African land snail. So then tilapia, no, not the tilapia, right? Okay. So A, B, C, and D. Answer number 5. No? Right. 149. An instance where metabolic process of microorganisms are used as end products, right? So metabolic processes of microorganisms, that is the keyword. Metabolic processes of microorganisms used as what? As end products, right? Okay. Uh, Metabolic process of microorganisms used as end products, right? So that is the that is the important thing. So instance where metabolic processes of microorganisms are used as end products. So the answer should be metabolic process, no, right? Then there are instances where there are several instances. Okay, one is microbial cells are used, and now as the end product is there, is the main thing. So end product is there. So then what are used as the end product? One is microbial cells. Microbial cells are used. Then once again, other thing is microbial metabolic products. It's one thing, right? Products, like not process. Other thing is microbial metabolic processes, right? Then genetically modified organisms used as end products, right? Okay, it means genetically modified microorganisms, right? Okay, so these are the pathways where end products can be used, right? So this is one, and this is another one. Okay, likewise. So the end product will be from these one. So metabolic process of microorganisms is here, right? So what are the answers? Then A, bioremediation, yeah, it is a process. Okay, metal extraction is a process. Then production of vaccines. Vaccines are produced by genetically modified organisms, right? So it is from the this one, right? This one, right? Vaccines from this one, right? So vaccines comes here. Vaccines comes here, right? Okay. Then retting is a process, that is okay. Then alcohol production. Alcohol is a what? It's a production, no? So it is a primary end product, no? Right? So primary end product. So there is a product. So microbial metabolic product. So alcohol comes here. Alcohol comes under products, not process. Products, right? Okay, so therefore that is incorrect. Then answer is A, B, D, and answer number 1. Got it? So that is what the most important aspect of this question. So keep in touch. So microbial metabolic products like alcoholic beverages, then this is primary. Alcohols are primary products. Then there are other one, that is the product. As a product you can use antibiotics, no, right? So when we discuss about antibiotics, they are secondary. They are secondary, right? Antibiotics are secondary metabolic products, right? Okay. Then when regarding microbial cells, then I will take it there. Single cell proteins are the example, right? Single cell proteins are examples for cells. Then then regarding microbial metabolic processes, now we have bioremediation, the metal extraction, retin, those three are there. Okay. Then genetically modified organisms, there you can have enzyme production, hormone production, and vaccine production. So GMO vaccine is one. Another one is right, enzyme. Another one is what, hormone, no? Those are the three methods: hormones, enzymes, hormones, and vaccines, right? Okay, got it. So put a message that those who have got the 149, and this is a kind of a entirely short note, right? A very important short note, very, very important, right? Okay. Okay, students, very important. So you have to understand the importance behind this, right? Really important. Okay. Then we'll move on to the 150 now, na? Right? 150 now, 149 done, na? Right. Okay. 150. Disease or diseases that can commonly be seen in freshwater fish that is or are caused by, right? Caused by unicellular external obligate parasite. Unicellular external obligatory parasites, right? Okay. External obligatory parasites. Now, okay, right? Okay. So, right. Then you need to be understand. Now we'll see each disease, right? So each disease is because of unicellular external obligatory parasite. That is correct. Then trichotinosis is because of unicellular. Okay, all right. Then external, all right. But the reason is not. It is not the obligate. That is opportunistic. Opportunistic. Opportunistic, right? Then hemorrhagic septicemia. Hemorrhagic septicemia is because of bacteria, right? Bacteria, therefore B incorrect, C incorrect, right? D. Columnary disease once again because of bacteria, right? Then gill infestation, yeah, it is obligatory, can be, but it's not unicellular, no? Or external. It is just obligatory, opportunistic, gill flux and skin flux, right? So it is only obligatory, can be either obligatory or opportunistic. No mention regarding that external log, maybe unicellular kind of that obligatory, opportunistic, then but gill flux and right, then skin flux. These are the right kind of uh, that parasites, right? Okay, so the only answer for this is A, each disease, no? 150 A answer number 5, right? Okay, nice. Now 150 done. 150 out of 500, right? Then of there are 5 3, then but 10, no? Then around by 100, around 30, no? Not actually 30, right? But around now 300, yeah, around 30%, no? Yes, 30% is done, right? So that is what up to now. Okay. Right. Around 30%, no? Not yet, but finished, but 30%. Now we'll come to 151. Okay, Lamai. Now give me a ready message quickly. Give me a ready message. Now 150 done. Now we have to move from 151. I need a ready message. Right. ඉක්ම ready කියලා message එකක් දාන්න. ටී චුට්ටක් බීලා එමු මේ 200ක් කරලා. හරි රයිට්. ටී චුට්ටක් බීලා එමු. හරි බොන අය බොන්නේ 100 එකක් ටී එකක් බොනවා. ටී බොන්නවත් ටයිම් එකක් නැහැ නේ. මට පොඩ ෆොටෝ ටිකක් දාගෙන කරපු කට්ටිය. ෆොටෝ ටිකක් දාගෙන මේකේ රිවිව් එක විවරණයම හදන කට්ටිය. ෆොටෝ ටිකක් දාගෙන යන්න හැමෝම පුළුවන් නම්. right. So send me some photos, right? Some photos which means that just you are doing the work to clarify, to just I don't know whether I'm just lonely describing things, no, whether you have not doing it or not, right? Right, okay. Then we'll move on with 151. We'll go quickly somewhat, right? Okay. Okay, no issue. Mouse. Then question number 151. A criteria which can be used to observe diversity among organisms, no? Diversity among organisms. Actually, what are the criterias there? Size, then shape, then form, then habitat, right? These are the four. So not order, not metabolism, not form, right? Not growth, not movement. So answer is third one, right? Am I excellent? Okay. Right. Then 152nd, the element present in the highest percentage within the human body out of the following is. Now you have to write the order correctly now from starting from the highest, right? Oxygen is the highest, then carbon, then hydrogen, then nitrogen, right? This is up to OCHN. Then calcium, then phosphorus, then potassium, then sulfur, then sodium, right? Then chlorine, then least one is magnesium, right? So human body out of the following, which has the highest, now no OCHN here, then it is, then the highest one is the calcium. Answer number 3. Okay, very simple. Now keep the order in your mind, right? This is not given in the resource book, but mind you, grade 10, right? Grade 10, that textbook include these percentages. Maybe they might check them, right? It's grade 10 knowledge, right? Even grade 10 student can write it, no, in that aspect. They might given it like that, okay, right? So that is the thing. Okay, right, nice. Then that is the order, right? So keep in mind that order needs to be not no need of percentages, but roughly with the ascending or descending order, right? Then 153, correct statement regarding water, which is a compound that is important for life on Earth, right? Nice, great, life on Earth. Most properties of water are based on the physical nature of water molecule. No, most are based on the what nature? Most are based on the chemical nature. No, they keep in mind chemical nature means that hydrogen bonds, right? Okay, then hydrogen bonds along with the angle, right? Those are all chemical bonds, right? Okay, the chemical features, right? Not physical nature. That angle, then hydrogen bonds, like all those things contribute in the chemical nature, no? Right? Okay, then polarity, right? Then polarity, all those things combined, right? Okay, these are the most important things to just maintain the properties. Physical nature is very important, but apart from, when comparing both, that chemical nature is the most important, right? Water provides a biological medium for some organisms, right? Yeah, water provides a biological medium for not some, for all the organisms, right? So therefore, this is incorrect. Then we come to third one. Formation of hydrogen bonds, breaking and reforming occur slowly. No, they are not occur slowly. They are taking place with great or higher, right? Higher frequency, right? They are taking place with higher frequency. Then hydrogen bonds of water at its liquid stage are very fragile. That is correct, right? Liquid stage are very fragile. The fragility is when compared with ice that in solid, their fragility is very low. It means very low. It's very strong. That ice is very strong. But when it comes to water, liquid water, that fragility is increasing. Are very fragile. Okay. Polarity is not the even charge distribution. It is the uneven charge distribution within a molecule. That now if we take a water molecule H2O, now this is delta negative, delta negative, delta positive, delta positive, delta positive. Now look at uneven charge. Here positive, here negative, here once again negative, here positive. This is the uneven charge distribution, right? So that is these charges lead into the what? Polarity of that molecule. Okay, right. Then we'll move on to 154, right? Correct statement regarding phospholipids consists of now phospholipid is made up of glycerol molecule, two fatty acids and phosphate group. No, therefore three fatty acids. That is incorrect. So phospholipid, if we draw the molecule like that, CH2OH. No need to just draw that. I think it's a waste of time. I just roughly draw that with the arrangement. There's the head part, then there is the tail, then there is the what? Phosphate group, no? Right? This is the arrangement, no? So here, once again went, right? Okay. Okay, now when it's come to phospholipid, right? It's the head, it's the tail. Now there are two fatty acids and there is this is 1 into glycerol, this is glycerol, and they are to find the 1 into phosphate. It means 1 into means 1, right? Okay, then right, okay, right, right, consists of a glycerol molecule and not three fatty acids. It is 1 glycerol plus 2 fatty acid plus 1 phosphate. This is the composition, right? An additional non-polar, not non-polar, right, molecule or a small charged molecule is attached to the phosphate group. That charged molecule attached is that what is that molecule attached? That is the choline. Choline is positively charged, right? So it is polar. It is polar and positively charged and hydrophilic. We have discussed that in the first, I think that in the early MCQs, right? Two ends of the phospholipid molecule show similar behavior to each other. No, that head is hydrophilic and polar. Hydrophilic and polar. What is that? That is the head, right? Head. Then the tail is hydrophobic and non-polar. Okay. They are not showing similar behaviors. It's a nitrogen containing lipid. Yeah, that is a very vital point. That inside in choline containing nitrogen. So therefore, choline is a must to maintain the neutral nature of the molecule. Therefore, phospholipid itself contains nitrogen, right? It has a positive charge. No, it is a not positive. It is neutral. No, neutral, right? Neutral charge. So answer is number 4. Fifth one, the most common glycoprotein in the extracellular matrix of a animal cell is what is the collagen. Correct statement regarding enzymes, right? All enzymes are globular, not all. Many enzymes are globular proteins, therefore not all enzymes. Most enzymes catalyzed reactions are reversible. That is correct, right? Most enzyme catalyzed reactions are reversible, not all. That is correct. All enzymes are substrate specific, right? Not all, most are substrate specific, no? Most are substrate specific, most, no? Enzymes are mostly substrate specific because there are enzymes like Rubisco that catalyzes two types of reaction, no? Now, Rubisco, if we come for Rubisco, it catalyzes that oxygenase reaction as well as carboxylase reaction. So therefore, there is kind of a deviation there, right? Okay. All enzymes are heat liable, not all. Many enzymes are heat liable, right? They take part in the reaction when the reaction takes place. They doesn't take place. They don't take part in the reaction. They are not taking part. They are not taking part in the reaction, right? In your resource book, just check whether for the substrate specificity, what is the statement given? Many enzymes are substrate specific, or likewise, what is the statement given there? Many or most, is there in such kind of a thing, or just given that enzymes are substrate specific? I think it's given as that only this that enzymes are substrate specific. I think it's given like that. That simply that enzymes are substrate specific without many or most or like that phrase, only substrate specific. No, if possible someone can send a photo of that to the discussion group. Okay. Right, then others will also be informed, right? That will be important, right? The reason is that Rubisco actually, no, we can't say that all enzymes are substrate specific. Rubisco there, is catalyzed two reactions. One is oxygenase reaction, and other one is carboxylase reaction that reacts with two substrates. No. So therefore the substrate specificity is not there, right? So it binds with O2 and CO2, two instances. Okay. Yeah, enzymes are highly specific to the substrate. That is what given, no? So not given as all kind of a thing, though that's the reason. But keep in mind that Rubisco is somewhat deviating from the scenario, right? Okay. But most of the time we generally say, if it is given as enzymes are highly specific to substrate, then substrate specific, then that is correct. But I think they won't give all kind of phrases, all or many more. But keep in mind that one, right? Okay. So the most correct one here is the second one. Okay, right? Right. Keep in mind, keep it in mind, right? Okay. Okay, right. Don't say all. If it is all, that is incorrect. Huh? Totally incorrect. If it is all, totally incorrect. Okay. Right. Right. පොඩි අවුලක් තියෙනවානේ. බලමු. Right. 157. 157. Right. Now we'll go to the 157. Right. Okay. Right. Which of the following is the correct statement regarding the Kelvin cycle in C3 plants, right? 157. Addition of RUBP to carbon dioxide is known as carboxylation. Addition of RUBP to carbon dioxide is known as carboxylation, right? What is regarding the Kelvin cycle in C3 plants, no? Right. What is that? Addition of RUBP to carbon dioxide? No, that is addition that carbon RUB that carbon dioxide is captured, or that is fixed by RUBP is known as a carboxylation. Addition of RUBP to carbon dioxide, that there is some that idea is not that much clear. No? Addition of carbon dioxide to RUBP is the correct one, I think. No? Addition of RUBP to carbon dioxide, no, not that addition of CO2 to RUBP. Likewise, the Kelvin cycle should take place twice, not twice, right? It should take place thrice, no? It should take place thrice, no? Three times, right? Three times. So therefore, this is incorrect. G3P, right? G3P is the stable product formed during only stable products formed during the photosynthesis. You can't say so. There are several stable products, right? It's not the only stable product, right? It is not the only stable product. The precursor of carbohydrate synthesis is not 1,3-bisphosphoglycerate. It is the glyceraldehyde 3-phosphate. It is the G3P, right? So the precursor of carbohydrate synthesis is G3P, G3P, right? G3P. Okay. Then okay. Right. So here, then come to fifth one. Reduction of carbon dioxide take place using the energy produced by ATP and NADPH produced in the light reaction. So yeah, that's reduction of CO2, right, take place using the energy produced by ATP and NADPH produced in the light reaction. Now, in the light reaction, ATP and NADPH are produced, right? Okay, ATP and NADPH are produced in the light reaction. They are produced in the light reaction. Okay, so what happened now? ATP and NADPH are produced during light reaction. So that energy is used to, now what is the reaction that taken place? Now if we take carbon dioxide and water, they are collected into form what glucose and what then glucose and oxygen. So here carbon dioxide with +4, carbon with +4, then here carbon is 0, then +4 is into 0 means -4, right? So reduction of carbon dioxide is taking place, no issue. That is taking place, that is correct. Then energy produced in the ATP and NADPH are used, no? So that statement is correct. So therefore, fifth one is the correct statement, 157. Right. Then 158. Right. Question number 158. Correct statement regarding the electron transport chain, no? Right. Consists of proteins and non-proteinous molecules that involve in the transport of protons and electrons through cristae. Consist of proteins and non-proteinous molecules that involve in the transport of protons and electrons through cristae. That is correct. That electron transport chain is found in the that inner membrane of the mitochondria, right? So it contain now different types of things like now protein, non-protein things, no, right, molecules. So they involve in the transport of just proton and electrons, no. Now protons and then electrons move through. So protons and electrons and then electrons just sorry, that direction had to be changed, right? So here what happened now, movement of protons and electrons taking place here, right? Okay. Right, may, okay, right. So then consists of proteins and non-proteinous molecules that involve in the transport of protons and electrons through crystal. That is correct. That is known as the electron transport chain, no? That is known as the electron transport chain, right? Okay. Then second one, the final electron acceptor is H plus. The final electron acceptor is not H plus. Final electron acceptor is O2, right? It's O2, right? It's O2, right? Then occurs in the inner membrane of mitochondria, right? It is not occur in the inner membrane of mitochondria. It is occurs through the inner membrane of mitochondria, not occur in the, right? Not occur in the inner membrane of mitochondria. It is occur, right? Occur through the inner membrane, right? Occur through the inner membrane. Occur through the inner membrane, right? Occur through the inner membrane, right? Then fourth one, ATP is produced by substrate phosphorylation. ATP is produced in the electron transport chain via oxidative phosphorylation, right? Via oxidative phosphorylation. The number of ATP produced by a single glucose molecule in liver cell is not 30, right? It is 32. It is 32, right? It is 32. Got it? Right, it is 32. The number of ATP produced by a single glucose molecule in liver cells is 32, not 30, because liver cell is a active cell. So an active cell, right? Liver cell is an active cell. In active cell is 32, but if it is become 30 is for inactive cells, no? 30 is for inactive cells, right? That is why this is incorrect. So second one, third one, fourth one, fifth, all are incorrect. So you have to get, this is the most important. Occur in the inner membrane, no? It's occur through the inner membrane, or across the inner membrane. The across, that is the best word. Across, not through. Actually, through is okay, but across is the best word. It occur across the inner membrane of the mitochondria. Okay, right. Then question number 159. The correct step where the respiratory substrate enters the process of cellular respiration is given by, right? So correct step where the respiratory substrate enters the process of cellular respiration is given by. Now see, glycerol glycerol enters through where? Glycerol enters through G3P. Now we have discussed this in the Poduratava seminar, right? We have discussed it in the Poduratava seminar. So therefore not through pyruvate. Therefore this is incorrect. Then fatty acids enters through what? Acetyl-CoA, not glyceraldehyde 3-phosphate. With this one also we discussed in the Poduratava seminar, right? So this is incorrect. Then amino acids through oxidative phosphorylation, that is incorrect. None of the substrate goes through oxidative phosphorylation, no? Right? If we take simply now from glucose, right? If we write it, we'll complete the discussion and then write note down it, no? Then then sugars, they enters through glyceraldehyde 3-phosphate, not through pyruvate. Therefore this is incorrect. Amino acid through citric acid cycle, that is correct, right? That is correct. So 159, fifth one is the answer, right? Okay. Then what is this? Now see, glucose is converted into G3P, then it is converted into pyruvate. This is what happening, no? Then pyruvate is converted into acetyl-CoA, then acetyl-CoA goes through the what? Is goes through the TCA cycle. Then TCA cycle, it goes through the electron transport chain, right? Okay. Then what happened? Glucose and G3P, pyruvate, acetyl-CoA, TCA and electron transport chain. Then what happened to glucose and the substrate? Then here carbohydrate is there. Then carbohydrate, what happened? Carbohydrate is breaking down into what? They are breaking down into glucose. Okay. Then glucose can again enters as the glucose, same one. Then O is goes through G3P. This is the carbohydrate. Then we come to what? Lipids. Lipids are broken down as glycerol and fatty acids, right? These are broken down as glycerol and then as what? Fatty acids, right? Glycerol and fatty acids, right? Fatty acids. Then what happened? Glycerol enters through G3P, then fatty acids enters through acetyl-CoA. This is the pathway. Now, when we come to what? When we come to here with proteins, with proteins, what is happened? Proteins are converted into amino acids. Then in that process, what happened? Ammonia are evolved. Then in that process, amino acid from accepting ETC, now can't, just you have to keep in mind that none can enter through ETC. So therefore I will cut it from the latter, from the most lowest part to the upper part. TCA, acetyl-CoA, then pyruvate. These are the entry pathways for amino acid. That is the summary. So therefore that is why the fifth one is correct answer. Okay, right? Then 160, right? Correct occurrence or time period combination out of the following is, right? Then 160, when we come to 160, evolution of sponges, it is around not it is not 670, it is around 700 million years ago, right? 700 million years ago. Then origin of ancestor of arthropods, that is 670 million years ago. 670 million years ago. Then third one, the time period combination out of the following. So colonization of land by fungi, plants and animals about 500 million years ago. That is correct. That is correct, right? Then origin of first tetrapod, that was about 365 million years ago. 365 million years ago, so not 250 million years ago. Then divergence of human lineage from primates, that was taking place 6 to 7 million years ago. No? 6 to 7 million years ago. Then all these 1, 2, 4 and 5 are incorrect. Correct answer is the third one. Correct answer is the third one. Okay. Right. Now that's a very important part, right? Very important part regarding the time periods, right? Time periods of that regarding the evolution, right? Very important. Okay. Uh, Now 11th one, sorry, 161, 61st one. Consider the following incidents, right? So it's regarding the eons and eras. So that, yeah, regarding the eons and that eras, right? Okay. Then we'll see origin of Earth took place at Hadean, no? Right? Then origin of first seed plants, that was at Paleozoic, right? Then radiation of mammals were at Cenozoic. Then origin of mammals were Mesozoic. Then fossils of older cells, they were again Archean, no? Right? What includes the answer with this order? Hadean, Paleozoic, Cenozoic, Mesozoic and Archean. That's the second one, no? Right? 161, answer is second one, right? 162, 162, right? 162. Select the characteristics of domain eukarya. All of them are multicellular, not all, right? Not all. Majority are multicellular, right? Majority are multicellular. Then all of them are aerobic. That is also incorrect. Majority of them are aerobic, right? Then third one, some of them can fix atmospheric nitrogen, not some or many or just little bit. None of them can, right? Okay. So here none can fix nitrogen. None can fix nitrogen, right? None can fix nitrogen, right? Okay, that's the reason. So then the fourth one, most of them show sexual reproduction. That is correct, right? Most show sexual reproduction. That is correct. Then some of them are prokaryotic. Actually, not some, right? Not some. Actually, all are prokaryotic, right? Not some. All are prokaryotic. So answer is answer number 4. Right? Just understand this domain eukarya. So domain, for in domain there will be a definite questions from domains, right? That's why I always include those things, right? Right. Okay, we'll come to the 163. Now, first one, right? The endodermis consists of a lignified belt called Casparian strip. The all are correct, but the constitutive is incorrect. Now, Casparian strip, is not lignified, it is suberinized, right? It is suberinized, presence of suberin. No, suberin belt is there. There is suberin belt, not lignin belt, right? Okay. Then the pericycle of a monocot root has the ability of divide, that none of the monocot, either root or stem, has no ability of dividing, right? Again, so it means that none of the monocots show secondary division. It means none of the monocots show secondary growth. Either either it is then either it is either it is you see, either it is then regarding the monocots, either it is uh, root or stem, no, right? Root or stem, right? Then right, okay, right. Then so the pericycle of a monocot root has the ability to divide, right? So no ability, right? No ability to divide, right? Okay. Vascular zones of monocot stems are surrounded by a sheath of sclerenchyma. Vascular zones of monocot stems are surrounded by a sheath of sclerenchyma, right? So vascular zones of monocot stems are surrounded by a sheath of sclerenchyma. That is correct. Now we come to the point where, right, vascular zone is taking, then then xylem here, then phloem is here. Then you can see in surrounding that one, there is a that sclerenchyma layer, right? Sclerenchyma layer surrounding the that vascular zone, right? So that is correct. That is regarding the monocot stem, no? So then we come to the fourth one. Then that is third one is correct, right? The xylem can be seen as a star-shaped structure at the center of the cross-section of a dicot stem. So not dicot stem, in the dicot root actually, that in the center we can see that xylem as kind of a star where phloems are in between, right? This kind of arrangement that is in the root, not in the stem, right? So therefore that is incorrect. There is a pith made of parenchyma cells in monocot stems at the middle of vascular tissue. Monocot stem, no. So therefore actually there is a pith made of parenchyma cell at the middle of vascular tissues, right? So that pith means that the space. Now if we think the vascular, sorry, right, if we take vascular bundles like this, if we take vascular bundles like this, right, this kind of an arrangement is there, no? This kind of an arrangement. So if we take these kinds, then in the middle means this portion, right? In this portion there is pith actually, right? This is pith and it is made of parenchyma, right? Parenchyma cells, that is correct, right? So therefore so that is correct regarding what? This is for dicots, right? Not for monocot. That's the point. So there is a in monocot stem that is incorrect. That is incorrect, right? Therefore fifth one is also incorrect. That's the reason, right? Fifth one also incorrect. Right. Then we'll move on to the 164. Correct statement regarding stomata. Correct statement regarding stomata, we'll see. Bean seed shaped in all plants, that is incorrect actually. In all plants, so we can't say that in all plants that is bean shaped, right? So bean shaped in actually what? That there's a difference in monocot. It is a kind of dumbbell shape, right? Dumbbell shape in monocot. In dicots, it is bean shaped, right? It is bean shaped in dicots, right? Uh, then guard cells are the only epidermal cells that possesses chloroplast, right? So guard cells are the only epidermal cells that possesses chloroplast. That is correct actually. So they are the only that epidermal cells with chloroplast, right? They have chloroplast in order for their light reaction to take place. Light reaction take place means then what happen when the light reaction is taking place, that produce energy, is used for the that K plus movement. It means for the opening and closing of the stomata, no, right? The gas exchange in plants occur only through stomata. That is incorrect. There can be lenticles, right? Then there can be kind of what? There can be cuticles, no, through cuticles. So these can be taken place. So therefore, not only the stomata. When guard cells are turgid, stomata close and they open when turgid is lost. That is incorrect. When guard cells are turgid, stomata is open, right? So when regarding the stomata opening, when the turgidity, right, when the turgidity increase, what happens? Stomata opens, right? Okay. When turgidity decrease, what happen? Stomata close, right? So that's what happened, right? So that's what happened. Then that is what happening, right? So this is very important, right? Very important point. Okay. Then right, then that is what's happening. So when guard cells are turgid, stomata close and they open when turgidity is lost. Okay, that is what happened. Okay, so that is what happening. So that is what happening. So then what is happening? Okay, then here it says therefore this statement is incorrect, no, right? The stomata close due to the loss of carbon dioxide concentration in the substomatal cavity. Stomata close due to the loss of carbon dioxide concentration in substomatal cavity. Now, earlier also we discussed. Now, they are the epithelium. The epidermis is there. They are the the stomata and the guard cells. Then they are the that adjacent adjacent epidermal cells. Then there is the space known as substomatal cavity. Then when the carbon dioxide here decreases, what happens? Carbon dioxide need to be taken inside by opening the stomata. So when the loss of carbon dioxide concentration, or the when the carbon dioxide in substomatal cavity decrease, what it tends to stomata opening, right? Stomata opens, right? Then therefore this is incorrect. Okay, this is incorrect. That's the reason, right? So 164 answer is number 2, right? Okay, these are the important points. Right. Okay. Okay. Right, 164. Then we are moving to 165, right? Right. Okay. Uh, right. Then coming to 165. Right. Okay. Then come to 165. Now there's a calculation, right? The water potentials of cells A and B above are respectively. There's a kind of a calculation they are going on, right? We'll see. Now we have to find the water potential of A first. Now psi A equals now here psi means first you have to write psi means psi P plus psi S, no, right? This is the equation. Psi A, it means psi of the A solution is equal, then plus 3 plus it means plus means plus negative 4.6, no, right? Okay, right. Then what happened? It is equals negative 1.6, no, right? Negative 1.6 megapascal, no, right? Okay. Negative 1.6 megapascal is the water potential of A. Then psi B equal plus 1.2, then plus negative 2. It means what is the water potential? 1 minus 2 means minus 0.8 megapascal. So this is the psi B. Then what is the answer? Right, answer number 5 for the 165. Right, okay. Then we'll move on to the 165. Now here it is 166, right? Okay, 165. That's the way to find the water potential, right? The equilibrium water potential. That's the way to find the equilibrium water potential of a given cell. It's a single cell. Now we'll go back to the other question. 166. The water potential after the cells A and B are joined together and allowed to reach equilibrium. So when we need to find a psi equilibrium of two cells, we need to psi cell 1 plus psi cell 2, then it is divided by 2, right? Okay. This is the psi equilibrium equation. Then psi equilibrium equal, right? Psi cell 1 was minus 1.6 plus then minus 0.8 divided by 2, and it is equal minus 2.4 divided by 2. It means minus 1.2 megapascal, right? So answer is 1. Got it? Right. Then 167. 167. Okay. 167. Okay. Then when come to 167, the pressure potentials of cell A and B when they are immersed in pure water and allowed to reach equilibrium are respective. So this is the right. This is the way to find the equilibrium. Now these are okay. Now the question is shifted. Now that the next part is somewhat not difficult. You have to apply a kind of a theory. The pressure potentials of cell A and B when they are immersed in pure water. When they are immersed in pure water means now you need to find now at the state now at this stage A cell and at this stage B cell. This is minus, sorry, right? Right. This is minus 1.2. This is minus 1.2, right? Correct. A and B. That's the case, right? So in A and B minus 1.2 and minus 1.2. So that is the water potential now. Now they are put in what? They are put into what? Immersed in pure water. No? So what is the logic? When cell is, right? When a cell is, right, put into a solution, right? Put into a solution, right? What happened with the normally the theory when a cell, now cell is here, when cell is put into a solution, now cell is inside the solution, what happened? Cell takes the psi of the solution. That is the point. No matter how much is the psi of the solution, it takes it, because the reason is now the volume of cell is actually can't compare with the volume of the solution. That's it, right? That's the reason, right? Okay. So that is what happening. Okay, that's what happening. Now then, what happened now after insert into the pure water, the water potential of A and B will be what? It will be 0. Here will be 0. Nice, 0, no? Final will be 0, right? Okay, final will be 0. But and another point, during these scenarios, keep in mind that psi s is not changing. Heck of points, right? Psi is not changing. You have to keep in mind these things. What is the first one? When a cell is put into immersed into a solution, it takes the psi of the solution, right? So that's why this is taken 0 megapascal, and this one also has took 0 megapascal, A and B, because they are immersed in pure water, right? Then what happened? We solution is higher than that of greater than that of V cell. Then what happened? That psi s is not changing. We are taking it as a this kind of a clue. No, psi s is not changing. So therefore, when we find the new psi a, we need to psi a. We know equal psi p. We don't know psi p dash, and plus then earlier psi s of the a. What is the psi s of the a? A is minus 4.6, then here minus 2, no? Minus 4.6, that's it, right? Then psi b equals psi p double dash plus minus 2. That's the equation, no? This is the first one, and this is the second one. Now for the first one, we get psi a means 0 equals psi p dash minus 4.6. Then we can get psi p dash as plus 4.6 megapascal, right? Okay. Then when come to second one, 0 equals psi p double dash minus 2. Then here we come, right? Psi p double dash equals positive of that psi s. Then what is the answer? Positive values of that. Answer number 2, no? Answer number 2. Right? It's a very good way of solving this question. So just make this correct. So if there is any mistakes, you can correct it, right? Right. Right. Okay. Then 168, right? Okay. Right. 168. Regarding the gametophytes of kingdom Plantae, right? We'll come to the 168. Now fine with the water potential. I think it's okay, no? Right. The gametophyte of Pogonatum is photosynthetic, but not completely independent, right? Okay. Right. It is actually it is independent, right? It is independent, right? It is independent. So gametophyte of Pogonatum is photosynthetic, right? Photosynthetic, but not completely independent, right? It says, but not completely independent. Actual thing is, it is independent, not completely independent, right? It's given that the gametophyte of organoid is photosynthetic, but not completely independent, right? So what is that? That is independent, not depending on, right? It is independent. So not completely independent. That is the incorrect. It is independent, right? That's the thing. It is independent, right? Independent. That is independent, right? Okay. That is independent, right? Okay. That is independent, right? So gametophyte of Pogonatum is photosynthetic, but not completely independent. It is actually independent, nah? That's why, because it is the dominant plant, no? Right. The gametophyte of Neprolepis is a monoecious bisexual plant. Now, when come to the gametophyte of Neprolepis, it is that what you know, that heart-shaped thallus, no? Right. So it have both archegonia and antheridium in the same plant. So therefore, it is bisexual. That is correct, right? Therefore, it is bisexual, and it is monoecious, means having in the that kind of the same plant, no? Right? Same plant. Then what is that now? The gametophyte of Neprolepis is monoecious, right? And bisexual, right? Then monoecious means both the sexes are found in the same plant, no? Right? It is monoecious, no? Monoecious. That is correct. This is Neprolepis, no? Right? This is Neprolepis. Okay. Right. Okay. Right. Then we'll come to the this one. The gametophyte of Neprolepis is monoecious bisexual plant. That is correct, no? Right? Monoecious and bisexual plant. There is no any point regarding is it that microscopic or macroscopic, right? Or anything, right? The male and female gametophyte of Selaginella are photosynthetic, right? The male and female gametophytes of Selaginella, right? They are photosynthetic. Okay. Then what is saying? But depend partially on stored food in sporophyte, right? Okay. So what is the point that here that female or that female gametophyte is photosynthetic? That is correct. Okay. But male gametophyte is not photosynthetic. That is the point. So the male gametophyte is not photosynthetic. Male gametophyte is non-photosynthetic. That is the point, right? So keep in mind that is non-photosynthetic. Okay. Female gametophyte, the male gametophyte is non-photosynthetic. Okay. Then this is what is that? It is independent. Then it is also incorrect. This is correct. This is incorrect. Then fourth one, both male and female gametophyte of cycas obtain nutrients from the nucleus, right? So both male and female gametophyte of is obtained from the nucleus. That is also correct. Then fourth one is also correct. Okay. The gametophyte of Anthophyta is made up of 8 cells. The gametophyte of Anthophyta is made up of 7 cells and 8 nucleus. That is the point, right? It is made up of 7 cells and 8 nuclei, right? 7 cells and 8 nuclei. That is incorrect. Then there are two answers, 2 and 4 for the 168. Okay, good, that it's a good MCQ regarding the life cycle, kind of an advanced one, right? Advanced one, kind of an advanced one, right? Okay, advanced one, right? So be careful of that, right? Hari. Right, then we'll move on to the 169. Uh, which of the following plant growth hormones inhibit early seed germination by promoting seed dormancy in plants? Which of the following, right? Which of the following plant growth hormone inhibit early seed germination? Which inhibit early seed germination by promoting the seed dormancy in plants? So this is actually in order for what that drought condition response, kind of a response for drought condition, right? Response for drought condition, right? That is the major thing, right? So inhibit the early seed germination, then promote the seed dormancy. They are promoting the seed dormancy, right? Okay. So that is the abscisic acid. So keep in mind that abscisic acid plays a major role in those controlling those drought conditions and such things, right? Okay. Okay, then it's done. Okay, 170. These are the molecule, the element composition. Definition of the which of the following elements would cause the above symptoms respectively, right? Okay. Deficiency of which of the following elements would cause the above symptoms respectively. Now we'll move on to the question, right? Okay, thin stems is due to actually phosphorus, right? Crinkling of leaf is due to zinc. Then light green color in young leaves is due to copper. So therefore, answer is number 3. No, answer is number 3, right? Answer is number 3, right? Okay, answer is number 3, right? Right. Then when come to 171, an epithelial tissue shows the following characteristics, right? Okay, 171 we'll see. Now we have to move towards what is the type of the epithelium here mentioned. Now single layer of cells. Okay, then it is we can definitely cut down stratified squamous. Presence of different nuclei at different level. There is only one tissue, that is the pseudo stratified column epithelium. Then answer is number 2. That's enough. No? That's enough. No need to go further. That's enough, right? So cilia can be seen under some instances. Cilia also seen. Then when the cilia present, we name them as pseudo stratified ciliated columnar epithelium. Pseudo stratified ciliated columnar epithelium, right? Okay. Then 172. Correct statement regarding the animal nutrition, right? Animal nutrition. 172. Now third one. Barnacles attached to whales are parasites, right? Okay. So then barnacles attached to whales are not parasite. It's kind of a relationship which includes commensalism, right? Commensalism kind of relationship, because there is no disadvantage for whales, but barnacles getting advantage, right? Okay. Or no any harm for the whales are even, right? So males, that is kind of a neutral relationship, but for barnacles that is a positive or advantageous one. Aphids sucks nectar from flowers, aphids sucks nectar from phloem sap, right? Not from flowers, they suck nectar from phloem sap, right? Phloem sap, right? Oh, therefore, that is incorrect. First one incorrect, second one incorrect. In substrate feeding, the animals eat through the plant leaf or animal carcass, right? In substrate feeding, third one, in substrate feeding, the animal eat through the plant leaf or animal carcass, right? So that is third one, that is correct actually, though this is called as the substrate feeding, that the organism is staying on the substrate and eating through it, right? Eating through it. Then fourth one, fluid feeders use tentacles, claws, and poisonous fangs, right? So what is they are mentioning now? Tentacles, claws, poisonous fangs are used by bulk feeders, not by fluid feeders. They don't need those kind of structures, right? They just feed on fluids, right? Therefore, this is incorrect. Fourth one. Cellulose digesting microbes in termites show symbiosis, right? So cellulose digesting microbes in termites show the mutualism because both are getting benefits from the relationship. So that is mutualism, not the symbiosis, right? Okay, that is the answer. Then 172nd, it is third one. Okay. Now take a 5-minute interval and come back at 5:25, right? We'll start from the 173, come back at 125, right? Sorry, 125, no, 5:25. Okay, right. We'll move from 173, no? Right. Can you hear me? I think okay, no? Right. 173. Correct statement regarding the absorption in the small intestine. Now, absorption in small intestine, the previous paper also, we discussed about, that absorption taking place in the small intestine, right? First one, there are two structural modifications in the intestine wall to increase the surface area for effective absorption. So, the point is two structural modification. This is the place that the mistake or that the false part is there. So there are not two structural modifications, there are three structural modifications actually, right? Three structural modifications there, right? So what are the three structural modifications? Permanent circular foldings is one. Permanent circular foldings is one. Then the other one is villi. The last one is microvilli. These all increase the what surface area. Okay, increase the surface area, right? Okay, right. Then right, that is what happened. Okay, then second one. Okay, second one, a brushboard appearance is visible due to villi exposed into the intestinal cavity. So brushboard appearance is because of what? Microvilli. Now look at now. This is the intestinal lining. Now permanent circular foldings are like this, right? These are like this, right? Okay. Then what is happening here? Now permanent circular foldings are there, and these permanent circular foldings, if we take one like this, they have kind of projections like this. These are the villi. These are the villi, right? So these are villi. Then at the end of the villi, if we take a villi here like this, if we take a villi, in a villi, there we can see these kind of projections in a villi. These are called the microvilli. So microvilli appearance is like this, no? So this is kind of a what? Brush-like appearance, no? Right? Uh, right? Brush-like appearance, no? Right? Okay. So there is brush-like appearance, that is because of microvilli, right? Okay. This is incorrect and this is incorrect. A total amount of 9 liters of water is added into the small intestine. That is correct. From the water intake and with the digestion process, a total of 9 liter water is added, right? That is very critical point. Keep in understand, 9 liter of water is added into the small intestine, right? 9 liter. All glucose molecules actively transport into epithelial cells, right? Not all glucose molecules, right? That many of the glucose molecules are actively transported into the epithelial cells. Many, right? Not most, right? Not all, actually, many. Fatty acids and monoglycerides enter the cell through pyloric splinter. Fatty acid and monoglycerides enter the cell through pyloric splinter, not through pyloric splinter. They are entered through pyloric splinter is where that the substance in the stomach is entered into the small intestine. This is mentioning about the absorption. No? So they are entered through the microvilli, not the pyloric splinter. Pyloric splinter is where that stomach stomach continue with now here it find the pyloric splinter, then here it find the cardiac splinter. These are the connection. Now stomach connect with the esophagus, there is cardiac splinter. Stomach connects with the small intestine, there is pyloric splinter, right? Okay, that's it. Then move into 174. 174, right? Okay, correct statement regarding the lymphatic system and its basic structure, right? The composition of lymph is similar to that of interstitial fluid. That is correct, right? The composition of lymph is similar to that of the interstitial fluid. So lymph and interstitial fluid both are same, right? Both are same, right? Both are same. So they are similar. Okay. හරි. මේ right. Then second one. Valves so that is correct. First one is first one already correct, right? Then second one, valves cannot be found in lymph vessels. So in lymph vessels, valves can be found. Valves are found, right? So therefore it is incorrect. Valves found in lymph vessels as well as in others. The valves are found in valves found in lymph vessels, right? Okay. Large lymph vessels are located very close to the capillary networks. Now, very close to the capillary networks found are the not the large lymph vessels. They are the small lymph vessels. Small lymph vessels are found closer to the what capillary network. So therefore, this one is also incorrect. Fourth one, lymph is moved by rhythmic contraction of wall of blood vessels and skeletal muscle contractions, right? Right. So lymph is moved by rhythmical contraction of walls of lymph vessels, not walls of blood vessels, right? So lymph is moved by contraction of walls of lymph vessels. Lymph vessels, right? Then fifth one, right? Lymph is drained into two veins at the base of the neck via two small ducts, right? Lymph is drained into two veins, right? Lymph is drained into two veins at the base of the neck via two small ducts, right? Okay. So two large lymph ducts actually, so lymph is drained into two, not two veins, right? So then what is that? Two large lymph ducts. So then lymph is drained into two veins at the base of neck via two small ducts. That is the mistake there. Not two small ducts, right? Two large lymph ducts are drained into the veins. So not the small ducts, right? So they are large ducts, right? Large ducts are drained into the veins, right? Into the veins, right? Okay. So therefore that is incorrect. Then first one is the answer. 175, right? Select the correct statement, right? The AV node is located, the AV node is located between the left and right ventricular walls. The AV node is located between the left and right ventricular walls. Now select the correct statement. Now look at the AV node is located between the left and right ventricular walls. Now you see, now you have to go through the structure of the heart. Now if we take the heart like this, right? Okay. So then if we now here, right? Now this is left atrium, this is right atrium, this is left ventricle, this is right ventricle. That's all, right? So AV node or the AV node, then here like this, and the right, sorry, these are no AV node, no, very sorry. I just note that this is, my mind says that they are the tricuspid and bicuspid. Very sorry. It's regarding the conductive system, no, sorry, right? The AV node, now AV node, right? Correct, regarding the conducting system. AV node is located between the left and right ventricular walls. Now, first one. So AV node is located between the ventricular walls, not left and right ventricular walls, right? Okay. So what is the mistake? The AV node is located between the left and right ventricular walls, right? So now if we take take the heart like this, right? Now like this, no, now it is separated atria walls, then ventricular walls like this, no, separated. Now SA node is like there in the opening of the superior and the inferior vena cava opening, and there you can see the atrial septum there. AV node is on the, not the ventricular, right? Okay. Left and right atria walls, right? Okay. So that is incorrect, between the atria walls, not the ventricular walls. So AV node is located between the left and right ventricle. The AV bundle enters through the fibrous ring that separate the atria and ventricles at the upper end of the ventricular septum and branches into left and right. That is correct. The AV bundle is a mass of cells, right? So now just okay, get into the lesson quickly, right? Okay, don't just yeah, the AV node, yeah, it is between the atrial walls, right? It is between the atrial walls. That's the AV node. That's what I told, right? This is the SA node. I just thought that are the bicuspid and tricuspid were also in my mind. Really sorry. That's why I draw the square heart. So this is the SA node. So this is the AV node, right? AV node is in between the atria walls, right? Right? AV nodes are in the AV node is in the atrial walls, right? Okay, got it. Fine. So then between the ventricular walls, no, left and right, atrial walls, right? Okay, got it. So this is incorrect. Then the AV bundle enters through the fibrous ring that separate the atria and ventricle at the upper end of the ventricular septum and branches into left and right. Now here it enters through like this and goes through the septum. So it is goes through the atria and right through the ventricular septum, right? That fibrous ring that separate the atria and ventricles, right? So that is correct. The AV bundle is a mass of cells. That is also correct. The AV bundle rises from the AV node. So when the AV node is like this, then it is going like that, and it is divided as left and right. Then left and right, then again divide, then goes through the other side. It means here one comes with left side goes to the right side goes to the right side of the heart. Left side goes to the left side of the heart likewise. Okay. So that is correct. The AV bundle branches are separated into thin fibers at the ventricular myocardium. That is correct, right? Okay. Now if I draw it once again with the clear representation, okay, I can draw it like this. Now here I will get the heart like this. Then this is the heart. This is the heart. Now here is the right atria. This is the left atria. This is the right ventricle. This is the left ventricle like this. Now I will draw this very easy. The SA node is here. Then AV node is kind of this kind of a location. Then then the AV node get into here like this, and in the ventricular septum it's divided into left and right and goes through the ventricles, right? Just like this. So then it forms fibers, that Purkinje fibers all the way, and goes through the right side the right fibers and left side the left fibers. That is what happening. Okay. So the AV bundle branches are separated into thin fibers as this is the ventricular, separated into left and right at the ventricular myocardium. That is correct, right? That is correct. Okay. So then fifth one is also correct. So there are four correct statement here. Second, third, fourth, and fifth. Right? Okay. Right. Then 176. The correct structure function combinations are given in, right? The correct structure and function combinations are given in. We now see hypothalamus, midbrain, medulla oblongata, and cerebellum. They regulate appetite. Those are given in order. So when come to the 176, right, we'll see hypothalamus regulates the appetite. That is correct, right? Then midbrain, coordination of the auditory and visual reflex. That is also correct, right? That is also correct. Then BQ. Then C with R, medulla oblongata, control reflex actions. That is correct. Then cerebellum maintain the posture and balance. Actually, all are matching. That AP, then BQ, then CR, and then DS. These are the answers. So answer is fourth one. No. Then 177. Correct regarding the innate immunity. There is a specific type of defense, right? So innate immunity is non-specific, right? It is non-specific. It is non-specific. So here pathogens are identified based on the characteristics that are common to the group of pathogens on which their defensive response are based on. That is correct. They are commonly found, right? They are commonly found, understand. No any specific understanding or specific finding, right? Shows slow response against foreign invasions. No, they are showing rapid response, so the quick responses, right? The activation of defense mechanism depend on the type of invader. The activation of defense mechanisms are depend on the type of the invader. No, they are not depending. Mechanisms are not depend on the type of invader, right? They are not depending on not depending on type of invader, right? Okay. Then can only be seen in vertebrates. Fifth one, right? Fifth one can only be seen in vertebrates. So that is incorrect. This is found in both. Can be found in vertebrates as well as can be found in invertebrates. Right? Okay. So answer is number 2. What is that AV? Here AV bundle, no? Right. AV bundle is a mass of cells. No, it is AV node. So therefore, third one is incorrect, right? Thank you very much. Third one is incorrect. AV node is the mass of cells. Okay, right. Thank you very much. Yeah. Yeah, here now, once again, I will, there's a issue now. The screen went off anyway. The AV node is located between the left and right atrial walls. That is, so therefore, this is incorrect, no? Not 8 ventricular walls, that is atrial walls. Then AV, that is correct. So this is incorrect. The AV bundle is a mass of cells. Mass of cell is the AV node actually. The bundle is a fiber, no? Mass of five, they are fibers, no? The AV bundle rises from AV node. That is correct. The AV bundle branches are separated into thin fibers at ventricular margin. Then 2, 4, and 5. No, not third one. Okay, right. Who is Herat? Is it fine now? Is it fine now, Herat? Right. Okay. Thank you very much. Okay. Then 178. Correct regarding spermatogenesis, a process which takes 6 weeks from start to finish. This takes not 6 weeks, it takes 7 weeks. It takes 7 weeks, right? Okay. Then this is the process of producing not female gametes, it's the process of producing male gametes, right? It's the process of producing male gametes, right? Occurs within the seminiferous tubules in epididymis, right? It is occurs in the seminiferous tubule, not in epididymis, it is in the testis, right? Hundreds of sperms are produced by spermatogenesis within 1 day, not hundreds, millions of sperms, right? What to do with hundreds of sperms, right? Millions of sperms, right? So produce sperms from primordial cells through a continuous sequence of process, that is true. Therefore, fifth one is the answer. Right. Then we'll move on to the 179. Correct statement regarding ovaries, no? Right? Okay. They are held in place within the abdominal cavity using ligaments, right? So 179, first one. They are held in the place within the abdominal cavity using ligaments, right? Okay. So what about the ovaries, right? Held in place, yeah, that is correct. Within the abdominal cavity using ligaments, that is correct, no? Right. They develop after ovulation, right? They develop before ovulation, no? They develop before ovulation, no? They are held in place within the abdominal cavity using ligaments, that is correct. Okay. Uh, right. Okay. Right. Then, okay. Ovarian follicles in different stages of maturity can be seen in the inner layer. Ovarian follicles in different stages of maturity can be seen in the, not in the inner layer, seen in the outer layer, right? Outer layer, okay. Connective tissues are present in the outer layer, which are covered by germinal epithelium, right? Connective tissues are present in the outer layer, which are covered by germinal epithelium. That is also correct. Egg cells can be seen only in some follicles. No, egg cells can be seen. Egg cells are seen in actually in not some follicles. All the follicles, no, right? All the follicles. So therefore, this is also incorrect. This is correct, and first one correct. 1 and 4 are the answers, right? 180. Steps of the sliding filament theory. What are the steps there? First, myosin head binds to ATP. That's the first one. Then, formation of cross bridges by binding myosin heads to actin. Second one. Formation of cross bridges by binding of myosin head to actin. Then third one. E, then B, then D. Fourth one is the reaching of low energy state, no? The fourth one, finally, myosin head detached from actin filaments by binding of a new ATP molecule. This is found in the first one, no? Answer is first one. Very easy one, right? Sliding filament theory, look forward for an essay, right? Might be given in as a essay. Right? 181. Correct statement regarding human sex-linked characteristics. Red-linked color blindness is not a Y-linked recessive disorder. It is a X-linked recessive, right? It is X-linked, right? Then 2. Hemophilia is an X-linked recessive disorder. That is correct. Hemophilia, right? In hemophilia, one of the proteins required for blood clotting are lost, right? We can say that one of the proteins, one or several proteins, right? One or several proteins, right? So therefore, you can say it's one protein, one or several proteins, right? Apart from sex genes, X chromosomes also carry minority of other types of genes. The X chromosome carry larger, means majority of the other types of genes also. But the minority of other types of genes are carried by Y, not by the X, right? Not by the X, right? By Y. It is related for Y. So apart from sex genes, Y chromosomes also carries a minority of other types of genes. Y chromosomes carry sexual characteristics as well as other characteristics that do not affect the sex of the individual, as well as other characteristics. Now regarding Y, it's carry just minority, no. So the sexual characteristics as well as other characteristics, those are carried out by not by the Y. It is by X. It is by X, no. Therefore, this is also incorrect. Then answer for the 181 is second. 182. Select the correct statement out of the following. Only genetic activation take place in epigenetics, right? No, not only activation and deactivation, right? Activation and deactivation both take place in, right, epigenetics. Activation is called as methylation. Then deactivation is known as demethylation, no, right? Only genetics inactivation take place in epigenetics. That is also incorrect because I now explain both take place. Activation is known as methylation. It means methylation. Methylation means adding of a CH3 group, no. Demethylation means removing of a methyl group, no, CH3. Epigenetics can be a result of signals caused by environmental factors. Yeah, that is correct. Okay, that is correct. Environmental factors can be a reason. Epigenetics are never reversible. No, they can be reversible, right? Can be reversed based on the condition. Can be reversed, right? Due to the expression of two DNA sequence, one among identical twins may show genetic disorder. Due to the expression of due to the expression of the same DNA sequences, right? Due to the expression of not two DNA sequences, due to the different expression of the same DNA. So not two DNA, they both having the same DNA sequence, both having the same. I will erase it like this. Both have the same DNA sequence, right? Both have the same DNA sequence, right? But it is expressed in two manner. For one in A manner, for other one in B manner, likewise, right? For other one in B manner, expressed in two manners, right? That is the reason for epigenetics, right? That is the reason for epigenetics, okay? Got it? That is the reason for epigenetics. Okay. Then answer is 3. 183. Correct statement regarding the packaging of prokaryotic DNA. Right? The diameter of supercoiled chromosome is micrometer 30. No, the diameter of the supercoiled chromosome is 2 micrometers, right? That is 2 micrometers. Okay. That is 2 micrometers. So therefore, that is incorrect first one. Chromosomal DNA as well as extra chromosomal genetic materials are cyclic DNA that are supercoiled. Yeah, that is regarding the packaging of prokaryotic DNA. No? So therefore, this statement is correct. Chromosomes cannot be loosened again once they become supercoiled. No, they can be loosened. They can be again when the when we require, we can loosen them. When we require, we can again right. Then again we can arrange them. Okay, got it, right? Rearrange them or loosen. Then again we can tighten them. Likewise, the independency of chromosomal loop is lost when the RNA protein core is removed. Yeah, when the RNA protein core is removed, it means RNA protein core means now like DNA are bound into kind of a core in the middle, no? Kind of a core in the middle like this, no? There's a kind of a core, right? There's a kind of a core like this. So this is the RNA protein core, right? So when RNA protein core is removed, what happens? This tends to again reform, no? Then this will, now imagine this core is not there, then what will happen? This again become a single molecule, no? Single circular one. So that is correct. That statement is correct, right? Chromosomes are, if this is not, if this is cut down, then what happens? This turn into circle once again. Those the main the structure or the arrangement is the structure is maintained by this RNA protein core. This is called the RNA protein core. This is called the RNA protein core. Got it? Right. Chromosomes are only attached to the RNA protein core. No, they are attached to the membrane also. So chromosome is attached. Now, if imagine this is the membrane, then what happens? Now RNA protein core is like this, and the rest of the chromosome is attached to the membrane. Now this is DNA. DNA are like this and also attached to the membrane, right? So that is also correct. Then they are also attached to the membrane. Attached to cell membrane. It is in the plasma membrane, right? Can be loosened. Right, this is the point. Then second one and the fourth one are correct. Fifth one is incorrect. There are two answers, 2 and 4, right? Hundred and 184, right? Correct statement regarding ecosystems in Sri Lanka. Okay. Tropical montane forests. Okay. Tall canopy, subcanopy, and thin shrub layer that is found in the what? In the not in the tropical montane forest. They are found in the tropical rainforest. No, no layering in that montane forest, right? No layering, right? In montane forests, no layering like that. Okay, no layering is there. Tropical wet lowland rainforest subcanopy is about, no, that is incorrect. That's not regarding tropical wet lowland forest. Subcanopy, no. Then tropical dry means evergreen forest, non dense canopy, tall canopy, and well developed shrub layer, right? Okay. So that is correct regarding the tropical dry mix evergreen forest. Tropical thorn shrubs, larger plants are dominant vegetation that consists of thorn shrubs, right? Large plants are not the dominant one. They are not dominant in tropical thorn shrubs. Right? Savanna, easily flammable plants, right? Right? Uh, okay. No, not related to savanna. Okay, right? Got it. Then this is the for the 184. Right. In savanna, there are fire resistant plants, right? So fire resistant plants, you have to write. Fire resistant plants are found there. And tropical thorn scrubs, large plants are not dominant. They are not dominant, right? Small plants are dominant, no? Right. Then 185. Then biome is tested here. So largest biome is northern conifers, no? So northern coniferous, right, is the biome. Then burrowing mammals and grazers, that is temperate grasslands. Then plants with needle-like leaves, that is northern coniferous once again. Then highest rainfall, tropical rainforests. Then largest temperature difference is found in deserts. So answer is number 2, no? Right? These kind of questions you need to be solved like this manner. Okay. Then 186, right? So in the above food chain, it was determined via calculation that 10 joule of energy was transferred to the eagle. Now eagle getting 10 joules, it means snake getting 100 joules, frog getting 1000 joules. These are very simple points. Drosophila getting 10,000 joules, grass having 100,000 joules, right? So the energy spent towards heat and respiration and the energy transferred to the next trophic level. Now energy spent towards heat and respiration means that energy loss, no? Then here energy loss is what? 90,000 joules, no? Right, here the energy loss is 9,000 joules, here the energy loss is 900 joules, right? Here the energy loss is 90 joules, right? Okay. So then energy transfer to the next level is given in this one, no? Right? So 1, 2, 3 and respectively. In one, energy loss is 9,000, 90,000, then energy transferred is 1,000. Then 10,000, right? Answer is 5, no? 90, 90 and 10,000. Then 9,000 and 1,000, 900 and 100, then 90 and 10. Then fifth one is the answer for 186, right? Right, okay. Then moving into 187. Now we'll see, right? A type of envelope virus, no? Envelope virus, there are the herpes simplex one, no? Answer number 5. So when we go to the rabies virus, it is a helical virus. You have to write the type helical. Then adenovirus, it is a polyhedral virus. Polyhedral, right? Polyhedral, polyhedral or icosahedral. It means polyhedral, under that icosahedral. Then tobacco mosaic virus. The tobacco mosaic virus, right? It is helical. Then bacteriophage virus. Bacteriophage virus, that is complex. Right. So therefore answer is 5. 488. Right. Bacteria that produce auxin, cytokine, and gibberellin plant hormones are respectively given by. Now, we'll see what is the answer. Now, auxin is by Pseudomonas fotida, cytokine is by Rhizobium species, and gibberellin by Acetobacter. No? Answer is fourth one. For the 188, answer is fourth one. Then 189, not an organism that contribute to nitrogen fixation, no? Right? So, Pseudomonas involved in what? It is involved in denitrification. It is involved in denitrification, no? So these are incorrect. These all contribute in, they all contribute in what? Nitrogen fixation. Right. Then 190. Right. The species that is an example for a runner. So runner is Chlorophytum chromosome. Chlorophytum chromosome is runner. Then when come to alocasia, alocasia comes under cones, right? Comes under combs. It's a comb. Then gladiolus also comes under combs. Then Cynodon grass, it's come under stolons. Then stachys comes under stolons. Right. Then we'll move on to 191, right? 191. Then okay, 191. Protists that possess a multicellular thallus and hold fast, right? Okay. So ulva possess of a multicellular thallus and hold fast, then gelidium, and then sargassum. But here, what is the error? Sargassum, there is no such organism like that. There's a spelling mistake. Therefore, that cannot be taken as answer. Then sargassum correct, diatoms incorrect. So answer is A, B, D, and answer number 1. Then 42nd. Select the correct response that show macro elements in plants, how they are absorbed and relevant to the deficiency system. Actually, this is a all, right? There's a error in the question. That is not, the element is not given, right? So therefore, you can't find an answer for this. This is all actually, right? All question means no answer because I have the element is missing there, right? Right. Then 193 function of the adipose tissue. Right, then we'll see. Calm. Insulate the body, that is correct. Now this had to be ABCD type question, right? ABCD type question. Act as a filler, that is correct. Transport of material is done by blood or that kind of thing. So therefore not a function of adipose tissue. Protection is not an function of adipose tissue, right? Then strength and no, only A and B. Answer is A and B. It means answer 3. 194 places where mucous membranes can be found is air, right? So in 194 places where mucous membranes can be found, then lining of the respiratory tract, okay. Lining of urinary tract can be found. Lining of alimentary tract, these once again need to be A, B, C, D, right? A, B, C, D, E, right? Okay. So lining of the reproductive tract, that is correct. Lining of the anus, not the anus, that lining of the anal tract, right? Lining of the anal tract, okay. So that is not the anus. Lining of the, it have to be anal tract. If it is to be correct, it has to be anal tract, right? Not anus. So therefore A, B, C, D, and answer is 5, right? Then come to 195. It has to be A, B, C, D equation once again, right? It has to be A, B, C, D. The hormones to which the heart respond, right? So in the resource book, they are mentioning adrenaline and thyroxine, right? If cortisol also affecting the heart response, but you have to follow the resource book. Cortisol is also correct, right? This is also correct. But these two are given in the resource book, right? Therefore, C and D, it means answer 4, right? Then we come to 196 here. The adverse effects of the addictive substance nicotine in cigarette smoke, right? So nicotine, these are once again A, B, C, D type question. This is A, this is B, this is C, this is D, and this is E, right? This is C. A temporary increase the rate of heartbeat, that is correct, right? Okay. Reduce the oxygen transport, that is done by carbon monoxide, not the nicotine. So then constriction of the peripheral blood vessels, that is correct. Temporary raises blood pressure, that is correct. Inhibit the action of cilia, that is done by the smoke, right? Not by nicotine. Therefore, that is incorrect. Then A, C, D, answer number 2, right? 197. Correct statement or statement regarding Hardy-Weinberg, no? Right? The Hardy-Weinberg approach is described based on the evolving hypothetical population. The earlier today earlier in discussion, we also discussed that it is not relevant in a evolving hypothetical population. So the population need to be non-evolved, no, not evolving. So therefore, the population need to be not evolving, right? Then this also need to be A, this is C, this is D, this is E, this is C. Then B, a population deviates from the Hardy-Weinberg equilibrium if it is too small. Yeah, that is correct, because the population need to be very large, no, right? Gene flow contributes, no, gene flow is not contributing, gene flow is adversely affecting. Gene flow is adversely affecting towards Hardy-Weinberg equilibrium, right? Okay. So this is also incorrect. Then most natural populations adhere to the Hardy-Weinberg equilibrium, right? Do not adhere, right? Most natural populations do not adhere because they are immigration take place, natural selection. So they don't adhere, right? They don't adhere. Slowly evolving population deviate greatly from the Hardy. No, slowly evolving populations, they are not deviate greatly, right? They do not deviate greatly. They don't deviate greatly because changes are minor. No, don't deviate greatly. The reason is changes are minor. Changes are minor. That's the reason, right? Changes are minor. That's the reason. Okay, changes are minor. Okay. Now 197 is done. Okay. This is incorrect. Therefore, answer is only 5. B, the answer is 5. Then 198. The physical changes that take place in food, they are the softening of food. This again need to be A, B, C, D, right? A, B, C, D, and E, right? A, then pigmentation. B, putrefaction is a chemical. It's a chemical change. Accumulation of toxin is a physical. Fermentation is also a chemical change, right? Okay, therefore answers are A, B, D, and answer number 1, right? Then 199, examples for endangered species, vulnerable species, Sri Lankan endemic plants, Sri Lankan endemic animals, and invasive alien species, right? So here, what is example now? A, B, C, D, E. These are the statements. We'll see. In A, elephant or endangered species, right? Buttercup, vulnerable, right? Sri Lankan endemic Garcinia quercita, that is correct. Sri Lankan endemic animal, Loris tardigratus, that is correct. Invasive alien, Lantana camera, that is also, sorry, that is also correct. Vesak orchid, buttercup, right? Vesak orchid is endangered, yeah, vulnerable buttercup. Sri Lankan endemic plant, Dipterocarp selingus, right? Puntius nigrofascicatus, Sri Lankan endemic animal. Yes, Pansigum macticus, yeah, invasive Pancum maxium, invasive alien species. Then dusky striped squirrel, buttercup. No, you can't take that. Elephant, no. Elephant is not an endemic species. Elephant is a endangered species. Here's a error. Then Vesak orchid, then elephant, dusky striped squirrel is not an endemic one, right? Dusky striped squirrel is not endemic, so therefore it is incorrect. Elephant, Vesak orchid, Pancum maximum, Loris tardigratus is not an kind of invasive alien, so therefore this is incorrect. Then A and B, answer is 3. Right. 200 processes that happen during post-harvest loss are. Processes that happen during post-harvest loss are. So we'll see process that happens during post-harvest loss. So what is that? Then come to the question number 200. Loss of mass due to spoilage, yeah, kind of a post-harvest loss. Loss of quality, this is also a post-harvest loss. Loss of nutrition is a post-harvest loss. Loss of seed viability is a post-harvest loss. Economical loss is a post-harvest loss. Then A, B, C, D, E, all are correct. Then answer is 5. Okay. Now, students, now okay up to 200 MCQs. Are you okay? Drop a message. Are you okay? Drop a message. Are you okay with the 200 MCQs? Okay, right. Nice. Then we'll stop it there, right? Okay, we'll stop it there. There is some sort of a issue there, right? Okay. I tried, the issue was there from I think just after the lunch interval that I gave you regarding that those the sessions which we are planning to do, right? That sessions we plan to do on the coming days. There's a huge issue and it has gone to the its upper level, right? Even for the ministry level, it has gone, right? The timetable. Uh, right? Okay, I think I don't think I let you realize that there is an issue, but I did my best. But here and there, I managed with the phone surrounding and just communicate with that, and the team is communicate with the problem. Then now I have to interfere with that, right? Okay, not the saturation. This okay, but I was trying to go and to discuss the 500 all the MCQs, but unfortunately not only this session, the coming sessions, I think really I think they are gone. I think there is no way that we can recover. I think it's almost over. I can't understand how people are like this, no? You tend to tell me that I saw that you also have shared before you. I got to know that our timetable and our process, all those are shared in Telegram discussions, and they are huge kind of a shout out is creating that they are asking that they are telling that that the government not to conclude sessions, but this guy is concluding sessions. Even this guy has posted timetables. That's how Sri Lankan people are behaving. No, but don't be frustrated. I think frustration surely there should be for students who didn't came to our classes, right? Because our class students, in as our class, we have done guest sessions, pilot papers, all those things. We are saturated with the efforts of the class. So, and I have given you the necessary for the we have given essay books, then CQs and all those things. So you have nothing to worry. You just need to study. I think the most the disadvantage is for the people, right? The other people, those who are actually majority, those who are there to be benefited. But I don't know. But with the behavior of them, that has become maybe a miracle, okay, or maybe a kind of a dream. Okay, anyway, we'll see. We'll see. But anyway, right? Anyway, I made my effort, and I made my effort totally on today's discussion. 200 MCQs done. I think it's the best. But anyway, we'll have to get a meeting. We have to take a meeting in a while. We'll inform that to you. And we are taking a meeting, right? Sudden meeting. We'll inform you quickly, right? Through the groups. Okay. Then till then, thank you. Take the use of this session, right? Okay. Have a nice day.